we don't need the two price with the two.
C'è il ladro.
Ecco, l'unica cosa che cosa mi devo recuperare se non lo vado a recuperare.
Is this church used for many meetings, or it's unusual?
It's a site of the university, the science faculty.
Okay.
It's
Amazing.
amazing. Yes. You always use this site for the meeting because it's well.
Amazing.
Mm-hmm.
Maybe another minute.
Yes. Aspettiamo qualche altro minuto
sì,
perché
sono
c'è
andato a vedere, c'è
un po' di coda, li facciamo entrare prima.
Poi acceleriamo.
Chi deve moderare? Monnes?
Sì. Giordi Monnes? Sì, certo. È lì.
Sono arrivati tutti gli stranieri.
Qualcuno senza voce, lei senza voce, Alessandro Invernizio senza voce.
Perché il freddo di Milano.
Esattamente.
Vabbè.
Però lui ha già eventualmente sostituito.
Lo può sostituire.
Sì. Qui hanno preparato dei casi che sono vedere
Giovanni Roberto lo sai, è diventato direttore del ha fatto una lavorazione 15 giorni.
Ma infatti qualcuno
Sarebbe bello se insomma questa collaborazione. Lei mi piacerebbe ritornare in casa.
Lei non fa un proposto.
Non fa. Secondo me con Alberto è un'ottima chance perché si sposterà tutto sulle malattie degli occhi. Considerando che non fa la medicina.
Certo, esattamente.
Perché ha ballato. Secondo me non verrebbe forse a là per fare il ritmo.
No, queste qua.
8 genetic therapies for visual restoration. Questo sì, per molto più pazienti si funziona.
Soprattutto perché se ci pensi è l'unico modo di mettere a posto non sembra funzionare
So Angela thinks we can probably start now because it'll take a couple of minutes for us to do the introductions, and there's still people coming, but anytime we want to. She said we can start anytime we want to.
Yeah.
Okay. Good morning and welcome. I'm very happy and honored to greet here in our city today so many important experts that represent the true excellence in the field of retinal diseases. Today, more than ever, there is a need to contribute in solving open questions regarding the treatment of inherited retinal dystrophies and age-related macular degeneration, in particular the atrophic form. Preclinical studies are fundamental to better understand the pathogenetic mechanisms underlying the disease. And this knowledge can help identify possible innovative therapeutic strategies, as is now occurring in the field of inherited retinal dystrophies with gene therapy and partly with new drugs for AMD. I believe that multidisciplinary teamwork can contribute to this aim with different points of view that can provide a broader reading of the phenomena and an enrichment in scientific knowledge. I hope this meeting serves as an incentive, especially for all the young people here today, to continue their research in this field and make their contribution to patient care.
So just to open, that this meeting in a little 2 words for, how it's working. What we want to is the discussion. So you will see that there is a lot of room for asking and, discussing, the topics that will be presented. The Retina update is a meeting that it's rotating every year in Italy, and so we hope that also here in Naples we will be able to bring the idea that the meeting should be not only listening but also discussing and asking to the presenters. Vas?
Thank you very much, Giovanni. Thank you, Francesca. So really grateful to Professor Simonelli for hosting us in this wonderful city, in this incredible venue. Giovanni actually had said that we have to send a reminder to the audience. Please pay attention to the slides. Don't look up at the ceiling, because it's easy to get lost in the beauty of this place. But instead, hopefully you'll get lost in the beauty of all of the advances in retina. Sometimes in our field, there's a joke, well, you can find a retina meeting every weekend if you want to attend one. And that may be actually true, but I will say that one thing about our field is that it's rapidly evolving. So even if you went to a retina meeting every week almost, you might be out of date until you get to the next one. And so that really is our hope in this meeting. I want to thank all of the faculty who are joining us today, really experts in the field. And we'll bring everyone hopefully up to date on the latest in what's happening in terms of therapeutic advances. And of course, many thanks to Angela and her Volpe and her team and AVEC for organizing this meeting. But we will thank them again later on in the meeting. So with that, we should start with the first session.
So we invite Jordi to come here to moderate with Francesca the first session.
Please proceed.
Okay. The first speaker is Professor Banfi from our University Vanvitelli in Naples, Genetic Testing in Inherited Retinal Disease, an update.
Okay. Good morning, everybody. Thanks a lot to the organizer for inviting me to this very, very interesting workshop. So basically what I would like to do today is to give you an overview of what is going on concerning the genetic test, starting from very, maybe, I don't know. Okay, so just starting from some basic and probably redundant information. So what do we mean by genetic diagnosis? Basically the identification of the molecular defect that underlie a genetic disease. And I don't know what's, okay. In the majority of cases, as you know, the molecular defect resides in, basically it's constituted by the change in a single DNA base, in a single letter in the genome. So basically 1 out of 3 billion bases in the haploid genome. So this makes this effort really cumbersome. It's a very difficult process that until 20 years ago, it seemed basically impossible. And, however, in any case, it is very important to it is very important to obtain a genetic diagnosis in all cases because, first of all, it confirms the clinical diagnosis. And we know that many times the genetic diagnosis may have an impact on the final diagnosis. So when the diagnostic information should make an important, play an important role in the final diagnosis. We can, knowing the genetic diagnosis, we can have also information about the prognosis. Not all the time, but in many cases we can. Of course, you can establish the recurrence risk in the family, which is an important information for the patient. And now, becoming more and more important, the possible access, thanks to genetic diagnosis, to possible gene-specific therapy, like in the case of the RPX65 that you will hear in the next presentation. And there are some other that are undergoing clinical trial. So the problem for inherited retinal diseases is that we are facing an impressive genetic heterogeneity with about 300 causative genes. There is high genetic heterogeneity, high clinical heterogeneity. The same gene, mutation in the same gene can cause different conditions. So this makes the diagnosis really, the genetic diagnosis really difficult. So in any case, there's been a lot of progress in this field. So we can, if we go back, if we look back, we see that there is, we can talk about genetic diagnosis before 2005, 2010, before first next generation sequencing, when we were using first generation procedure, PCR, Sanger sequencing, and other technique. And in those years, the diagnosis of such a highly genetic heterogeneous disease, such as inherited retinal disease, was very difficult. In the best scenario, this is an example of a study that we performed in collaboration with Professor Simonelli almost 20 years ago. You could reach a genetic diagnosis in no more than 30% of cases. And this was a really fortunate event. And of course, the number of genes that you find mutated was very limited. Now, with after 2005, with the availability of next-generation sequencing, which is a technology, a revolutionary technology that allows the sequencing of an incredibly high number of genes, or even the whole genome, you can now, all these figures have been changed. Of course, you don't need to sequence the whole genome when you use next-generation sequencing, when you want to diagnose inherited retinal disease. You can start from targeted sequencing, so the sequencing can be restricted to a number of genes. Maybe in this case, for IRD, you can decide to analyze only the genes already known to be responsible for this condition, or you can go for the whole exome sequencing, or WES, in which you sequence all coding elements of the genome, so basically all protein coding genes. Or at the extreme, you can do all genome sequencing, so you can sequence the entire genome, and this will be the most detailed and complete analysis. But in for diagnostic purposes, we are not using yet whole genome sequencing. Either we use targeted sequencing, or we use all exome sequencing. And with the NGS, now the figure, the effectiveness of the genetic diagnosis has been completely changed. Now what we see here is that we can solve about 60 to 70% of all patients. We can reach the genetic diagnosis in inherited retinal disease patients in about 60 or 70% of patients. Here I want to show a study that we performed in collaboration with Professor Simonelli on about over 3,000 Italian patients with inherited retinal diseases. And this clinical cohort of 3,500 patients are estimated to cover about 20% of all cases in Italy, and even more than 40% in southern Italy. Out of these 3,500, 2,800 were analyzed by NGS, either panels or whole exome. and we obtain a diagnostic success rate of about over 70%, and we identify mutation in 132 genes. Results of this study are in line with what already reported in other countries. And so this is a study that was published 1 or 2 years ago. This point, however, so we are very happy about this result, but still we notice that there are about 20 to 30% of cases that remain unsolved. This is the challenge to be solved in the near future. Why these cases remain unsolved? And we have, here I indicated 4 possible causes. I will give you brief, short example of one of each of these cases, apart from the 4th one. So we can distinguish these 4 possible reasons. First of all, the variants that cause the disease in this undiagnosed patient is a variant that we cannot detect by NGS. NGS is very good in detecting point mutation in protein, point mutation, in particular in protein coding genes. But what happens is sometimes the mutation can be present in not an exonic region, in an intronic region, a region that normally, unless you sequence the whole genome, you will not sequence. And this deep intronic variant may cause an alteration of splicing. So these are variants that are frequently missed by using standard NGS techniques. Or maybe there can be other kind of variants, structural variants, small deletion and duplication, or there are some regions, some genes responsible for IRD, like or 15 RPGR. RPGR is a very frequent cause of retinitis pigmentosa, but yet the region that contains the majority of mutation is very hard to be sequenced using standard technique. So taking all this information into account, how is it possible to increase the sensitivity of diagnosis for inherited retinal disease? There are several possibilities. One is to do whole genome sequencing, which is good, but not the final solution, because we need to interpret then the meaning of the possible intronic variant that we identify. We can perform, instead of doing analysis on DNA, we can do analysis on RNA, but here we face the problem that many of the genes responsible for this condition are not expressed in easily accessible tissue, such as blood. But most of the cases, we need to do functional validation of the variant that we identified, or we need to consider also RPGR or 15 screening. So basically, this, just to tell you that to find additional variants that are in region that escape detection by NGS, this effort is mostly focused on research effort, not diagnostic. So there are still labs working hard to solve this issue. And here I just want to show an example of a study performed in the laboratory of Professor Franz Kremers in Nijmegen, that if in the case of the gene ABCA4 that is well known to have a significant number of mutations that are in the intronic region, he performed a very detailed analysis. He sequenced the entire gene in all the patients, and then he systematically carried out functional tests for each of these intronic variants to see whether they could alter splicing. So this gives us an example of the effort, the strong research effort that we still need to undertake in order to solve this issue. Then the second possible reason for undetectable mutation is that we find the mutation, but we cannot interpret correctly this mutation. So basically, these are the so-called VUS, variants of uncertain significance. In this case, again, it is necessary to perform functional studies to solve this problem. But we can do something very simple sometime to simplify this work. So let's assume that you find 2 variants of unknown significance in the RP65 gene, and the variant that that have not been described so far. The first thing that you can do, and this is very easy, that doesn't take research effort, is to look for the segregation in the family. So you find the mutation in the proband, in the patient, you analyze the parent. What you would expect to see is that these 2 variants are present, one in the mother and one in the father. In this case, if it is the case, then the pathogenicity is compatible. If instead you find that the 2 variants are only present in one of the parents, you can exclude this result as pathogenic. So this is something very simple to do. So this again invites also the clinician to collect the blood, the DNA of the parents as soon as possible, because this can be very helpful for the diagnosis. Third and last aspect I want to share with you, sometimes you have unexpected genotype-phenotype correlation. So you find mutation in one gene that is not associated with phenotype that are compatible with what you see in the patient that you are analyzing. So the solution here is to be open-minded. Not to think, okay, so if this patient has Usher syndrome, then we can analyze only the genes that are already been shown to be mutated in Usher syndrome. So don't use too restricted panel. Just I want to give you the last example, two families that we have recently analyzed, two families that had the diagnosis of Usher type 2. We performed exome sequencing, and we found mutation in this gene, AGBL5. This gene, AGBL5, was reported so far to be causing only isolated RP. But this was very strong mutation. At this point, another aspect that we need to implement is it is necessary to collaborate with with international centers, consortia. So we asked this consortium of genetic labs, European Retinal Disease Consortium, whether they found similar results in their cohort of patients. And indeed, there were five more families with this finding. So mutation in AGBL5 associated with RP and inherited hearing impairment. So at this point, we concluded that AGBL5 is a novel gene responsible for Usher-like, let's say, RP and the deafness phenotype. So in conclusion, the NGS led to a significant progress in the genetic diagnosis of IRDs. Still a significant fraction of patients who remain undiagnosed. The analysis of family members is essential to finalize diagnosis. The resolution of the undiagnosed patient require a multi-step procedure involving collaboration, research effort, and data sharing and cooperation with other groups will also be crucial. And with this, I finish by thanking all the collaborators, particularly Francesca Simonelli, all the other people involved in this genetic analysis. And I thank you for your attention.
Thank you very much. I will proceed with the next one with Francesca Simonelli on update on RP65 gene therapy.
I will speak you about our experience in real life with RP65 gene therapy. These are my financial disclosures. RP65 mutation-associated inherited retinal dystrophies are the first ocular diseases treated with gene therapy. We treated 29 patients in our eye clinic in Naples from 2020 and today, and you can see they came from different regions of Italy. I will show you the results in the first 12 treated patients who reached 1 year post-treatment follow-up. The age of patients was from 7 to 35 years. They had early onset retinitis pigmentosa or Leber congenital amaurosis, and all patients showed variants in both alleles of RP65 gene. We performed best-corrected visual acuity, semi-automatic kinetic visual field, full field sensitivity threshold, FST, chromatic pupillometry, and color fundus imaging at baseline, 1 month, 6 months, 12 months post-treatment follow-up. We found a statistically significant improvement in best-corrected visual acuity 1 year post-treatment. We also found a statistically significant improvement, enlargement of kinetical visual field 1 year post-treatment, in particular with 1,4-E and 3,4-E stimulus sites. We performed full field stimulus thresholds with 3 different stimuli, white, blue, and red, associated with the response of the photoreceptors, blue rods, red cones, and white mixed response. And you can see that we observed a statistical significant improvement in white, blue, and red FCT 1 year after the treatment. But these 3 tests are subjective tests, so they can be influenced by the collaboration of the patients, in particular in pediatric age. So we decided to introduce an objective test to evaluate in real life the response to the treatment, such as chromatic pupillometry, using the same stimuli of FCT, blue, white, and red. And you can see we found a statistically significant improvement in 10 lux blue and 10 lux white maximum pupillary constriction, confirming the data of the subjective tests. We correlated the results of FST and chromatic pupillometry, and we found a statistically significant correlation between the 2 tests. But we had some adverse events, such as chororetinal atrophy. We considered 25 patients treated with at least 12-month post-treatment follow-up, 41 eyes, 16 patients treated in both eyes, and 9 out of these 25 patients, 36% showed chororetinal atrophy about 6 months after treatment in both eyes. We observed three different patterns. Nummular pattern, predominantly involving in the periphery, is observed in 8 patients in both eyes. Mixed pattern of chororetinal atrophy, nummular and peripheral atrophy, observed in 1 patient. And focal atrophy at the injection site, so-called touchdown atrophy, is observed in 6 patients. And we found an enlargement of area in all patients with nummular and mixed atrophy from a 6-month to 12-month follow-up visit. We found no further enlargement in any patients with touchdown atrophy. So we collected the demographic and clinical features at the baseline in our patients, and we subdivided the patients in a group with CRA, 9 patients, and a group without CRA, 16 patients. And we observed that the younger patients and those with liver congenital amaurosis had more frequent chororetinal atrophy. We also collected the functional and morphological clinical features at baseline in the 2 groups of patients. in eyes with the CRA, 18, and in eyes without the CRA, 23. And we found no statistical significant differences in visual function, such as visual acuity, visual field, FCT parameters between the two groups at baseline. We found that eyes with disrupted EZ band at baseline are more likely to develop CRA compared to eyes with absent EZ band at baseline. The question is, can the onset of atrophy influence the treatment effects? We evaluated the treatment effects in two groups, in eyes without and with CRA. And we observed a statistically significant improvement in all parameters in patients without CRA, and a statistically significant improvement in all parameters except visual field in patients with CRA, probably due to the localization in mid-periphery of the chororetinal atrophies. But we need to collect other data to evaluate the consistency of these preliminary results over time. In conclusion, our results showed the improvement in visual function in patients treated in real life with vorticiguat neparvovec, prevalently observed by subjective testing and further confirmed by an objective test such as chromatic pupillometry. We need more data to understand the causes of CRA and their impact on visual function over time. These are our recent references on the topic. And I would like to thank my team for the great and passionate work. Thank you. Professor Moniz, optogenetic therapist for visual restoration.
Morning, thank you for inviting me. It's my pleasure to be here in Naples. And it's my pleasure also to discuss optogenetic strategies for retinal disease. Disease could be some sort of science fiction because with this technology, it's not preventing loss of vision, it's restoring vision. And optogenetics has been in the field for a while, but until recently, there were not clinically meaningful results. So these are my disclosures, mostly related to AMD. So in inherited retinal diseases, we lose the detector of light, photoreceptors are gone, and those patients have very low vision. There are no therapies. The main inherited retinal diseases are retinitis pigmentosa, a Stargardt disease. As we already heard, there are many different genotypes and phenotypes of retinitis pigmentosa. So finding a therapy that would be agnostic for the gene that causes the disease would be ideal, and not to do a specific cause by the gene origin. These patients have very low vision, and ETRS do not work. ETRS do not work worse than logMAR 1.9, and those patients are mainly in this range. So for testing these people, we need to use the Freiberg acuity test. That works for that very low level of vision, and changes on 0.3 logMARs are considered clinically significant. They would correspond to a 15-letter change. Also, These patients need to be tested by mobility test, and this mobility test specifically, patients have to save some obstacles and going to some lights that are in the other side. And depending on the level of light that they pass those mobility tests, they have this score that goes from -1 to 5. So optogenetics is attempting to improve retinal sensitivity to light. It can transfect ganglion cells or bipolar cells. And the idea is on these retinal diseases, the photoreceptor is gone. And sorry, I lost my screen. But the cable is maintained. The bipolar and ganglion cells is maintained. Only the cable, but not the detector of light. So optogenetics, what it does is making the cable, those cells, to be able to detect light. The targeting bipolar is better than ganglion cells because there are 10 bipolar cells per one ganglion cell. So targeting bipolar, you target more cells. So now the cable that was only transducing the electricity is now able to detect light. So there are different options. This protein that has the capacity of detecting light when you transfect ganglion cells or bipolar cells. So in normal vision, that would be on the left. If the opsin only detects red, you would only see whenever it's the red light. If opsin detects blue, we would not see the traffic light. If the opsins detect the 3 lights, then you would have the signal. So it's very important, the opsins, if it's for red, for blue, or for all. Then the sensitivity is very important. Some opsins do not have enough sensitivity. They would need huge light that is not in normal life. So sensitivity is also very important in opsins. And then the kinetics response. If it's not fast, the images would be blurred. So the kinetics have to be fast too. So opsins have been in the field since 2012, approximately. And the first trial was Allergan. I think that this has been stopped. That was a fast but low sensitivity only to blue. Then Crimson and Kronos and Gensight and Bionic, they're still in phase 1. They are fast, but again, low sensitivity and only to red or blue. And now the most recent, the multi-characteristics opsin, it's high sensitive, it's fast, and it's for all colors. And this is the MCO clinical trial of nanoscope that finished phase 2, and now it's going to enter into phase 3, and those are the results we're going to show. So the first 4 are the ones who've been in clinical humans. The other 3 are preclinical. So of these 4, only nanoscope is targeting bipolar cells. The only one is high sensitive. The 4 were fast. Allergan was stopped, and this one has already given results of phase 2 and entering into phase 3. So MCO is a transgene for high-performing opsin that sensitizes bipolar and uses an B2 virus for the transportation. So the RESTORE phase 2 was a single injection of 2 doses of gene therapy versus sham, and this was 9 and 9 patients. There was some oral steroid prevention therapy. And the primary endpoint was at 52 weeks. And best corrected visual acuity was assessed with a Freiburg's test, the shape discrimination test, differentiating these 3 shapes, and the mobility test, and also a composite of the shape and mobility test. So this study, as we mentioned, enrolled 27 patients, 18 subjects received gene therapy. And the high dose and the low dose had a statistical significant improvement in visual acuity. And also there was a 3 increase, that was not a statistical significant, but 3 increase score in the mobility test. The adverse events were similar across the studies, across their arms, and they were not important. So on the mobility, patients went from this level to the other level. 12 of 18, about 67 at least, improved 2 levels in the treated arms compared to 3 in the sham patients. So a few patients with Stargardt have been also tested, 6 patients. That was a very small trial. And there were improvements in visual acuity, clinically meaningful improvements, not a statistical power for that. And the mean improvement in visual acuity was about 5 letters. And 2.6 decibels improvement in microperimetry. If we have time, I think so, yeah, I will. Do we have audio for this video? That's a testimonial of a Stargardt patient, one of those 6. I don't think the sound is on. Do we have sound?
Watching TV, you know, I know who the characters are by their voice, that, you know, now, like, I watch and I can see their face structure. It's like, and any little bit of vision, I mean, is better than, you know, what I had before.
At the beginning, she was explaining that she was able to detect more colors than before. So this is subjective, this is testimonial, this is not very scientific, but only 6 patients were treated, so we need to get this kind of inputs. So in summary, treating inherited retinal disease and unmet need, very limited treatment, only for RPE65. Optogenetics is an innovative therapy that is agnostic from the genetic cause. It can treat any blind area of the retina. And it's not a therapy that prevents visual loss. It's a therapy that pretends to restore vision in patients who have very, very low vision. And these positive results in phase 2 now have allowed that phase 3 will start shortly. Thank you very much.
Thank you. And I'm pleased to introduce Professor Alberto Auricchio, expanding EV gene therapy in the retina.
Thank you very much, Francesca, and I thank the organizers for this invitation. So I'll tell you in the next 10 minutes some of the things that we have developed in our lab at Teleton Institute of Genetics and Medicine here in Pozzuoli in order to enlarge the patient population that is suffering from inherited retinal diseases that would benefit from in vivo gene therapy. And today, the most effective tool to transfer genes to the retina, and for that matters also to other tissues in vivo, it's based on adeno-associated viral vectors, which are small viruses that are safe and effective in humans. There are 7 approved products that are actually based on AAV, and one of them is actually Luxturna for RPE65 associated inherited retinal dystrophies, as you heard before. So one of the features of AAV that makes it so attractive and so effective as a tool for in vivo gene therapy, it is that it's highly diffusible, which means that it's able to overcome physical barriers in vivo due to its small size. And of course, the small size also comes at the cost of a small packaging capacity, which means that a small virus can only package a certain amount of genetic payload of DNA, which in the case of AAV, it's 5 kilobases. So I'm going to discuss about the efforts that are ongoing in my lab and also in other labs in order to overcome this challenge. And this is a challenge because there are a number of inherited retinal diseases, and you're only seeing a few of them in this table, in which the gene that is mutated that would need to be transferred to photoreceptors or RP cells in the retina, it's actually larger than AAV cargo capacity. So you would need to fit a large gene into a small virus, like a large foot into a small shoe, or a large group of people into a small car. So how do you solve this? So the strategy that we have thought about is that what you cannot fit into a single AV, you actually break it into two pieces, into two halves in terms of genetic payload. And at this point, these two pieces of DNA can fit into a single vector. So if you have a large coding sequence or a large expression cassette, you break it into two, and each of these two halves are independently packaged by a single AV vector. So your drug is actually a mixture of two AV vectors, each transferring one piece of a large expression cassette. And so the first disease on which we started to work testing the efficiency of this platform that can actually be actually declined in different flavors that I don't have the time to go into because of the limited amount of time. But the first disease in which we have tested this dual AV strategy, it's the retinitis pigmentosa associated with Usher 1B. And in this case, the gene that is mutated in patients with Usher 1B, it's called myosin 7A, and it's actually a gene that has a large coding sequence that would not fit into a single AV. And so we developed a set of two AV vectors, each carrying one half of a large expression cassette expressing myosin 7A. And we delivered this mixture of two AV vectors to the subretinal space, initially of mice that have Usher 1B, that have basically deficiency of myosin 7A. And if you look on the left of this busy slide, you'll see that upon subretinal administration of the two vectors, we are able to reconstitute full-length myosin 7A expression in vivo in the retina of mice. And the levels of expression are quite meaningful because with doses of vectors that allometrically scaled to humans would be clinically relevant, we actually are able to reconstitute between 40 and 75% of endogenous myosin 7A levels, which in a recessive condition are expected to be therapeutically significant. And indeed, if we look at one of the features of the phenotype of the mouse that I don't have the time to get into the detail, we see basically a dose-dependent improvement of the feature of this phenotypic aspect of Shaker1 mice phenotype. So these another preclinical data were instrumental to actually apply to European funding and to establish a European consortium coordinated by TGEM and of course Francesca Simonelli was the major clinical partner of this consortium, where we were actually paving the way to a phase 1/2 clinical trial testing the safety and efficacy of dual AV vectors in the retina of patients with Usher 1B. And through this consortium, we were able to basically produce a GMP clinical vector that can be used in the clinical trial. We were able to show in IND-enabling studies in non-human primates that the subretinal injection of this material was well tolerated. And we submitted our CTA in both UK and Italy. And I'm glad actually to announce that the CTA was cleared as of May 15 in UK and is about to be cleared in Italy. So we will hopefully soon be able to move into patients with this innovative approach. That's not the only disease on which we've been working in terms of large gene transfer to the retina with the AV vectors. The other important indications that would benefit from such approach is the Stargardt disease, which is basically the most common inherited macular dystrophy in humans. And again, the gene that is involved in this condition that is inherited as Usher 1B as autosomal recessive, it's a BCA4. And a BCA4 has a large coding sequence that would not fit into a single AV. And so we developed a slightly different dual AV platform than the previous one. And again, unfortunately, I don't have the time to get into much details. But this novel dual AV platform, when delivered to the subretinal space of pigs, and we actually developed an ad hoc model of Stargardt disease, a pig model of Stargardt disease, where there is a knockout of the BCA4 gene, we were able to reconstitute high levels of ABCA4 full-length protein in the retina of these animals. And when we compared this to the previous platform that we've used for Usher1B, it's quite evident that this novel platform that is based on protein transplicing is actually superior to the previous one. And we were also showing that this platform works, this two-vector platform works in human photoreceptors in human retinal organoids developed from Stargardt disease patients' iPS cells. And we have also shown, that, and I don't have the time to show that this approach is effective at reducing lipofuscin accumulation both in mice with ABCA4 knockout and in pigs with ABCA4 knockout. And so both these programs and the technology that is associated with the development of these two products has transitioned from my lab at TGM into a spin-off that's called Avantgarde Bio, that it's now basically progressing this program towards the clinic. And I told you, Usher1B is about to start and the phase 1/2 clinical trial. For Stargardt disease, we need to wait until 2025. And so with this, I would like to finish. I would like to thank actually the people in my lab who have contributed to the development of these two dual AV platforms and the funding, and of course, all of you for your attention. Thank you very much.
Thank you, Alberto. Questions?
On this part?
Okay, so I'll ask my question first, I guess. Then my question is for Professor Riccio. Really great presentation. All four were really great talks. You talked about a dual vector approach. Can you comment about liposomal or non-viral delivery systems, because they can also accomplish this, I think, right?
Sure. So liposomal, so non-viral technology it's a fastly growing field, particularly because genome editing it's fastly developing, and so these non-viral technologies are particularly useful for in that regards, in my view. So the problem with delivering large genes or large mRNAs with liposomes is still that the particle is quite large. So when you think about AV, this is a 20 nanometer particle. There are very few non-viral nanoparticles that are smaller than 100 nanometers. So if you take a 100 nanometer virus, it doesn't get to photoreceptors if you inject it subretinally, mostly because it's a large particle that doesn't diffuse easily through the physical barriers of the subretinal space to the inner segment of photoreceptors. So until we get to small nanoparticles, this is going to be a challenge in the retina. But I think it's just a matter of time. We'll get there.
I was just curious, a great talk, whether you're limited to dual fragments. Can you go higher if you wanted to do larger fragments?
Yeah, we also developed and published triple AV vectors, so you can, of course, I mean, theoretically, you can increase the number of vectors and the packaging capacity or the transfer capacity, DNA transfer capacity. But there are challenges, of course, with this. With the more complex is the system, the more difficult it is to manufacture, and also there could be aberrant products that are produced. Once you increase the number of vectors that you put in your syringe, so.
I have a question for Francesca. Fascinating data that you showed, Francesca, fabulous. So I'm interested by the atrophy that you observed. Now, in terms of the mechanism of atrophy, it does look that although AAVs are considered to be not so immunogenic, that there may well be, because there have been cases of acute hemolytic uremic syndrome in patients who have received AAV therapy in some of the larger systemic diseases that we see. Do you think that it could be complement activation that's causing this patch of geographic type atrophy after AAV therapy with Luxturna?
Thank you for the question. This is a problem. I don't know the answer. I think that in the pathogenesis of chororetinal atrophy
Very nice talk.
can be crucial two factors: the surgery and probably a toxic effect of the drug. About the surgery, on my opinion, we saw retrospectively all the videos of the surgery in our patients, and we found that the patients in which we performed one bubble and we detached the fovea had more frequently chororetinal atrophy. About the possibility that the toxic effect of the drug, from a practical point of view, we decide to change our follow-up plan and to examine monthly the patients to capture some inflammation signs, perhaps hyperreflective foci at CT, and so in that case restart with the corticosteroid treatment. But it's a mystery.
I have another question for all of you about, with the use a viral vector. How is the inflammation, issue? It's still an important issue, or and it depends on which kind of AV vector you use, or not?
Can I take this one? This is an excellent question. The inflammation is an issue with both intravitreal and subretinal administration, maybe more with intravitreal than with subretinal, but we do see inflammation also with subretinal, and it's AV serotype independent. So you basically get it whether you inject AV5, AV8, AV2 subretinally. And it's managed by preventive immunoprophylaxis with corticosteroids. And there are various protocols to deal with.
And you have to maintain the treatment, the corticosteroids.
So there are various protocols, yes, but typically you start before you inject the vector and you keep it for 2 months, or around 2 months after vector injection. And then depending on whether there is still inflammation or not, you may continue with the immune suppressive regimen.
Thank you.
Let me ask,
Corticosteroids regimen.
let me ask a quick follow-up question. Does transplicing occur under normal physiologic conditions in cells?
Right. So depending on what transplicing we're talking about. So if it's, let's say, RNA transplicing, this is what normally, so what we are doing for Usher1B, this is what normally happens in cells. So that's not something different. If we're talking about protein transplicing, which...
From two different transcripts, the transplicing in vivo. Single transcript.
Oh, no, but this wasn't the transplicing from two independent RNAs. So you have two pieces of DNA, they get together,
Yeah.
they get transcribed, and then splicing, maybe transplicing, it may not be the right word, but splicing basically splices away the recombinogenic region that are in the middle. So this is something that happens for any gene inside photoreceptors or inside the cells.
Yeah, but it's usually a single transcript, not two different.
This is a single transcript. So the two pieces of DNA get together, and then you have a single transcript. And then from this single transcript, the central region that are the recombinogenic region are spliced out.
Got it.
So this uses a absolutely physiological splicing signal and is a physiological phenomenon. What is different is the second platform that I mentioned for Stargardt disease. That is protein transplicing. That's something that doesn't occur in mammalian cells. It's something that occurs only in unicellular organisms from which we copied this mechanism, where you have two separate half proteins that are then joined together into a single full-length protein. So this is not something that mammalian cells normally do.
And Jordi, let me just run along because I know we're short on time. Jordi, how much residual vision should a patient have? Because you would think that the optogenetic approach would result in visual confusion for some patients if enough vision was still
Right.
available.
Well, yeah, there are two things which are theoretical that did not occur. The first thing theoretical is that we would expect neuroremodeling, that when you lost photoreceptors, the bipolar and ganglion degenerate and they never work. That's neuromodeling, but it appears that this neuromodeling is not that bad. That's one thing that it causes me a surprise. And the other thing is that Stargardt patients, you would suppose to be confused. And those 6 patients at least did not have issues on confusing. But yeah, theoretically, you should be having flashes everywhere, but it was not the case. It was not the case. Yeah.
And then, of course, you don't have the processing that you're used to having, right? So now you've bypassed the photoreceptors theoretically, and then the brain is getting some strange information, but I guess we learned to figure it out.
Right. Yeah.
Okay, well, thank you.
Congratulations, but I have a question.
Sure.
In the selection of the patients, retinitis pigmentosa or Stargardt, did you consider the duration of the disease?
I did not.
Can influence?
Yeah, sure. And I think that it can influence very much with all what I mentioned of neuromodeling, because you have had a disease long standing. All these circuits are degenerated by neuroremodeling. So probably the earlier you get, the more chances that these bipolars and ganglioners are more still alive and working enough. Yeah.
I
Yeah,
think
sure.
that in the advanced cases can be different.
Yeah,
from
that's true. But some cases were really almost blind, and they had some improvement. So this is defeating the theoreticals that we have as prejudgments. So yeah, it's nice that sometimes our prejudgments are not correct.
Jordi, the last questions, because otherwise Vasa will be mad with me. You inject the optogenetic things, and it works, but it's not working on the disease. So you can actually have, for example, in Stargardt, an increased atrophy and more deep atrophy of all the retina. How can work in that way?
Yeah, that's correct. But this therapy probably is intended for very end-stage disease that you don't care anymore in preventing progression, because whenever you have a disease at its early stages, you prefer to prevent progression than restoring the little part which is blind. But this has been done in patients who were beyond 2800 and more. So they were counting fingers and this kind. So there probably you don't care anymore in preventing progression, but you care on restoring something. So the optogenetics fits those patients that we would never treat for preventing progression.
Okay, thank you.
Great. Thank you to all of the speakers and our moderators. Fantastic talk. So we're going to move on to session 2 on imaging. So I'm pleased to introduce Phil Rosenfeld and Janet Sparrow, who will moderate this session.
Do you want to introduce the first speaker?
I'm here. Right?
Yeah.
I'm here, so you should do Christine.
Okay.
And then I'll do
Okay.
Fraser, and then we do
Okay. Thank you, everyone. So this session is on imaging, and I'd like to invite our first speaker, Christine Kershaw.
Thank you, Janet and Phil. I'm going to apologize in advance for my voice. I'm going to try to keep right next to the microphone. I'm going to be talking about imaging histology correlations in dry AMD, specifically gliosis and hyperautofluorescence in geographic atrophy. These are my supports and disclosures, and here are my takeaway messages. In geographic atrophy of AMD, the normally vertical Mueller glia bend to form the border of atrophy, which is the ELM descent. And they also form processes that adhere to Brooks membrane I'm getting flashing here, and wrap individual drusen that lack RPE. Hyperautofluorescence at the atrophy border can be caused by dysmorphic but continuous RPE, degenerating photoreceptors, or both. And histologies, especially of eyes imaged during the patient lifetime, can elucidate pathophysiology and assist in the interpretation of clinical imaging. So let's start with the Mueller glia in atrophy, these three features. So what we learned from the Sarks and the Killingsworth group that did all the fundamental ultrastructure of AMD, we learned about the RPE at the edge of the border of atrophy and the progressive degeneration of photoreceptors. But importantly, we also learned about the ELM that ended in a curved line, and the outer nuclear layer also disappeared. So what is the ELM? This is junctions between Mueller glia and photoreceptors. We can see them by EM right here. Those are the junctions. When we look at them on FOS, we can see that the Mueller glia form basically a Swiss cheese that leaves holes for the photoreceptors. The ELM is also an important physiological barrier that regulates fluid balance in the outer retina and maintains the environment of the photoreceptor in inner outer segments. So here is a dissected Mueller cell, and you can see that it's vertically oriented. What we described, the Sarks described it and illustrated it. We named it the ELM descent. You can see it right here. And we saw very early on that this was formed by vertically, there it is, formed by bent Mueller glia that were gliotic, as recognized with GFAP, glial fibrillary acidic protein. In OCT, the screen's flashing up here, by the way. In OCT, the Bond group described the curvature of the ELM and the disappearance of the photoreceptors. Other studies that I won't go into show that the ELM is reliably, and the descent is reliably present in histology, and it's visible in high quality OCT. Many OCT scans don't show it. You need good signal averaging. So Malia Edwards and Jerry Luddy showed several years ago that the reactive Mueller glia are concentrated in the atrophic area. You can see the ELM descent, and in there, all the Mueller cells are highly concentrated. And what they showed was that the Mueller cells processes repositioned outwardly, and they basically replaced the ELM with a glial seal. Now, in the next slide, what I'm going to do is effectively flip the retina up so you can look at the bottom of that glial seal in recent work that Malia has done. Sorry, it's the next slide after that. This is how the Mueller glia adhere to Bruch's membrane. This is in vivo OCT of a case that we could follow with histology afterwards. And you can see the adhesion to Bruch's membrane, the basal laminar deposit. The rest is artifactual detachment in histology, but it showed us the tremendous adhesion of the Mueller glia to the remaining tissue. So here's the new data from Malia showing the glial scar right here. That's what they called a subretinal glial membrane. These are all reactive with a GFAP, and you can see how the glia wrap and invade the individual deposits. And this is what the deposits look like. You can see on the here basal laminar deposit and a calcified drusen. In the on FOS view, there are the calcified drusen, and then the green of the activated glia. So there's a huge glial membrane and adhesion and scar. So now let's talk about the hyperautofluorescence at the border of atrophy. This is a recent case. I work with Bailey Freund. This is from his practice in New York. This is a white woman who was seen for 5 years. 6 years before her death at age 98, and we were able to study the two eyes. One was the right eye that I just showed you that Malia's lab is working on. Here is the eye that we studied multimodal imaging at the last clinic visit. You can see the three modalities. Interestingly, a thick choroid in the OCT scan that was visible also in the histology. So let's review for a second the subcellular sources of blue autofluorescence imaging. This is a complete reconstruction of a human parafoveal RPE cell showing all the organelles and membrane specializations. You can see the melanosomes in the apical processes, and right here are the organelles that are responsible for blue autofluorescence. That's lipofuscin and melanolipofuscin together. Tom Hock's group has classified these organelles showing with structured illumination microscopy, so you can see the different shapes of the organelles that are the source of the blue autofluorescence. So there are several routes to hyperautofluorescence. One is rounding, stacking, and anterior migration of individual cells. Another is reduction or loss of outer segments that absorb light. You can see the difference between the long outer segments and the shortened ones. And these can result in hyperautofluorescence, and we in combination. And we showed direct histology, clinical pathologic correlation to support this in the past. So this is magnified fundus autofluorescence 7 years before death. And we're going to focus on this area that I'm going to call a peninsula, which is caused by two adjacent areas of drusen-driven atrophy that pinch together over time, but it's long-lasting. And what you can see here, this is the bright autofluorescence. There's the ELM descent. Here are the photoreceptors that still have outer segments, but they're greatly shortened. And then here is continuous but dysmorphic RPE. In the next scenario, we'll look at this area where you see three zones of autofluorescence, bright, mottled, and dark. We're going to look at that. This is, again, the ELM border of atrophy right here. This is the outer junctional zone on the right, the atrophic area, the interjunctional zone on the left. And now we're going to look at this area at higher magnification. And you can see that the bright autofluorescence is inner segments lacking outer segments and continuous RPE. The mottled area is a lack of photoreceptors and dissociated RPE, or individual cells scattered across the atrophic area. And then at the left-hand side is an area of no photoreceptors, no RPE, and a large glial scar at the yellow arrowheads. So the takeaway messages then are here about how the glial cells form the border of atrophy at the ELM descent and a glial scar, and we looked at some of the causes of hyperautofluorescence at the atrophy border. And we can still learn a lot from histology of clinically documented cases. I want to thank all my collaborators, particularly Bailey Freud, who set up the donor program at his practice, and Malia Edwards' lab that did the confocal microscopy of the glial cells. Thank you very much for your attention.
All right, it's a pleasure to introduce Scott Frazier, who's going to be talking on imaging at the cutting edge.
Well, thank you very much. It's very nice to be here. And what I'm going to try to tell you about today, if the slides will advance. Can you advance it one slide? What I'm going to try to tell you about today is the way that we are using tools that we're developing in the lab for looking at sand dunes. Anyway, hopefully this will come back. Yes, we're looking at embryos and understanding the developmental physiology of embryos to build technologies that let us look at human retinal structures together with colleagues from the ophthalmology community. And one of the reasons I'm so happy to be here today is because it allows me to outreach some of these techniques. We've tried very hard to translate some of our techniques, so we have a number of different disclosures to make. But what I want to try to talk to you about today is a follow-on to something that you just heard about, and that is autofluorescence. And so we as a field are trying to make sense of the autofluorescence, or the reflection signals that come back. And this is still really in its infancy. It looks mostly at the amount of reflection. And what I want to argue for today is that one of the things we can do is to use the fluorescence, the excitation spectra, the emission spectra, and the time it takes between the two to get information about the chemicals that are underlying that autofluorescence. And so what we are in the midst of trying to do is trying to use technologies that we've developed for looking at embryos, which are very light sensitive and so very similar to some of our issues with imaging in the eye, to try to deal with what I call the pyramid of frustration. What we want is to be able to image throughout the depth of a specimen very rapidly and with no photo damage. And back here in this cloud is the photon budget, the threshold for damage, or the ANSI standards that would be necessary for us to meet to do imaging in the human eye. So one of the things that is a huge problem is that the spectra overlap tremendously. For example, if we take two commonly used experimental fluorescent proteins, green fluorescent protein and yellow fluorescent protein, even though they are called two different colors, they actually overlap in spectra tremendously. And if we add in some of the endogenous fluorochromes, the background fluorochromes that are there in all those settings, you can see the challenge. There's green fluorescent protein and yellow fluorescent protein, but in the presence of a huge number of other endogenous and exogenous labels. So if we take the normal approach of using most instruments, and that is to try to take a very narrow bandpass filter to only take that fluorescent signal from a subset of the spectral range, we make the problem of decreasing the amount of signal because we're throwing away all the signal that's here. And even within this narrow band, no matter how narrow we made it, it would not be selective for any one fluorochrome. Now, this is a problem that shows up often in imaging, and I will see if I can advance. Can you advance one slide, please? This problem shows up in imaging constantly, especially in the satellite world, where people were trying to use pictures from space to see what was beneath. And what they came up with was the idea of taking spectral fingerprints and unmixing them. An example of two very overlapped spectra, fluorescein and fluorescein in the red shown here, GFP in green, you can see the spectra overlap tremendously. But using this approach, a mathematical approach called linear unmixing, we can separate the signals out. Now, the problem is that this is, if I can have the next slide, please. We may have a battery that is dying in our remote control. Okay, the problem is that using those approaches are computationally expensive and computationally slow. And so one of the things we have been involved in is to use a Fourier transform of the spectra. So the green spectra becomes that green dot, the red spectrum, that red dot. A 50-50 mix of green and yellow becomes a yellow dot halfway in between. Because of this, it makes the mathematics much easier. If I can have the next slide, please. That means that we can get much better results as shown at the bottom instead of those at the top, which are trying to separate it out optically or with our old school mathematics. And the next slide, please. And click one more time. This shows a live animal separating out the multiple signals of 5 different extrinsic labels and then several intrinsic labels. Now, this problem is one that we face all the time with trying to image complex mixtures. You can see that the spectra, the spectra overlap tremendously here, and yet what we need to do is separate those signals out. We also have to deal with the fact that we want to do this with minimal numbers of photons. And so while this really works, this approach is showing clean separation of both exogenous labels, which you're seeing here, and now some endogenous labels, which have to do with the metabolism of the cells. So that brings us to a challenge. And the challenge is, can we extract more information from this pyramid of frustration by using this approach? And can we do it with less photons? The problem as we try to do it with less and less photons is it makes it much easier, especially in a diseased retina or a diseased situation to image. But when we do that, these nice spectra, which overlap but are well posed, become very, very bad spectra. And this is where artificial intelligence has, over the last year or two, really helped us. We've been able to teach the AI network, a UNet, how to reconstruct those spectra from a minimal amount of photons. And that is allowing us to both increase the number of spectra that we can separate out and decrease the amount of light, and also increase the dynamic range. And so if I could have the next slide, please. That's allowed us then to reconstruct these spectra. Next slide, please. That takes images that would otherwise be too corrupted to be able to use and be able to see the perinuclear signal and the cell membranes very clearly. So the sort of light dose that we can see through a pupil would be able to see signals that Christine would like to be able to see. To give you an example of that, if we can have this is separating out several extrinsic signals from an experimental animal. And what you can see here is the background noise, the autofluorescence. And in the same presence of signal, we can see the background parsed into its many different parts. For example, the retinoids, NADH free and bound, which gives us the metabolism of the cells. And this has allowed us then to be able to see extrinsic signals like the metabolism and like the autofluorescence that Christine was just telling us about, but in the presence of high levels of autofluorescence from things like lipofuscin or other age-related deposits that would otherwise confound our ability to see things. Now, this intrinsic imaging that we've been able to do allows us to take experimental animals and separate them out into metabolic signals, retinoids and retinoid metabolites, and other metabolites within the cell. And as powerful as this has been spectrally, it has not been as powerful as we would like, because especially in older eyes, the optics get imperfect. And in those imperfect optics, we really take a great power from looking at the lifetime of the fluorescence instead of the color of the fluorescence. This just allows me to say that the same tricks we use for interpreting the multispectral imaging works for the autofluorescence, where we can see the difference between a cell that's undergoing oxphos or glycolytic metabolism. And this allows us to look in a single image and be able to see the metabolic state of the cells. Here you're seeing dinner time per group of epithelial cells in culture. We're now using these techniques to allow us to separate out the different endogenous metabolites from the different exogenous signals. We've recently gone from being at light levels that wouldn't be meeting the ANSI standards, so we had to use retinal organoids, to now meeting the ANSI standards. So that's allowing us to be able to do this in intact subjects, intact patients. And if we can have the next slide then. I'm just going to say what the take-home messages are beyond my take-home message, which is don't use a Macintosh for presentations here. Next slide, please. Okay, the take-home messages. We can use tricks like the spectral phaser analysis to buy ourselves about 10-fold in the efficiency of collecting the photons and about 1,000-fold in processing it. The machine learning buys us another 20-fold or so in the signal that we can get. And where we are right now is combining the spectral and the lifetime imaging to image the metabolites and other autofluorescent components. And we're using that now, taking advantage of this 50-fold more sensitivity to start to collect this through the pupil of intact subjects and trying to get a better idea of some of the processes that Christine told you about in histological sections, but in our case, imaging them in the live specimen, the live subject, sorry. Thank you.
Thank you, Scott. The next speaker is Phil Rosenfeld, and his title is OCTA Imaging of Non-Exudative AMD.
It's a pleasure to be with you this morning. I'd like to thank Giovanni, Vas, Francesca for the opportunity to share with you the work we've been doing looking at OCT angiographic imaging of non-angiographic structural features, which is what we do routinely. And I'm going to talk specifically about predicting the progression in intermediate AMD using the area of hyperreflective foci. We're going automatically, just so you know. Hyperreflective foci and drusen volume. I do receive research support, and I'm a consultant for Carl Zeiss Meditech. I'd like to thank my fabulous OCT research team, particularly Alessandro Berni from Milan, who's going to be back in September, and he's a force to be reckoned with, and you'll get to know much better in the years ahead. Ricky Wong's group for his fabulous algorithm development. So the total macular burden of hyperreflective foci can be measured in both the intraretinal space and along the RPE. The total area of macular hyperreflective foci actually outperforms drusen volume in predicting disease progression, but drusen volume is more easily obtained using algorithms that are available. Now, in intermediate AMD, drusen with pigment progress to these area of atrophy, which is characterized not only by the atrophy, but by the calcifying drusen that we'll get into later. So the role of hyperpigmentation as a risk factor for progression from intermediate AMD to GA is well established. Just so you know, we're still losing our video up here. But it was really Jay Duker's group that first showed that pigment could cast this hypotransmission defect into the choroid. And this led to the ability to identify these intraretinal hyperreflective foci, which have been shown to really be associated with disease progression by a number of groups. And a recent systematic review of meta-analysis showed that these intraretinal hyperreflective foci really increased the odds ratio and the hazard ratio significantly. But it was really back in 2012, a combination of the AREDS group and the OCT group at Duke that, just so you know, it didn't get fixed, so it's still flashing up here, that show that there's really two components that need to be appreciated. Those components are the intraretinal hyperreflective foci, but also the hyperreflective foci along the RPE, or what we call RPE HRF. These are really important. And as the Duke group showed, it actually starts off with the increased pigmentation along the RPE, and then that migrates into the retina as intraretinal hyperreflective foci. So we showed using on-phos OCT imaging and dense raster scans that both these forms of hyperpigmentation can be seen on on-phos imaging. And when you look at a slab from 64 to 400 microns under Brooks membrane, you can see these dark spots. And these dark spots both reflect the intraretinal and the pigmentation along the RPE. And as you can see beautifully in this color from this image that has many of the features in AMD, dense raster scans can yield not only drusen maps from the same scan, but you also see these hypotransmission defects and hypertransmission defects. And these hypertransmission defects are the early stages of atrophy. You can see on the B scans that are easily obtained from these on-phos OCT imagings, the calcifying drusen that also cause hypotransmission defects, the hyperreflective foci, here additional hyperreflective foci and the hypotransmission defects, and the hypertransmission defects even before the drusen collapse into more typical atrophy. So on the on-phos image, you can delineate the hypertransmission defects and these hypotransmission defects that correspond to the hyperpigmentation. Now, Ricky Wong's group came up with a fabulous algorithm replacing signal intensities with optical attenuation coefficients. And this is a technique that was first developed by Vermeer in 2014. And it actually highlights the intensity of the RPE in terms of its attenuation coefficient. And as those hyperreflective foci move into the retina, you can easily distinguish the intraretinal and those that punctuate the RPE layer. So we performed a longitudinal analysis in patients with intermediate age-related macular degeneration. 6 by 6 millimeter swept source OCT angiographic scans were performed at baseline and follow-up. We detected and quantified the intraretinal hyperreflective foci and the RPE hyperreflective foci using these sub-RPE slabs. And we associated the total area of these hyperreflective foci with the onset of persistent hypertransmission defects. So this is what the on-phos image looks like from the algorithm, and the grader can look at individual B scans through these regions and pick up the RPE, intraretinal RPE, as well as the RPE along the hyperreflective foci along the RPE. So this is an example of how the disease progresses and how we can quantify these areas of hypotransmission defect. And then we develop these masked outlines that are easily quantifiable in terms of area of hyperpigmentation. Okay, and you can see very easily the individual B scans and how they correspond to these findings. So what are our results? We followed 171 eyes for average about 60 months who had 3.3 on average visits annually. 78% showed these hyperreflective foci, and 48% developed these hypertransmission defects. And we looked at the univariable and multivariable analysis for disease progression. And while individually, both the hyperreflective foci and drusen volume predicted disease progression, in the multivariable analysis, the hyperreflective foci were really important. And these are two cases that show why. This is an area with large drusen volume and significant hyperreflective foci burden. And you can see as the disease progresses, the hyperreflective foci give way to hypertransmission defects that progress and form the more typical atrophy, which you can appreciate on this slide. This next case shows an area with very small drusen volume, but a big burden of this hyperpigmentation. And as the disease progresses, you get multifocal hypertransmission defects that merge and enlarge. So this is just a video that my fellow put together showing that these dark spots are really the hyperpigmentation or hyperreflective foci. And those dark spots are where the hypertransmission defect form and grow and coalesce and merge into your typical geographic atrophy appearance. So when we did receiver operator curves, looking at the thresholds in which disease prediction is important, you can see that a hyperreflective foci area of 0.07 millimeters squared and a Juzin volume of about 0.22 millimeters cubed really is a nice threshold to look at prospectively in these Kaplan-Meier type plots. And these are looking at Juzin volume on the left that's greater than 0.22 or less than 0.22. Sorry, everything's progressing automatically. All right, and this is for the pigment. You can see the pigment is actually a little bit of a better predictor of at least 0.07 millimeters squared or larger. And when you look at a combination of all of them, picking a Juzin volume of 0.22 millimeters cubed or greater and an area of 0.07 really gives you the best predictor. And this is what's interesting here. You can appreciate that if you look for the appearance of the second or third hypertransmission defect at day 0 here, you actually, in most eyes, once you form one, there's a high likelihood of forming a second or a third. And it doesn't matter what the risk factors were getting into this stage. Once you get to this point, additional hypertransmission defects form. So this is the first study that really quantifies the total burden of hyperreflective foci in the macula. And if you have to choose one, you'd rather choose hyperreflective foci. It's a better predictor. But just Juzin is easier to measure because there are a number of algorithms that can actually predict Juzin volume on any OCT device. So I'll stop here and thank you for your attention.
Thank you, Phil.
All right, it's a pleasure to introduce Bailey Freund, who's going to talk on novel findings from high-resolution OCT.
Thank you, Phil. Thank you for the organizers. So I'm going to show some interesting cases using the high-res OCT from Heidelberg. My disclosures were there. Briefly, this is a technology that functions just like our standard spectralis unit, but it increases the axial resolution to 3 microns. The lateral resolution is unchanged. And here's just some examples of the increased detail that we can see with the high-res OCT. We can see greater delineation of the outer retinal bands. We can see the distinction between Henle's fiber layer and outer nuclear layer. And we can see much better vascular details. We also have a real-time module. Let's go back. Is that? These worked before. If the videos do not function on this, this will be a very boring talk. So is that fixable? Because if not, this may not. We may not get too far. I'll skip that video. It's a very nice real-time showing vascular flow. Here's a 49-year-old healthy male, central visual disturbances, right eye 20/40. Here is the high-res OCT on the right, where we can see subretinal fibrin, subretinal fluid. This is obviously a case of central serous. You can see choroidal congestion there. Here we can see something that's been described a while ago, which is a lucency in the fibrin or subretinal hyperreflective material, which if you look very closely, you can identify this in a high percentage of patients that have a focal liquid CSC. So basically that's where the RP defect, you can see the hypertransmission. Below that, fortunately, this is functioning here. So this is a dense raster pattern through that defect, and you can see the serous PED, and that will be contrasted with the next case I'm going to show after that. So this lucency originally described with time domain OCT by Tomohiro Aida. Here it correlates with the focal hypofluorescent area on autofluorescence, the focal leak on fluorescein, and then the intense leakage later. And this is a paper by Nicholas Unitzi describing this on SDOCT. So now I'm going to move to the second case, and it's quite similar to the first. This is a 54-year-old male, central visual disturbance in the left eye, and if we blow this up, you can see that there's a serous neurosensory detachment, so another case of central serous. But there's something a little different about this case. If we look right here, that doesn't really look like a serous PED. It looks more like a shallow regular RP elevation. And if you look closely at some of your patients with CSC, you'll see a double layer sign of splitting. And if this doesn't show, that's unfortunate. So here's the vascular flow movie, but it's not showing as a movie. Is it possible to present this with the laptop that I tested my slides with? Because otherwise, we won't be able to see any of the real-time OCT.
There we go.
Yeah. Well, I can move on, but now I think I can't advance. Okay. What I will show in the next slide is, since we have some time to kill, is I find that quite a dilemma when you have a patient that looks like typical central serous. You're all ready to do PDT because it's chronic, and then you realize that they have type 1 MNV right in the area that you plan to do PDT. And should that warrant doing anti-VEGF in addition, or should we try anti-VEGF first? Because it's then not completely clear whether you're dealing with a case of exudative neovascular AMD or just a straight central serous that has an incidental finding of type 1 MNV. I tend to use anti-VEGF, pre-treatment, treat it, and then maybe give one more, and then just so I don't have to worry that I'm going to make the neovascularization more aggressive by upregulating VEGF at the time of PDT. So, there's a lot of sort of speckle noise and there's some movement. And so sometimes it's hard to tell what's just sort of bulk movement, the speckle noise, or what's really vascular movement. Had the first video played, it was very obvious where there was movement within the vessels and what was just the tissue around it. And it correlates very nicely with this. Looks like it's going to take a while. So just tell me if I should go on. I think we'll get to the point where you won't really need OCT angiography because you can just see the movement, and that's what you're measuring with OCTA, and then you avoid all the issues with artifacts, right? Because sometimes you're not sure if you're dealing with a projection of flow onto another layer, or the flow's really in that layer. You get some of that issue with the real time because it will cause speckling kind of below a large vessel. But it's very neat to be able to see the flow in the tissue itself. I'm going to keep moving ahead because I don't want to take up other people's time. Here's just showing confirmation with OCT angiography that there definitely is neovascular flow in that lesion. And then I discussed earlier that might represent a treatment dilemma. This was a patient referred to me by a former fellow. They were concerned about the possibility of a nevus, just a nasal to the disc, where you can see that alteration in pigmentation. I'll blow it up here. There's also some subretinal fluid here. You can see that with the autofluorescence on the right, that there's sort of a gravitating detachment. Here's OCT angiography. Again, that should have been a movie that would go through all the different layers, and what you would have seen is dilated veins beneath the area of neovascularization, which also showed aneurysms or polypoidal lesions, and also choroidal hyperpermeability. Here's the OCT AB scan, so you can see the small aneurysmal lesions in that type 1 neovascular complex nasal to the disc. I'll blow that up here, and you can see that here, and this should have been a movie as well, which would have shown the increasing choroidal hyperpermeability and the hyperfluorescence of the aneurysmal lesions. This movie will probably not play either, but then we've imaged a lot of these aneurysms, polyps, glomeruli, whatever you want to call them, and it's very interesting to see the vascular flow, both in the branching vascular network that leads to the aneurysmal lesion, and then just the flow in the lesion itself, which tends to be sort of slow as we might expect, but hopefully there's some clinically valuable information. When you look at the way that moves in there as to what type of lesions are more likely to exude and which ones aren't. What I want to show here is that with the high resolution OCT, these were really not visible with any of the other en-face imaging and very difficult to pick up with the standard OCT, is this patient has very small hard drusen, cuticular drusen, nodular drusen, and we'll often see these patients who have secondary macular neovascularization, overchoroidal vascular hyperpermeability, congestion, is that presumably these are patients that have at least some genetic risk for AMD. So maybe high risk and complement makes them more susceptible to develop neovascularization in the setting of the insult of choroidal vascular hyperpermeability, which leads to damage of the choroid capillaris. So it looks like I'm running out of time. This is just a nice segue from that, showing a nicely imaged case of cuticular drusen. I'm particularly fascinated with the retromode image on the right there, where we can see a very strong correlation between the starry sky pattern on fluorescein of cuticular drusen and then those tiny little bumps that we see on near-infrared reflectance. This is more of an image that is intended for the CAM meeting, which follows this one, which is something that I'm quite confused about, which is that if you have a cuticular drusen, which are tiny little excrescences of the intercollagenous layer, and then you develop a double layer sign, sort of as we see here, are the cuticular drusen now up here, or are they still at the base of Bruch's membrane? It's something that Christine and I have talked a lot about, and there's something that I think we just don't understand from that transition from the cuticular drusen through the soft drusen and how basal laminar deposit fits in. Here is the final case, I think, a 71-year-old male. You can see has drusen. There's something very difficult to see right there. It's a tiny little red lesion, which turns out to be type 3 macular neovascularization. Here we can see the lesion as it sort of infiltrates through a defect in the RPE, and the Mueller cell activity that Christine discussed earlier infiltrates the basal laminar deposit, forming adhesion between the outer retina and the sub-RP space, but just going into the apical surface of that drusen. Here is a higher magnification of that. Unfortunately, I don't think the vascular flow in real time will show, because you can actually see on this image the nice correlation. If the movie was working here, the movement within the type 3 lesion that corresponds perfectly to the vascular flow here. So you'd see movement right there if the video was working. And you can see here that when you segment the lesion, that's entirely sort of at the level of the deep plexus with no vascular flow in the segment right below. So nothing's gone deeper than the basal laminar deposit. So truly neovascularization of retinal origin. And since I'm way over time, I'm not going to talk about the histology, which I've worked on with Christine. Love to show that. And then the overlap between these microvascular abnormalities that sometimes are called PVAC or drama, as we refer to them, that can masquerade as type 3, but really don't penetrate through the RP and are truly not true neovascular complexes. Here's histology of one of those. I want to acknowledge the work of my fellows, Olivia Fays and Jacques Dijon, my prior fellow, on this work with high-res. Thank you for your attention. Just addresses Scott's question before that if you have a patient that's cooperative enough and you acquire, this is not perfect real time, right? Because if the patient moves, it has to track these images, and then depending on how quickly you're acquiring, then you can adjust it to the actual time it took to acquire. But you can see quite clearly that, you can see vascular movement. Giovanni's talked quite a bit about this feature and how the vessels appear and why they appear in certain ways.
Our next speaker is, or the last for this session, is Vasada, and the title is Clinical Applications of Peripheral OCT.
Thanks, Janet. Thanks, Phil. Sorry, Bailey, that you had all the difficulties. I guess the codec for the video just wouldn't play there, but hopefully this will work okay. So I'm going to talk about peripheral OCT. These are my disclosures. And just to sort of define what I'm going to be speaking about, I'm really talking about imaging the far periphery based on the consensus nomenclature definitions. We're really talking about imaging the retina beyond the vortex veins. So, okay, so again, this is actually not a new concept. This has been possible for many years. If you can adequately angle the patient or angle the OCT device, it's possible to scan in the far periphery. But this could be a challenge sometimes. You really need wide dilation and a skilled operator. So not every patient can do this. Nitin Chaudhary published this very nice paper where he was able to achieve really pretty spectacular imaging beyond the vortex vein ampulla and demonstrated these types of abnormalities that we all expect to see in the peripheral retina, right? These kinds of tractional abnormalities and the like. So most of our experience, though, comes with a newer device, the Apto Silverstone device, which actually makes this much easier because you essentially can get an OCT scan wherever you can get a color image. So I really want to talk about how this can be helpful in clinical practice, because remember when OCT first came out, before we had a lot of clinical trials, I think for many of us it became intuitively obvious that it was quite useful. And that's been our clinical experience with this technology, although I'll show some data as well from some early studies. So this is an example of a patient of mine with a confirmed gyrate atrophy. I always wondered about these areas of preserved pigmentation. I was thinking about the earlier case that was shown with the bridging areas between areas of apparent preserved autofluorescence. So we wondered, well, what does that area look like with OCT imaging? Of course, now we can access these areas so we can study them. And in fact, actually, we couldn't find any real preservation of the photoreceptors in these areas that we thought were actually preserved. There was a little bit of a thicker retina, thicker RPE in those regions. You can see the thickening of the RPE band. That actually seemed to correspond to those areas, but there was no photoreceptor structure. This has actually been a very useful tool for evaluating patients with various retinal tumors. This is a patient actually has a regressed retinoblastoma, but we've been monitoring her over time by scanning these lesions to see actually if these regressed retinoblastoma lesions change. Another patient with pigmented preserved pararterial RPE, and due to a CRB1 mutation. This patient actually was one that we recently published on because they actually have an astrocytic tumor as well associated with this particular manifestation or phenotype in this patient that we were able to obviously obtain scans through, and you can see that it's an intraretinal lesion. Another patient, courtesy of a colleague in India, Pradeep Venkatesh, who's a von Hippel-Lindau and a capillary hemangioma. Again, you can actually scan through these areas. You can actually see the vitreous relationships, the significant exudation. And actually, you can even, I'm not going to go through it in detail here, but actually spot the bridging vessels and the like. The feeding and draining venules. Here's a patient who was referred to me from one of our oncology hospitals locally, who's known to have CNS lymphoma, had new vitreous floaters. We were already suspecting that this patient had intraocular involvement, but it was nice to be able to actually scan through these regions because you can actually see the accumulation of material, as you'll see as the volume scan goes through in the sub-RPE space in this patient, typical of what you'd expect in a patient with CNS lymphoma. We found this to be a very useful tool for mapping the extent of peripheral schisis. I've been tracking a lot of myopes this way, and by mapping their schisis systematically over time. This is a patient actually we had published who has an extensive schisis, very unusual case with an optic pit retinopathy, but the schisis goes through significant portions of the retina. The patient even has an outer retinal hole. But actually, you can see even way out here supratemporally, or supranasally, excuse me, this patient has evidence of schisis. The whole retina is not schitic, but it was interesting to see how far this extended. Again, you could map this through this kind of scanning approach. Again, as I've already hinted, that it's been very useful for tracking tumors like nevi and the like. Again, we don't really guess anymore with any type of peripheral pathology. We'll just get an OCT through it, right? So we really don't get the 3-mirror lens out anymore. It's just much easier to do this with OCT. This is a patient with dome-shaped maculopathy. But again, for peripheral retinal lesions, seeing the vitreous relationships and the like, I think it's going to be very helpful. It's a patient with a dialysis where you can see the vitreous adherence at the vitreous base. And in fact, we've been recently very interested in lattice lesions. I think these have been well described historically, histologically, but we're able to study them now in OCT in patients. One of the interesting questions for us is, can you actually, with different types of peripheral retinal pathology, can you actually distinguish patients who may be at higher risk for developing a retinal break or other complication based on evaluating them with OCT? We recently, this is just more examples of patients with lattice. It's interesting, not only do you see retinal thinning, but many of these patients can have hyperreflectivity within the retina. And this was unpublished material, but something we just presented at ARVO on a large series of patients with lattice degeneration. We've characterized their OCT findings. Again, as I said, when we have questions about lesions now, we pretty much just will do an OCT through those areas as a patient with a cystic retinal tuft. We have many patients who come in with barrier laser, which for a doctor who suspected this patient had a retinal detachment. Most of these patients, in our experience, actually turn out to have schisis that has been walled off. And again, another patient who was referred with a retinal detachment had barrier laser, but actually, as you can see, her has just evidence of schisis. Again, it's very interesting to see this patient we were going to treat with laser anyways, because even though they had an operculated hole, they were symptomatic, and we can understand why, because there was vitreous adherence around the margins of the hole in this patient's case. And again, as I said, this is helping us understand why I think many of these patients have symptoms. Here's another patient we suspected had a pigmented tuft, maybe even a little hard to see on the color image. But when we actually scanned through the area, it actually was not what we expected. There was actually some area of retinal elevation that obviously would have been pretty challenging to appreciate this, even with a contact lens by microscopy. More examples of that. I don't want to bore you all, but more examples here. Another operculated hole where, again, you can see significant vitreous relationships around the margin of the hole. And another patient with more extensive proliferation on the retinal surface. So again, I just showed you a number of clinical cases because, as I said, it's pretty intuitive in terms of the clinical value from a perspective of, the patient has a peripheral lesion. You're trying to evaluate this before we'd evaluate it using ophthalmoscopic techniques. So again, now we're able to study that with OCT. But I want to acknowledge Nitin Chaudhary, who did a more systematic study of patients presenting to his practice and demonstrated that in a large proportion of patients actually getting the OCT scan changed its decision-making. And largely it's because of this sort of undetected traction, or at least clinically difficult to establish a traction, which you can see much more easily when you apply OCT through these areas. Again, this is imaging at the vitreous base near the ora serrata in this particular patient, where you again can see the vitreous relationships and some of the peripheral cystoid degeneration that's commonly present. We know this from histology, so this is not really new information. A patient with a retinal detachment that Nitin had imaged, and again, you can do imaging through these kinds of retinal breaks in the detached areas as well. Maybe it's helpful to see these vitreous relationships before surgery. Another patient with actually was supposed to, this is showing you the fact this patient had a macular hole, but also had a retinal hole in the periphery. So in any event, we have found from just a clinical utility standpoint this to be useful. I don't do much in the way of 3-mirror lens peripheral examination because it's just much easier to obtain an OCT through these areas. So that's become a routine part of our practice. Our photographers, when they spot peripheral pathology, will automatically get the OCT. So through those areas, I don't have to send them back. And much of this, as we sort of experienced kind of an interesting revolution with macular OCT, which is essentially shown and demonstrated in multiple well-conducted studies to be superior to slit lamp biomicroscopy. The thought is that we may have a similar experience with peripheral OCT, but of course those studies have yet to come. Thanks very much.
All right, I'd like to invite the speakers up here, and we'll take questions from the audience. And we have Carl Chalky's got his hand up here. I've got a question for Christine Curcio, and I know she's the one that can answer this. So, Christine, what causes drusen to start the calcification process? What's going on histopathologically? Any insights?
Excuse the voice. Thank you for the question, Phil. What we speculated on, and the we in that sentence is Emery Lengyel, myself, Boss was a member of the authorship, was that the RPE is really undergoing a state of degeneration at that point. That the metabolism it's changed, and which causes a change in the extracellular space and an imbalance of the calcium and magnesium ratios. Actually, the RPE, when it's releasing lipoproteins as part of drusen, is pretty healthy, but as the degeneration proceeds, the extracellular matrix, the extracellular space changes, and that's what promotes the formation of calcification. That's our hypothesis.
Right.
Carl?
So this is to you, Phil. Fantastic talk. What is the role, do you think, of anti-complement and these hyperreflective foci? Are you seeing changes in the appearance, anything in the dynamics that might suggest that there's complement involved in either the generation, progression, increase in the number, anything? We haven't had, we have enough time now of treatment that we could see in patients a change. What have you observed?
So I've learned a lot from Christine, and what I've come to appreciate is these hyperreflective foci, first along the RPE and as they migrate into the retina, probably indications of epithelial mesenchymal transition. And these cells are under great distress, and they're leaving the monolayer, they're aggregating, they're accumulating, they're causing the hyperautofluorescence signal, and then they're escaping. And it's really a point of no return. And those foci, particularly along the RPE, that's where the atrophy, the hypertransmission defects, the atrophy is going to form. So once you start seeing those foci, and it could be variable in terms of when they first start appearing and when they progress. That's why once a certain burden is achieved, I would love to have a treatment, and we're going to be talking about this at CAM. I would love to have a treatment to implement at that point in time, because you know what the future holds.
So I just wanted to make one quick comment about that. I totally agree with the discussion about the hyperreflective foci. We'd also used Ricky's algorithm to do some quantification for the Amish study. We actually had to do a reasonable amount of manual correction because of segmentation.
It's semi-automated.
Because it's definitely semi-automated, to be sure. But exactly the same observation we published, that once you get the first hyperreflective focus, that's a bad transition. That's why I don't know that you actually even have to quantify them in terms of as a biomarker. There's no question if the greater quantity is an increased risk, but just getting one is a bad feature. And we'd recently published also what are the risk factors for developing them. And this is an observation I think David made as well. Maybe he's going to comment on that, that it does seem to be that after the drusen get to a certain height, at least it's one of the mechanisms, it's not the only mechanism, that when the separation from the choriocapillaris to the RPE location increases to beyond a certain limit, then these RPE cells say, get me out of here, and they start heading for greener pastures, or they think are greener pastures.
That's why I showed those two cases. One clearly was associated with increasing drusen volume, but the other cases didn't have a significant drusen volume, yet developed a hyperreflective foci burden, but probably the underlying mechanism is the same. Those eyes have these thin double layer signs, basal laminar deposits accumulating, and you probably have a choriocapillaris deficit that's accumulating.
I think that's the key. That's the key, is that there can be many different pathways. Drusen height, I think, is one, but if there's underlying choriocapillaris ischemia from other etiologies, that's going to lead to RPE breakdown. You see it, for example, in pentosan retinopathy. Hyperreflective foci are very prominent in that condition as well, and that's from underlying toxicity to the choriocapillaris.
So we've obviously identified two broad categories here, and we're always focusing on the drusen because that's what we've seen and that's what we follow. But as was shown in the ARED study early on, these changes in pigmentation are really, really important and can serve as identifying eyes at high risk for disease progression that we can implement therapy on.
Bill, can I just ask a question related to that? If you're looking for hypotransmission defects, if that's what you call.
Well, yeah, that's what we look for. The hypotransmission defects can occur from calcifying drusen and from the pigmentary.
Well, that's okay. How do you tell one from the other?
On B scans.
So you have to look at.
You always have to look at. What's so nice about en-face imaging is it's comprised of B scans. So you always have the B scans. And when you look, you can just move the cursor to where the lesion is and you see the B scan.
So if someone were to grade using hypotransmission, the sub-RP slab, it would require also looking at B scans simultaneously.
Yeah, but we do that all the time. We're always looking at it either in the algorithm or the review station, and where you see a lesion, you just move the B scan there and you see exactly what it is.
One thing I wanted to ask, I didn't get a chance to ask, I think it's really cool that you showed the sequence from the hyperreflective foci to the hypertransmission defects, but I think there are a significant number of patients where you just see the hypertransmission defects without necessarily seeing the hyperreflective foci. And that might be an interesting.
Say that one more time.
So there's definitely a sequence where you go from the hyperreflective foci to the hypertransmission defects, and that may represent an earlier pathway to the hypertransmission defects. But in my experience, you also see hypertransmission defects without any prior hyperreflective foci, and it may be interesting to just see what percentage of patients who have hypertransmission defects start with the foci or don't start with the foci.
Right. Well, we have that data, and that's going to be an intense discussion at CAM over the next few days. But that's why there's at least two categories: drusen-driven hypertransmission defects, drusen plus these hyperreflective foci, and then the hyperreflective foci by themselves.
When we did the CAM5 paper, there were two types of sort of hyperreflective foci. There was intraretinal migration of hyperreflective foci, but in my opinion, there was also this sort of hyperreflective thickening of the RPE band at the dome of drusen. Also, you see a ton of that on drusenoid PED. Do you think one of those is more predictive of progression to atrophy?
This gets back to the ARED's work with the OCT and Cindy Toth. That's how it starts. It starts with the thickening of the RPE, the renegade RPE cells then migrating into the retina. But it all comes from the RPE.
But I don't think they always will migrate. Some will, but some don't. Sometimes it's just, there's also the underlying, wherever you have that hyperreflective thickening, you have thick basal laminar deposit. So that's another thing that has to break down as well to get a collapse.
Sorry. We are running out of time. Last question to Scott. Sorry, Scott.
How about Usha? I thought she had this.
No, we already.
I thought that. I'll take her time.
What about what you show in? It will be possible to do in VI, so Retina, and how small you think we can get, details. We can go to the cellular level also at the Retina, and if yes, in how long do you think it will take to get that point?
So the question is, how many molecular species could we separate out and what spatial resolution?
Exactly.
And that's what we're trying to push right now. I would say within one focal volume, so within 1 pixel, I'm hoping we're going to be able to separate out dozens of specimens in a relatively real-time image. And that's why I was excited about following Christina's, is thinking about which of those hyper-reflective signals and hyper-fluorescent signals we might be able to parse apart and be able to segregate.
Okay, sorry, we are a little bit late, so there is a coffee break now.
Just a little bit.
So that's one of the things I'm curious about is,
Giovanni, you want to mention what time to come back?
Could we see the calcification
10:50,
process,
10:50,
for example?
10 minutes.
Could we see some
Yep,
of
thank you.
all the different chemistries that are happening?
I think so, because, you know, I didn't give a very complete answer to Phil's question, but in the science translational medicine, it showed
Yeah,
loss
but
of
did you want to do this?
loss of
Yes,
autofluorescence
absolutely.
being
What are we
signaled,
going to do?
Well, we
over
can
the
actually do
drusen
now,
that were
or if we
calcified.
don't, but we
So,
don't
I mean,
have
that's
time.
loss of
I
simply
think for you,
intensity of blue
I think we
loss.
can actually
So they're
move
giving you a
during
full
the
range of
That's what I'm
color.
curious about.
Yes.
Is if you could see
Exactly.
a spectral shift there.
Exactly. We can. Why don't we do like here? It will start him, and then we can actually do film before the exactly.
Or you can just join it.
Exactly. What's that? Yeah, because we have quite a lot of time here, so we can actually.
Well, you know, this.
But particularly because we already introduced, we really introduced.
Oh, okay. You decide.
No, but I think it's fine because we will introduce the drugs,
good.
and then the
Perfect.
That'll be perfect.
Yeah, all this gets at designing the clinical trial and who's your most at-risk patient.
So I'm thinking Rick is going to be sitting there. So that stays, I'm sure. But you're so right that there's very interesting scores many cases that they've been based on that. And our approach is to see
They're all mixed up, right?
I don't know why. The abstract is based on method of composite is the diffusion characteristics of which is that line.
It's going to be fun.
Because we have a very nice theory about the elevation, the distance the moves away. It's more to the, but I'm saying, but the thing is that we should be able to develop mathematical model of why.
But did I answer your question?
No, but I thought you were going to invoke something about the real sense. Don't you think the real sense contribute to changing that environment?
Maybe, but I think it's a secondary effect.
I never paid much attention to my when I used to get books. I need to. I'll be back.
Well, I mean, it's the end stage.
It's game over.
It is, it's the end stage. I have something wrong with you, but you can see how the classification is growing.
We have it all.
Yeah, well,
No, but you look at different stages.
see, your study is why we did our study. You know what you're looking at. You can. That's it.
Learned a lot. I read a lot. So really, I wanted to say, well, let's introduce it
Yes,
it'll be willing.
well, not a little, I mean, like the beginning.
Of course. It's about size. Right now, we're waiting for the atrophy to form. Donut shape. Once it's done, it's done. And you can see now that
Even, see, I don't have a sense of suppose going on. Sit there.
So methods for versus the chosen body. What is it really apples to apples if you're doing after segmentation versus you're just about comparing the two methods of measurement of getting air of hypereffective foci using volume. So HRF, you're getting an area and you very carefully segmented that graders of the circle an algorithm versus a slab, which isn't a careful measurement.
You say a slab. You use slab. A slab always is truth bound.
Yeah.
So what's your slab?
Same.
RP Brook?
Yes.
So that's choosing volume.
But you could say you're missing a bit because you're choosing volume.
The algorithms have been pretty well bounded. The big difference between swept source and spectral domain is that swept source, the segmentation is where the spectral domain sometimes. So there's a small volume difference. We've done the comparison.
Okay, so you're happy with that. Yeah. And the other problem with choosing volume is choosing volume changes.
So we studied this years ago. So the
regionally there's changes.
Turungu senza nessuno. Però capite pure sui cancellare, non pensa che perché sono tre.
increases, and then like the hypertransmission defect. Before and before baseball. That's why I like the hypertransmission. It precedes the
I like hypertransmission defect and hyperreflective because the choosing volume still you don't
And remember
know.
the hyperreflective focus are the hypo transmission.
Yeah, oh yeah.
So you can use the same slab to look for both.
Yeah. I just think choosing volume is
for the next session.
because you don't know if they're on your way up in terms of increasing volume.
It's a threshold. Well, so yeah, because we're fine. So Jews and volume are useless.
Yeah.
Jews and volume can go away, but no sequelae. It's rare.
Yeah.
But most of the time, Jews and volume is a nice cutoff for a high risk.
Yeah.
Once it gets to a certain point, that's why we deal
Yeah.
with that threshold. So
Yeah.
undulates go way up. Once it gets to that threshold,
Goes back down. What if you catch the patient in the down and you don't quite realize?
well, that's why we use some legal activity, right? We discussed this. So if like 0.22 is your cutoff, so 0.22, and throw in hyperreflective focus.
Yeah. See, that's I like all of that, and I think.
Yeah, you'll see this clinical trial design.
Yes, I saw that.
Throws in this.
I mean,
Shows you you're at the point of
yeah. In a broader kind of perspective, I think it's like the beginning DR, right? Retinopathy, what did they know? They didn't know. So they looked at every variable, right?
Right.
So hard exiting didn't really matter. Soft, hard was soft, didn't matter. But they left it.
Right.
So, good. I'm glad you agree. Because I think we got it collectively.
So, but once it gets to a certain 0.2, once it gets to that 0.2, it's pretty rare.
Yeah.
But going there, it can go like, yeah.
Ne manca uno, quasi quasi, però cioè, è un po' brutto, però mettere qualche bicchiere di plastica. No, ci metto un cazzo.
Stufile candela?
I mean, we got a lot of guys that talk in town. We just can't afford to look getting people in here.
Well, it's
like, for instance, are the people giving the talks in here?
I think so. Well, you know what? Where's Saray?
He's outside talking. If everybody could come in and start taking their seat, we're going to be getting started so we can stay on time. So we can try and stay on time, put it that way.
So I should not introduce Chalky. Or Usha.
Okay. Well, I can introduce them.
So there are 3 of us.
Yeah, I don't know where Giovanni is. I know. Maybe you could introduce David's here.
I'll stay here until Giovanni shows up.
Or we're okay either way. Why don't you?
When I do Robin, Giovanni.
Okay.
Or I could just do Robin.
Well,
No,
it'll work.
I'll do Giovanni.
Yeah, why don't you do Giovanni? If everybody could take their seats, please. We're going to start right now. Do you want me to introduce?
You want to start, Rick? You can open the session. Or Glenn.
We're going to start
Yep,
the
go ahead.
geographic atrophy session. I'm Glenn Jaffe, and my esteemed co-moderator, Rick Ferris, will introduce the first presenter.
So the first talk is current approaches for diagnosis and monitoring of GA, and David is presenting.
Thanks, Rick. Appreciate it. Thanks very much to the organizers for the opportunity. I'm a little bit nervous about what I'm going to encounter with this remote, but it seems to be working really well. So that's a good sign. Thank you. So I'm going to talk about geographic atrophy. I just want to acknowledge Alessandro Feo, who is Italian and from Milan, and is doing some really amazing work with us in Los Angeles, and was very helpful in putting this PowerPoint presentation together. So this is just a summary of the talk that I'll give. We're going to look at ways to various imaging modalities that we use now to assess GA. Of course, the traditional way is with color and fundus autofluorescence, but the challenges with using these modalities is that they don't give you a discrete way to measure the margins and also to identify precursor lesions. And OCT has become really a remarkable breakthrough because it allows us a much more granular, much more precise way to measure qualitatively and quantitatively atrophy, and also to capture the precursor lesions that are so important in the evolution of this complication. So traditionally, we used to, we define geographic atrophy, traditionally defined with color fundus photography and fundus autofluorescence as a circular area 175 microns in size with bearing of the choroidal vessels, and autofluorescence has become the important adjunct in measuring geographic atrophy, and other modalities can also be used. Here, Giovanni Staranghi made the identification of the bright and dark atrophy on ICGA to distinguish the profound choroidal atrophy that you have in Stargardt's versus AMD. And we can even use fundus autofluorescence to capture high-risk patterns of progression, the banded and trickling pattern that lead to 2 to 3 millimeter squared progression per year. But really, we don't have a great way to identify earlier stages of atrophy. And some fundus autofluorescence is not always amenable in terms of identifying and capturing the margins quantitatively, and in terms of really capturing precise enough images that in all patients. And because of these limitations, OCT has really been a breakthrough because we can now capture these areas of atrophy more precisely and also identify earlier stages of evolution before the development of GA. And the CAM group, which we'll meet tomorrow or tonight actually in Ischia, was the first to develop this OCT classification system in which we define C-RORA as an area of hypertransmission 250 microns in diameter with overlying photoreceptor loss. And IROR, and here it is on the B scan, and IROR is just the same thing but less than 250 microns. And by using this OCT classification, we can now more reliably capture geographic atrophy and also capture the earlier stages in evolution before IRORA, C-RORA, and GA. And here is just an example of progression from IRORA to C-RORA that was originally published in the CAM paper. And in addition to these more end stage, more later stages of GA, we're now able to capture earlier signs of evolution to atrophy. And Phil very nicely described the importance of hyperreflective foci, also mineralized drusen, perhaps two of the most important features of early onset atrophy. And in this paper by Christine and Bailey, you can see the different forms of hyperreflective foci. Sometimes they can be punctate, other times they can track in the Henle fiber layer in this so-called plume pattern. And these are very high-risk signs of evolution to atrophy, probably indicating RPE breakdown and dissociation. Hyperreflective mineralized drusen can also be readily captured with OCT, and they have a very distinctive appearance with this hyperreflective cap and these punctate hyperreflective opacities above an area of hyporeflectivity that shouldn't be confused with hyperreflectivity that you see in PEDs that are in the form of in the process of breaking down due to hydration, which I'll show you later. And there have been various, and in addition, we've also been able to identify other important risk factors, including STD, dot SDD and ribbon SDD are an additional sign of choroidal or interchoroidal ischemia and another important risk factor in the evolutionary progress to atrophy. And there have been various studies, several from Vasada's group, showing that perhaps the most important risk factors on OCT for progression to atrophy are the hyperreflective foci, the SDD, the calcific drusen, and drusen volume. We wanted to look at drusen volume a little bit more specifically and actually make the distinction between height and diameter. And so we correlated the two parameters with various OCT signs of future atrophy in a cross-sectional survey. And we found that while hyperreflective foci were significantly associated with both the height and the diameter, the height was also significantly associated with many other risk factors, OCT risk factors of atrophy, indicating that perhaps height may even be more important than volume, than diameter, I should say. And we also looked at where these OCT risk factors developed, and they predominantly developed at the apex. And as Dr. Sada mentioned in the previous session, this may relate to the displacement from the underlying choroid, where the apex may be at greatest risk of ischemia and RPE breakdown. Other OCT signs of degeneration or disruption that we've been able to detect include fluid. We now have been able to associate fluid with the non-neovascular stages of AMD, and fluid can be a sign again of RPE pump dysfunction and RPE ischemia, and it typically develops again at the apex and in eyes that have the largest or the tallest drusen with the greatest displacement from the underlying choroidal supply. And what's interesting now, in a more recent study, and we hope to have this published, you can also see evidence of hydration, not just above the RPE in large drusenoid PEDs, but also within the PED. And these hyporeflective areas can be another sign of RPE breakdown and drusenoid PED collapse. Again, an indication of RPE dysfunction. And what happens is that it seems just before these drusenoid PEDs collapse, they enlarge. And there's probably progressive degradative hydration of the large drus or the drusenoid PED, and then ultimately collapse due to RPE breakdown, probably from RPE ischemia. And here's some more examples of hydration above and within a large drus or drusenoid PED, and these three separate examples that we hope to publish in the near future, indicating these large hyperreflective areas within drusenoid PEDs and large drus, a sign of progressive RPE dysfunction. Maybe the most important precursor of geographic atrophy and C-RORA is I-RORA. And in this study, we looked at the progression of I-RORA to C-RORA, and we found that upwards to 30% of patients progressed from I-RORA to C-RORA on OCTB scans, and over 2 years, almost 100% of patients progressed from I-RORA to C-RORA. And you can see that in these two sequences, these two cases that Giulia Corradetti published with us while she was a fellow with Voss and now his research coordinator. Yet the most recent studies have still used fundus autofluorescence, and we now have Syfovre and Izervay that are approved, but they use fundus autofluorescence as the clinical endpoint within the trial, and essentially using one of the furthest along markers of atrophy, geographic atrophy on fundus autofluorescence, and they found this 20% separation both in the Syfovre study and in the Izervay study. But what's interesting is if you look at the phase 2 trials, there was a sub-analysis done, and if you look at I-RORA to C-RORA, instead of that 20%, they found a 50% reduction. And so by using these more granular or earlier stages of atrophy, you can maybe perhaps identify even greater benefits of the treatment. And in this case, in the analysis of the Philly data, it was a 60 to 30% reduction of I-RORA to C-RORA in the Syfovre treatment group, and here 40 to 20 in the Izervay GATHER1 phase 2 trial, indicating that using these earlier mediators of atrophy, you may be able to uncover a more significant therapeutic effect. In the last half minute, I want to show you how we now integrate OCT into our clinical management of patients. This is a patient with geographic atrophy. You can see the fundus autofluorescent changes, the C-RORA with degenerative cysts, and here is the OCT progression of the B scans, which we use in clinic regularly now to judge rate of progression of atrophy in both eyes. But what is very helpful for us is the on-FOS OCT, and using both the subretinal and the outer retinal B scans, you can see significant progression and delineation of the area of atrophy that actually evolved from the cluster of drusen. And that's very nicely captured with on-FOS OCT, both qualitatively and quantitatively. Another patient who has a progression of C-RORA towards the fovea, but you notice that the fovea remains preserved after many years, and in this case we decided to defer anti-syfovre treatment or Izervay treatment because of the relatively slow progression of atrophy. And in this final case, I'll show you a patient who progressed to atrophy on the left, has neovascularization on the right, and actually in the other eye, in the left eye. And by using both on-FOS OCT and OCTA, you can actually overlay the areas because of the dynamic capability, and you can see that the area of neovascularization is actually protective. So again, a way we can integrate these advanced modalities to better assess our patients. I'll leave you to read that summary since I'm over time, and thank you again for your attention.
Thank you, David. Now is Robin Geimer, structure and function correlation GA. I think it's one of the most important topic right now, particularly for the evaluation of a clinical trial. So, Robin, thanks.
Terrific. Thanks very much, Giovanni, and thank you for inviting me to present today. So yes, as was alluded to, I'm going to talk about the structure function correlations in geographic atrophy. First of all, I'd like to introduce my macular research unit group that's 28 years old in Melbourne, Australia, and particularly single out Zi Wu, who really is the brains behind what I'm going to present to you today. So currently we're in an era where we have many clinical trials aiming to slow the progression of geographic atrophy. At the moment, the outcome is a structural outcome, so looking at the area of growth of GA as we've been talking about today. If we look at function, we all have realized that visual acuity does not really comprehensively capture the functional impact as the GA lesions grow. And so people have been using microperimetry to try and capture visual sensitivity. However, to date, the way we've done microperimetry is what's called threshold strategy, where you use a staircase to try and work out exactly how bright a spot do you need to shine on the retina for each spot to be seen. And to do that, it actually takes some time. You have to present a stimulus at different intensity over and over again to the one spot. And so if you want to limit the size of a test to, let's say, 6 minutes, which is really what we aim for, you're really limited to the number of spots you can test. And in this particular Mayer perimetry pattern, there's 37 spots that you can test. And previous studies, particularly big longitudinal studies such as the Chroma and Spectri study, have shown that in cross-section, there is a moderate correlation between the mean sensitivity and the GA area. There's perhaps a stronger correlation if you look at the number of absolute scotomatous points. But in a longitudinal study, there's really only a very weak correlation between a change in either the visual function, the mean sensitivity, or the number of scotomatous lesions and GA area growth. And so this is a quote from one of the papers from Chroma Spectri saying that people with bilateral geographic atrophy do experience a drop in visual function over 48 months. But measures of visual function were not correlated strongly with the GA lesion area, and that indeed this threshold-based perimetry might really have quite limited ability to help with the outcomes in GA treatment trials. And we know this when we look at the pegcetacoplan results. We see very nicely that there was a reduction from sham in the growth rate of the GA area. But when you look at the, this is the mean sensitivity in those trials. This is the Oak study with pegcetacoplan. They were unable, when they looked at mean sensitivity, to see a difference, despite we knowing that there's a difference in GA growth. So we would like to propose a different way to look at visual function changes when there's a patch of geographic atrophy. So you would expect as the atrophy grows, there would really be an increase in really deep visual sensitive losses. Do we really care about the actual number worth of decibels that someone sees? So why not optimize to quantify the extent of the deep visual sensitivity losses? And by doing so, we can actually use much more of our time, our 6 minutes, to actually have many more spots to test. So we think that this is perhaps a very useful concept. So here, we took people with geographic atrophy greater than 175 microns based on fundus autofluorescence and a visual acuity that was 2100 or greater. We did the OCTs on the Heidelberg and the FAF images, and we annotated the areas on the FAF. We then used the Maya to look at the extent of deep visual sensitivity abnormalities, which was basically presenting a 10 decibels stimulus to see, could you see it or not? And 10 decibels is more than 2 standard deviations above what you would expect somebody to see. And to do that, you can get 208 locations within an 8-degree radius, and we in that same duration of 6 minutes. And in this case, we did 2 tests on each patient. We also sort of allocated a category to where these spots were. So for example, the blue spots here are in what's called the outer non-lesional zone. So they were all more than 500 microns from the edge of the GA lesion. There are ones in the junctional zone. There are ones that overlap the edge of the GA as seen on the fundus autofluorescence. There were ones sort of slightly inside the edge, and then ones that were further inside the edge. And so what we were interested in is looking at the percentage of the region sampled by the microperimetry that was also occupied by the GA. And hopefully you can see here, we looked at the percentage of missed locations, or PLM, and a very nice association with the region of GA. So as the GA increased, there was this beautiful linear association with the number of non-seeing points. And then when we looked more locally, just at a particular lesion, and as you might expect, if you look here, this is here the distance from the actual edge of the geographic atrophy. So the probability of missing a spot was very low, a long way from the lesion. But as you got closer, so these ones are at the GA margin, the probability of having a deep scotoma was greater, and then clearly inside the lesion, it was greater. So there's both this local and global association with the number of spots missing. And this is just 2 examples then of change over time. So in the first example on your left, you can see that over 21 months, this is over, there was this increase in the GA area by about 18% from baseline to 21 months. And very similarly, there was a 17% increase in the number of lesions missed. And in the second location, the GA growth by 20%, and again, there was a 20% increase in the number of deep scotomas. So a very nice correlation between the two. So in terms of this defect mapping, there really was a very strong global structural functional relationship between deep visual sensitivity losses and the extent of GA. And our findings suggest that this strategy would be very useful when you're trying to capture treatment effects, and that we would certainly recommend that perhaps we think about using this strategy rather than the typical threshold-based microperimetry when using treatment trials for geographic atrophy. And then secondly, well, what about what we're talking about, perhaps a bit more interesting, is what about those smaller lesions, so the rawer lesions? So in this case, we wanted to know what the function was over patches of C-RORA. And here we sort of concentrated the 37 spots that would be in your normal Mayo perimetry down into this 1,000 micron diameter spot placed over the top of the Aurora lesion. And so here we had a prospective study of people over 50 that had Aurora and had vision 100 or better. And they had completed 2 microperimetry tests. And so we tested these 37 spots in this 1,000 micron area. And the final cohort, we had 171 visits from 60 eyes of 35 patients, and we had therefore 342 individual microperimetry tests. And what we did then was we measured on the autofluorescent, the OCT image, we actually measured the size of the hypertransmission, the size of the RPE attenuation disruption. We also measured the complete RPE loss, and then the subsidence or wedge. And we also then had the FAF image and the color fundus photo images. And so we were interested to know the association between the number of these locations that had a deep visual sensitivity defect, defined in either 2 ways. They either had an absolute scotoma where they had 0 decibels or less than 10 decibels, so not quite as deep scotoma. And we wanted to know the correlation between having 0 twice or -10 twice. And what I want you to take a look at here, if we just look here at these columns, these are the amount of hypertransmission you need to see a difference, a statistically significant difference in the number of lesions that had -10 decibels or 0 decibels compared to Aurora. So Aurora is the reference. And hopefully you can see here for the hypertransmission, it wasn't until you got over 500 microns that you started to see a significant increase in the number of scotomatous lesions in both the less than 10 decibels and less than 0. If we just looked at attenuation or disruption of the RPE, we never got to a point where there was a difference between Aurora and C-Aurora in the number of those lesions. But if you had complete RPE loss, then over 250 microns of complete RPE loss, you could see a significant increase in the number of spots that had either less than 10 or less than 0. And so finally then, that study showed that the presence of hypertransmission more than 500 microns or complete RPE loss more than 250 microns independently was associated with an increase in the number of those deep visual sensitivity scotomas compared to those without those lesions. And not all C-Aurora lesions were characterized by that deep visual sensitivity loss. Only those with hypertransmission more than 500 microns or complete RPE loss more than 250 showed a significant difference in deep visual sensitivity compared to Aurora lesions. And so we hope that this provides that much needed functional evidence to help define what we would define as a atrophic lesion that was associated with deep visual sensitivity defects. Thank you very much.
Thanks, Robin. The next presenter is Usha Chakravarthi, and she's going to present on pegcetacoplan update, Gale, and latest on safety.
Thank you. Here are my author disclosures, and of course, I want to thank the panel for allowing me to talk here today. I really don't need to go much into GA because it's been talked about much about already, but the recent trials using inhibitors targeting the complement pathway have been shown to reduce the rate of GA expansion, and I'm going to concentrate on the GALE study, which is the open label extension of the pivotal Oaks and Derby trials. So just a brief bit about complement inhibition and what exactly the complement pathway constitutes. So there are 3 ways in which complement can be activated. That's the classical, the lectin, and the alternative. And all these 3 converge onto C3 and C3b. It's sort of a central molecule which has to be activated in order to generate C5b, which is the molecule, which is the end result of this pathway that destroys cells, causes lysis of the cells. And in geographic atrophy, it is assumed that that chronic activation of the alternative pathway leads to cell death and GA. This, of course, formed the basis of the Oaks and Derby pivotal trials, and I'm just going to show you the reduction in GA growth relative to sham. So this is initially data from month 12, and PN stands for every month, and PEOM for every other month. And what you observe is in the Oaks, there was a statistically significant reduction even by year 1, but not so in Derby. But over the 2-year period, the curves in terms of GA area growth reduction diverged even more, and both trials were statistically significant, showing a reduction in GA growth of approximately 20%. Going straight into the Gale open label extension study, as you will see here in the box, if I can make this work, those are patients who were randomized to pegcetacoplan monthly, were continued into pegcetacoplan monthly, continuing in the same regimen every other month into every other month. But what happened to the sham arm was a switch to treatment, and they continued again monthly or every other month. So various planned analyses were carried out, and these continuously treated eyes were compared to sham, both true and projected sham. The sham crossover was compared to the projected sham, and the untreated fellow eye was compared to sham and projected sham. I'll just give you a brief example of the estimates of the RP cells saved as well, and talk you through the safety data through 36 months. So in terms of the patient disposition, approximately 3/4 of patients who were originally enrolled entered Gale, but of course those who in the first year of Gale, there was only a few patients lost to follow-up, 92% retention in the first year of Gale. Comparing all study eyes that were included in Gale in terms of the reduction in GA growth after 36 months of continuous pegcetacoplan, you can see that there's something like a 30% reduction in year 3 in the every month, and a 24% reduction in every other month. Non-subfoveal GA, the reduction in GA growth after 36 months of continuous pegcetacoplan, when you look at the subset of about 286 eyes, the reduction in GA growth was even greater, and around 2.5 millimeters squared of retinal tissue appeared to be preserved in the every month, and just under 2 millimeters squared of retinal tissue preserved over the 36-month period in the every other month arm. When you examine the GA growth in untreated fellow eyes, these are eyes that were never treated, it tended to overlap with the sham arm. There were adverse events in the study eye over the 12 months, and the most important, obviously, was the presence of the development of exudative AMD, and this was seen in the monthly arm as well as the every other month arm, but the rate of development of exudative AMD slowed during Gale and was only between 2 and 2.9%. Other severe adverse events was the most important one, obviously, is intraocular inflammation, and there was a very low rate, as well as the patients who were in Gale did show the development of optic neuropathy. In Gale, there were no study events of occlusive or non-occlusive retinitis or vasculitis, but it is important to note that there were other events had been reported in real-world settings, and I'm going to cover that in a moment. So the REST Committee of the American Society of Retina Specialists looked at all the cases of intraocular inflammation that developed in patients subsequent to introduction of pegcetacoplan, now known as Syfovre, into routine care. And in this committee has published its data in the ASRS. This is the original manuscript by Andrew Witkin. Recommend that you read it. And what the REST Committee have shown is, I'm going to go back, is that occlusive retinal vasculitis was reported at a rate of around 1 in 4,000 per patient and 1 in 10,000 per injection. So to conclude, 12 months after release from protocol, continuous treatment with pegcetacoplan reduced GA expansion compared to sham during the extension phase by 35% in the monthly and 24% in every other month, 42% and 28% in the subset of eyes that were non-subfoveal GA. And although occlusive vasculitis was not seen during the pivotal trials or GALE, data accrued after introduction into routine care do indicate rates of occlusive vasculitis of 1 in 4,000 per patient, 1 in 10,000 injections. So cautious and targeted management with appropriate counseling will be needed if current complement treatments are going to be considered for patients with GA. Thank you for your attention.
So the next talk is on avacincaptad update to your outcomes by Giovanni. And it's an opportunity to thank him for 10 years of developing these meetings and all the work that has been involved. We all appreciate it.
Thank you, Rick, and thank you to all. First of all, I have to thank also Raminta Dayoni because actually it was the guy who has to present this paper. So again, just a good reminder from him. So these are my disclosures. And as you heard from Usha, the pegcetacoplan, it's inhibiting the C3 complement, and either way or avacincaptad pegol is actually inhibiting the C5. And all are, a complement, and the inhibition of complement seems to work with the patient with a geographic atrophy. The inclusion criteria is a little bit different from the pegcetacoplan, and in particular, there are no area of GA that should involve the fovea. And you heard from a previous presentation that there is a little bit difference from the patient that has atrophy outside the fovea comparing the ones that have the fovea involved. And here just an example of a study that where the patient were included and the patient were excluded from the clinical trial. Gator 1 was a phase 2-3 study, but the Gator 2 is the phase 3 study. And first year, the patient were randomized to every month injection or placebo, of course. But the second year, the ones that has an injection were re-randomized to every month or every other month. And of course, the sham continue with the sham. These are the baseline characteristics that seems very well balanced between the two groups. And the other interesting thing is that the data. The results shows that there is a decrease in the 12-month that actually was the data that was used for registration of a drug, like the pegcetacoplan. FDA wanted just 12 months. So these are the difference between the Gator 1 and the Gator 2 study. And but as you see, there is a reduction comparing, the treated and untreated patients. These are the a particular, representation of a visual function. In particular, you have not to think like the ones that you usually see with visual acuity, but this is a risk of probability of changing or preserving visual acuity. And so this is why, the patient with treated with lower but not the ones that sham, of course. This is at 12 months. But then we will see the data on the 24 months. And as I said, the 24 months was re-randomized. And again, the most interesting thing is, of course, the area of atrophy. And you see that, again, also the 2 years, you have an advantage in the patient that were treated comparing with the sham. What about function? And you see that in that case, the risk probability of decreased visual acuity seems that it's merging after 2 years. Why this happened? There are a lot of explanations about that. And visual acuity, as you heard from the Robin presentation, is not probably the best way to assess the function in these patients. And again, if you divide it by 10 letter persistent loss or 20 letter persistent loss or 25 letter persistent loss, it seems that there is an advantage. But again, I think, there are two explanations why it's, visual acuity is not the best test. There are some anatomical and some functional. For anatomical, we have to think, and that it's a post-hoc analysis that was used by the PET Cetacoplan, but Ursula Schmidt-Erfurt show with the AI the possibility to identifying the photoreceptor loss and the RP loss. And you see that the photoreceptor loss are preceding what we actually see usually with the autofluorescence, that is RP loss. And both for the study eye and the fellow eye. And of course, the data was showing that in that case that the photoreceptor loss was slowing down if you treat the patient. But because of that, if you think when the drug will work, I just tried to explain with the drawing, the dark spot is the area that RP is lost, and all the yellow area are the ones that you have already hyperfluorescent. And probably, as you heard from Christine's presentation, the increased autofluorescence around the geographic atrophy can be or an accumulation of RP with more lipofuscin, or just the initial loss of the photoreceptor, because you see much better the fluorescence that comes from the RP. So if you think what we actually have to expect from the drug, probably the drug will be visible, the effect of the drug will be visible only if you go much farther from the area of atrophy. And this is why what I'm expected, and actually the data from atrophy will show, you actually do a better steady state after 2 years, and then Gale, for example, shows such a things. So why visual acuity is not the best way is because probably we are not able to, exactly concentrate in the area that we can actually preserve. And then there is also a functional explanation. And visual acuity, again, is not the best way. If we think, patient with a GA can increase the size of the lesion, but the most important thing is a preferred retinal locus, and it's already demonstrated by many studies that if you have a not stable PRL, it will be much more difficult that the patients will improve or stabilize vision. The other interesting thing is that a patient with PRL can be very influenced by the psychophysical situation. So if the patient is not convinced of what he's doing, it can actually be not very or is depressed. And you know that the depression in AMD is the third cause of, AMD is the third cause of depression. You can actually think that the patient cannot collaborate when you do the visual acuity test. And just to show what Marco Morales showed many years ago, the visual acuity and PRL are very well correlated. So if you have a PRL much farther, from the center of the fovea, the visual acuity, of course, will be low. And again, the fixation stability is another issue about that. So what about going back to the from either way study? What about the ocular side effects? And you see that there are not so many, intraocular pressure and choroidal vascularization are the same issue. And if you think the choroidal vascularization can be indirectly a sign that the drug, the anti-complement drugs are working, because if you have a cells that is not very healthy, of course, you are not having the possibility to in a ischemic area to produce a VEGF, and if you reduce the inflammation in the cells, that the cells can actually work much better, and indirectly you can see. These are the choroidal vascularization that you see at the year 2 of the Eiserway, and again, it's about from 5 to 7%. But also, if you look at the sham group as a quite high number, and again, it depends probably on the type of lesion that you have, probably more type 3 neovascularization. So the adverse events, for inflammation, it's almost 0. It's very, very low, and but not all of them as a visual threatening situation. So in conclusion, both monthly and every month at year 2 seems that works. Of course, the problem, again, if we consider the function, it's still a challenge. And so in that case, for us in Europe, we will be a little bit afraid that EMA will still discuss the idea that even if you have a decrease in growing of the atrophy, you will not, having, based on the visual acuity, a very good effect for the patient. I actually do not agree personally, but I hope it will be possible to demonstrate it in some way. Thank you.
Thanks, Giovanni. The concluding presentation in this session will be given by Carl Chalky, who's going to be talking about next in the pipeline for GA, Annexon's ANX007 and Avicida's AVD104.
Well, thank you, and thank you, Glenn, and I want to thank all of the organizers for this great meeting. And I'm going to run through some interesting new drugs that I've been involved with in the clinical trials. Let's see if it's, where do I point this to get going? It works now. Okay, great. So here are my disclosures, and I'm definitely telling the truth. Yes. So that got to make sure you tell the truth in this environment. Otherwise, something bad will definitely happen to you. So I'm a consultant for both Avicida and Annexon. So here's the first. So this is the anti-C1q Annexon approach. And I think what's very important to understand with both of these drugs that I'm going to be talking about and the approach is that it's something different. And the idea here is that C1q, while it's considered one of the initial complement drugs, it really has a different mechanism of action. Yes, it initiates a complement cascade, but what's really important to understand is that it gets activated at the photoreceptor synapse. So when you're using, or when we're using this drug that inhibits C1q, the target, unlike some of our other drugs, is really being focused on photoreceptors and synapses. And this is really an important concept, as we will see some of the data that came out of the phase 2 trials. And there's plenty of evidence that, in fact, when you inhibit C1q, it's this prevention of photoreceptor synapse degeneration that's occurring, and that could be a very invaluable aspect as we go into the clinic. So the trial was the Archer trial that was completed about a year ago, and essentially the idea was to give this drug either every month, every other month, or no drug, and compare the results. So the important point here was that the visual acuity results is what was most dramatic out of this study. So here we are at 1 year, and what we're looking at is the number of patients who lost 15 letters or more during that 1 year period. And you can see that in the patients receiving every month or even every other month, there was a very significant decrease in the rate of vision loss in these patients. And in fact, that loss every month was further increased, suggesting that the more drug you gave over time, the more benefit you had in terms of preserving visual acuity. And in fact, this also was seen in both the foveal and non-foveal patients. So it was irrelevant where the lesion was. And seeing this photoreceptor kind of effect on these patients. And interestingly, for the first time, we also saw an effect on low luminance vision, again, suggesting that all of these effects was happening at the photoreceptor level. Of course, what was most interesting is the study was designed so that after a year, you stop the drug. And what happened when you stop the drug, those patients you can see here at the bottom who were on the drug, once you stop the drug, they started losing vision again, suggesting that there really was a preventive effect of this drug on photoreceptor loss. Now, what was most interesting was that the outcomes that I had been involved in developing very early on, which is what we've seen all along, this decrease in the rate of fundus autofluorescence loss was not shown with this drug, interestingly. Again, not unexpected because, again, the drug is targeting the photoreceptors, not the RPE. But what was most interesting is when you started looking at the EZ area, which is a direct indicator of photoreceptors, there we saw a preservation with drug and a reduction in the rate of EZ loss areas. As you can see here, almost a 30% loss of total or what we call attenuated EZ loss. So again, suggesting that in this case, the appropriate anatomic outcome was EZ preservation, not necessarily fundus autofluorescence. Again, very little side effects of the drug, and there's now an ongoing larger phase 3 study along with an extension of the Archer 2 study that's going to be started. So I'm going to transition to a similar kind of newer drug, and this is the anti-Siglet drugs from Avicida. The concept here is these are actually nanoparticles which are coated with an anti-Siglet molecule. Then the Siglet molecule, or the receptor for the Siglet molecule, is very interesting. It has two areas. The complement factor 8 is one of the factors that Siglets bind to. The other thing that's very intriguing about the Siglet receptors is they're overactivated in macrophages. And we know that in macular degeneration, there are a lot of inflammatory cells that come in and are around these areas of geographic atrophy. So now we have a drug that's targeting this specific receptor, the Siglet, both on CFH and on the macrophages. And again, the idea is that by downgrading the inflammatory response in a very specific manner, we might see that effect of anti-inflammation, especially on macrophages, which we know may have an effect similarly on photoreceptor degeneration. So in this trial, this was a very early trial. This was only a phase 1 trial. And here what happened is we simply looked at a dose escalation after one injection, then followed these patients for several months after this injection. And again, with one injection, you wouldn't expect, and there really wasn't any significant adverse effects with the drug. But what was most intriguing was that even after one injection in these patients who had primarily foveal lesions, when you started looking at the dose response, and you can see that over here at the very end, you actually started to see patients who gained vision when they were given this drug, again, suggesting that our approach to treatment of geographic atrophy needs to be kind of rethought, and that yes, we need to target both the loss of RPE, which we have now with our anti-complement drugs, but that we also need to be targeting photoreceptors and preservation of photoreceptors. And in this case, through a anti-inflammation aspect, we're starting to see the potential of having an improvement of vision in some of these patients as we give them their drug. What was most intriguing, again, you see a difference between the study eye and the fellow eye and the number of patients or the amount of vision that was gaining in this very short-term study. And what was interesting, there was an effect to some degree on the dark area of fundus autofluorescence in those patients receiving drug. What was most intriguing, though, was also the area of increased hyperautofluorescence, which we see around the GA lesions. And with this drug, there was a change, again, suggesting that there's something going on in this region around the dead cells where there's clearly inflammation, and perhaps targeting the Siglet receptor and the macrophages can alter that peri-GA environment, which may again be beneficial in terms of visual acuity. And again, even here, the EZ area was also some degree of preservation. So now there is an ongoing phase 2-3 study, which we're involved with, very exciting. And we again can start to see what is the effect of these added therapies potentially on photoreceptor preservation, targeting photoreceptors, and how we can keep them alive in this disease so we can get not just some anatomic benefit, but potentially visual acuity benefit as well by targeting these different receptors. Thank you very much.
Thank you, Carl. I think we will discuss in the panel discussion just after. We have to ask to Phil to tell us about the real life with using pegcetacoplan. So if you can, if you can come and.
So Giovanni and I were talking. I don't have any slides. I was just going to have a discussion.
Yes, absolutely.
And we've treated quite a few patients. We're up to about 140 patients treated with Syfovre, and this is after approval, and just a real-world experience and some words of advice. I'm happy to answer any questions you have.
Yeah. First of all, how the patient, like to be injected every month.
So what's most important is you have to talk to your patients and set expectations. This kind of reminds me of the early photodynamic therapy days where vision improvement wasn't to be expected. So you really have to sit down with your patients. And we're doing a natural history study. It's called Impact Swagger. We've been doing it for a number of years. And I've really had the opportunity to sit down with these patients. And you have to educate them about their disease. You have to instruct them to measure the vision in both eyes separately. But more importantly, they have to embrace what's going on with their eyes. You just can't see atrophy and start injecting. You have to make them appreciate, and I say this over and over again, your vision will not improve. Your vision will not improve. And of course, patients come in and say, my vision's not getting better. Your vision will not improve. I even treat my mother-in-law, who's 92, bilaterally, avid reader, atrophy encroaching into her fovea. I could show you the pictures of it progressively moving in. And right now, she's preserving her vision, and she's coming in bilateral injections every 4 weeks, which, by the way, is really hard to maintain in patients in any protocol every 4 weeks because life happens. They have events, you have events, and sometimes you can't see the patient. And that's why you have the flexibility with Syfovre. And that was the first drug approved in the US, and we had experience with it in clinical trials. So we started using it, and what's nice is you have that flexibility of 4 to 8 weeks, and I try to start every 4 weeks.
Practically, do you inject both eyes the same section?
Okay, so how do you start? right? After you educate the patient, and there are many times I say no, and I want the patient to come to me telling me that they're losing vision. I never treat a patient with either wet or dry macular degeneration unless they're experiencing vision loss. You know, a patient can come in with exudation, but if they're not experiencing vision loss, I'm not going to treat them. They need to understand the disease. They need to appreciate what's going on. And the same thing is true for dry macular degeneration. So after educating the patient, experiencing vision loss, the general consensus now, because of this rare risk of occlusive vasculitis, which we have not seen, we haven't had any problems, 140 patients starting off every month. It takes some effort. Don't assume this is like an anti-VEGF injection. It's a larger volume. The drug is more difficult to draw up. Take your time with the injection. When we decompress the eye, we soften the eye first. I haven't had any elevation of intraocular pressure of any concern because we use a cotton tip compression, which we've been doing for years. I have a secret, and you don't tell anybody. I inject 100 microliters of every drug into the eye. Okay, because it's there. I'm not going to throw it out. And based on our
we want to
previous
add
experience-
that's off-label.
Off-label,
to
right.
be
But we have a technique that we've been using since the CMV retinitis years with foscarnet against cyclovir, which is where we do 2 20-second compressions with a lidocaine tetracaine soap swab, and we just push at the injection site. And then you drop the betadine. So the bottom line is, the consensus is start with the worst seeing eye first. If there's going to be a severe inflammatory episode, it's going to occur usually with the first injection. Haven't had it. And then after you inject the first eye, then the patient considers both eyes being injected or just treating the better seeing eye. I don't care if it's subfoveal or non-foveal. If the patient is experiencing vision loss, and there's a lot of vision to preserve in subfoveal lesions, there really is. And that PRL, very important. Maintaining that PRL makes patients' lives wonderful. And we presented our results at ARVO. That's how this discussion started. And we have a population, 140, but only 101 have 9 months follow-up, and 70 have 6 months follow-up. And we're seeing a 40% reduction in the growth rate of GA. But remember, this is our population of patients that we've been following, and we're comparing pre-injection to post-injection. These aren't really large lesions. These are lesions that should continue to progress. And it's a bit of cherry picking because we're picking cases that are actively losing vision. 40% reduction in the annualized growth rate so far.
Don't you think, sorry, don't you think it probably, if you can bring, don't you think that doing pre and post, there is a bias to the fact that the lesion can grow, and so it could be less than 40% because some of those has already reached a bigger area?
That's why I mentioned these aren't really large lesions, and we've done natural history studies of growth rate, just using the square root transformation, right? But these patients are not end-stage disease patients. I'm not treating end-stage disease patients. These patients have lesions to grow.
So, Phil?
Go ahead with your question, Bailey. So we're going to just make a changeover to get my panelists. Rick, you stay.
Okay.
Glenn gets to wait till his next panel assignment, and then I'll call up Sergio and Emily. And who else am I? Who did I forget? I'm so sorry.
Well, I'll just ask my question as you guys are getting set. Phil, if you've just seen the patient for the first time and you don't have any history, looks like they might be a good candidate, what do you do in that situation if you don't really know the history?
I never treat a patient the first time I see them. I send them home. I make them monitor their vision, consider the procedure, and sometimes I don't inject them when they come back. But they have to experience vision loss, and they have to truly embrace.
Well, let's say they tell you I'm losing vision, but you don't have any, evidence of progression yet. You you just go, would you want to see progression of the atrophy before you treat?
It's always great to see anatomic progression for confirmation, but I just don't inject the first time. I actually send the patient home with the information, and I have them talk it over with family and friends, because it's a big inconvenience for everyone in the family, particularly for my patients. Many of them have family members bringing them in, that they're going to have to come in every month initially with some wiggle room for the injection.
So, okay, so thanks very much, Phil. We added Phil to the panel because he's got the most.
I have a final question for Phil. I'm here.
Okay, Francesco, please, go ahead.
Yeah, Phil, is it important to start with your mother-in-law?
Actually, she refused to be the first patient. Yeah. You know, what's interesting, I injected my father-in-law many years ago. And to show you.
Your father or your father-in-law?
My father-in-law had wet AMD, and I injected him. And I remember her saying, "You're never going to do that to me."
So I think Francesco's getting at a difference between your mother and mother-in-law.
And I like my mother-in-law. I do.
I can't believe you.
Francesco's
I can't
main
believe
question.
you. It's impossible.
So,
92.
Okay.
So we're going to have a brief, we're going to abbreviate the panel discussion a bit because we're over time. But I just have a few questions for our esteemed panelists. These are my disclosures. But these are expert panel. We've added Phil because he has a lot more real world experience, I think, in terms of using the drug. But I actually want to take a step back because obviously we were in the past where maybe we had no treatment, but maybe that's not, and again, obviously we already heard about the data on the drugs. So what I want to skip, and we heard about things in the pipeline. I mean, we weren't able to cover all of the different agents, but this is sort of a summary slide of some of the different agents that are being considered. But really, I guess what I want to ask the panelists to weigh in on. So this is, I'm going to share a case of mine. 81-year-old woman had previously treated macular neovascularization, had a scar in her right eye, and now she's complaining of problems in her left eye. And you can appreciate even on the ONFOS OCT image there that there is an extensive area of injury in the right eye. In the left eye, you can see there's a atrophic lesion which is still outside of the foveal center, which explains what her symptoms are. And so I guess actually my first question, and maybe this is to Emily or Rick, I mean, patient with advanced AMD in both eyes, micronutrient supplements for this patient?
Well, I think that's a very good question. That's often asked of us. And in the past, we've looked at our AREDS1 and said a certain visual acuity, these still seem to be effective. But more recently, we've actually looked at a post hoc analysis looking at supplements for patients who already have geographic atrophy. And these are cases who don't have the fovea involved. And when you look at that and looking at the progression rate towards the fovea in those with and without treatment, in the randomized, this is the randomized, post hoc randomized evaluation of those in AREDS1, which was vitamin C, vitamin E, beta carotene. And if you remember AREDS2, we actually eliminated beta carotene. We actually had randomization of lutein, zeaxanthin, omega-3 fatty acids versus the actual AREDS1. And in both cases, we actually saw a reduction by as much as 35% in the progression of the geographic atrophy towards the fovea. These are post hoc analysis, and we needed to do some more on this. And in actual fact, I've spoken to various funding agencies who are thinking perhaps we need to look at that in the future, do a longitudinal study. So in the past, I would say, no, don't do it. But now there's suggestion that there might be some helpful aspect. The other aspect, in addition to supplements, is, of course, the diet. We are what we eat, and the diet has certainly been important, looking at both the nutrients and the diet itself. But we also found that there was a reduction in the progression of geographic atrophy. Patients who already have the disease, if your diet is of a certain amount of, especially high adherence Mediterranean diet, there was also a treatment effect. So I think don't underestimate the importance of diet and the supplements.
And you might reduce, these patients can still develop macular neovascularization, so might be of some benefit there too. So, okay. All right, great. Good. Thank you for that advice. So again, getting back to this patient, again, I want to point out, if you look at the OCT, you can see why the patient may be having some visual difficulties. Even though the atrophy is outside the fovea, there does seem to be some areas of EZ disruption. And so I guess, Carl, maybe I'll ask you the next question. What information would be helpful to you in deciding on pursuing treatment for this patient, given our options?
Well, again, I think when Phil was talking about the cases that he's treating, he has a very good natural history of his patients. And I think it's very important to get some idea of what are the aspects that will be driving this patient's GA into the fovea. Because it's different if it's just growing outside. You may want to say, look, it depends on, are you really growing into the fovea? And also, what are the characteristics? Do they have increased rim hyperautofluorescence? Do they have STDs? Do they have things which really suggest are driving? And from what you're seeing here before, this was 12 months before that, 4 months before that. So, this is a multifocal lesion. It's probably going to be growing a little bit faster. So, you can begin to understand where you can begin those conversations. I'm also a believer in the kind of nutrient approach, at least initially, if they haven't been on it, to try to get some sense of what exactly, what else we can do to control the rate of growth.
Phil, go ahead.
And I just would want to add, these patients are symptomatic long before they enter the fovea. So even though this lesion is non-foveal, you could see it from the OCT, the ellipsoid zone affects the reticular pseudodrusen. These patients are having visual problems that don't necessarily involve the fovea.
I think that's an important point. And one of the things I was going to say is that I wouldn't consider, my own opinion, treating a patient without having some information about their progression, because I think it's very hard to keep these patients on therapy because they don't perceive a benefit. And I think that's where, the information is. So maybe, I don't know if Robin and Sergio or Rick haven't weighed in yet, but their thoughts on the kind of patient you would consider. We can start with you, Sergio.
We have these conversations with the pool of patients that we have in our clinics for potential recruitment in clinical trials. And it is hard for them to understand the benefit without visual loss. There are two ways to talk to them, especially in a case like this where they lost the vision for whatever reason. They are very concerned. One is on the scan. The widespread EZ loss is something that we can make them understand and what it means and get that point. In addition, if you are lucky enough to have microperimetry, I will do a microperimetry. And if I find that one of the central forces involved, this is something they can see, that's not enough. You also want to see changes over time. So we can bring them back in 6 months. And if they are progressing and they already experience symptoms like, they would often say, I'm struggling to drive at night. And they can maybe put all these things together and they might consider treatment. I doubt it will go for monthly, but every other month is a possibility.
Rick, and then Robin.
So I have an epidemiology sort of approach to think about this. And I think it's clear that there are benefits in these eyes that have already developed geographic atrophy. But I think we need to be thinking about a little bit upstream from that. And the dark adaptation information we have suggests that there may be widespread rod loss in these eyes. And reticular pseudodrusen may be an example of diffuse disease that almost surely is going to eventuate. And so these treatments are good, but we need to be thinking of treatments that are upstream, preventing this from happening in the first place.
For sure. Robin, and then we'll let Bailey.
So I guess I was just going to say similarly that some of the symptoms may not actually be the atrophy, but the reticular pseudodrusen and the dark adaptation, and they often stop driving at night long before they get geographic atrophy. So not all symptoms are necessarily due to the dead RPE. in Australia, we've just revised the referral guidelines to try and get optometrists to send patients. We don't have the treatment yet. It's probably going to be another few months at best to actually do the imaging now while we're waiting. So we've actually actively changed the referral guidelines so we can do that.
Just one other comment related to what Phil was saying. One of the things that I think has been striking over 30 years of looking at patients with geographic atrophy is how linear the growth rate is. The rates may be different depending on the subgroup of patients you have, but whatever the group, almost all of them have been absolutely linear. So his comparison with the growth rate previous to starting and after starting is not a silly thing to do.
Yeah, well, that's why I think it's vital. That's why wouldn't consider treating someone without having some previous data, because I'm going to get to it about how to motivate someone to stay on therapy. But maybe Bailey and then Phil, you can make your comment.
Vas, I saw a couple drusen on your example, but let's say there was just SDD or reticulocytic, no drusen. Would that influence whether or not you would treat that patient with these drugs?
So any panelists want to comment on if the patient had, you didn't see any regular drusen, but they had, STD. Emily, obviously, in the AREDS group, they've shown that patients with STD, they're at a higher risk category in progression in general, but we know that the atrophy may progress faster in the setting of STD. Maybe that's what Bailey's getting at, but.
Do they have AMD?
The same well, that's a whole different discussion. Well, we're not going to talk about that in this panel.
I know, but if there's no drusen, is that complement mediated?
And Greg would say there's chromosome 1 and Greg would say chromosome 1 and chromosome 10, and one is complement.
So I guess what we haven't heard is from either of the two approved therapy trials, specifically whether such patients were allowed into them, right? They had to have atrophy associated with AMD, but I don't think that we've heard clarity on how many cases had primarily STD driven or all STD or what have you. So I guess we don't know, but maybe that data could become available.
You wouldn't know that because they had big patches of atrophy, by which time the STD has gone away. So
could be.
You wouldn't know what was there in the first place.
So do you think so Bailey is asking, and then Usha's got a question. Bailey is asking, do you think that an eye has largely STD-driven disease, maybe that's not complement-mediated? Do you think that that's a possibility, Phil?
You have to look at the totality of the data. I look at choroidal thickness. I look at the subretinal drusenoid deposits. You usually see a drusen here or there. A lot of the times they've resolved. But if the patient's progressing, and it's got a thinner choroid, and you have STD, and it's the right age group, it's pretty much AMD.
Usha, you might have one of the last questions. I think Rick has a comment.
Very quick. Basically, in the Oaks trial, when they did microperimetry and they looked at the central 4 points and progression to absolute scotomas, you could see that there was a very clear relationship with visual acuity loss. So coming back to the point that Phil was making, that there is depression of visual function. Patients are symptomatic, even though they may be in the situation where you cannot find a very clear-cut association between visual acuity and GA progression. So the point I'm making is that when you look at the central 4 scotomatous points, as central 4 retinal sensitivity loci in microperimetry, you see a clear relationship with visual acuity, and then progression to absolute scotomas was associated with much greater drop in visual acuity. So that kind of ties in with the discussions you're having about the function and structure associations.
So I think Rick and then David, and then we're going to probably have to break for lunch. So sorry about that.
Well, to Billy's point, if they have no drusen at all, I think you've got to ask yourself whether they have AMD. And particularly central areolar type lesions, it may not be AMD at all. So whether those lesions benefit from treatment, who knows?
One thing that hasn't been discussed in this case, if you look at the original lesion, you don't have it up, it was at the same central location. It did not encroach. It was right at the edge of the central ring. All the growth is taking place almost in a curvilinear fashion outside that central ring. You see the the first lesion is right at the central ring. So I'm not sure that I agree that it's always entirely linear. That central foveal ring is clearly resistant, and we know that. And in this particular case, I would argue that especially if the patient is over 90, it could be another several years before the patient loses vision, central vision.
So that's one of the reasons I asked, do other factors matter? So you brought up age, but Phil?
Remember, it's not about foveal and non-foveal. And Rick came up with this term at Reimer years ago, dead retina walking. The atrophy is clearly dead, but you have this zone of dead retina walking that if you can slow down that photoreceptor loss and maintain the patient's quality of life.
On the other hand, you're talking about a 92-year-old person, you're asking to come in every 4 to 8 weeks, and the central vision is clearly going to be the most important function to her.
Depends on the 92-year-old, and you have to talk to the patient. And one of the points that Rick made also is the linearity of growth. These multifocal lesions, when you incorporate the perimeter and you look at the growth, these lesions are continuing to grow. The trouble, David, is that if you don't, and it grows where you don't want it to grow, it's too late.
The problem I'm having is I am talking to these patients, and I'm kind of surprised that you have so many you're treating, because when I talk to them, none of them want the treatment. And I'm just simply telling them the data. So it's a surprise to me.
So we're going to have to continue that conversation into lunch. Giovanni's given me the sign. So we're going to break for lunch, and we do need to start back on time. So we're going to start at 1 o'clock. So that's going to, we're about 5 minutes past right now, our scheduled stop time. So we're only 5 minutes behind, but.
One thing, the fellow eye, don't forget the fellow eye. That's very influential in what patients need.
Okay, thanks everyone. So try to come a few minutes before 1 so we can get started promptly at 1. Thank you.
If you look at the actual area, it's not the same. The growth rate's the same. But towards the center, towards the center, that's what it's protected. Yeah.
That's why I think there might be some role in this central testing.
Yeah.
So, I got the analysis the central, the first point, it's 1 degree from the border. So if there is a dysfunction there is something functional already very close to the
Right. Well, the other thing about the genetics is that if you look at GA that's already formed, I think there's different genetics that causes the GA. But then when you look at this progression, arms 2, chromosome 10 is really important. If you have chromosome 10, your rate is much faster. And why is that? Is it because they have RPD or SCD because they have chromosome 10? We looked at that. The mediation, it's independent. So the arms 2 and pseudogenes.
No,
increases the risk.
It doesn't go via the
No, it doesn't. It actually it's two separate risk associations with progression that's much faster. So I think that's really intriguing as well too. What's driving it?
The arms is also common.
It is common. That's what we thought. Maybe it's mediated through that. It did not show that. It's totally independent, So that's kind of interesting. And then when we looked at what we call phenotype versus genotype, that we call the PHEROS, that was Fakia who did the work. When we look at that, complement factor H was really important for drusen formation. And then ARMS 2 was really important for people who got hemorrhage and visual acuity loss. So there was almost like a two hit kind of thing. Something happens there's two roots, something happens for the actual etiology of the disease. And then when it comes to progression to the later disease, something else happens. It has to happen. Like the two things have to occur, somehow. We haven't looked at them.
So I have a question about this reduction in growth rate toward the center, because I think that's really interesting. And I wasn't sure whether you had looked at both AREDS and AREDS 2. And was AREDS 2 more effective than AREDS?
No,
because
it had
lutein.
AREDS 2. Remember, AREDS 2 also had beta carotene already. So in the AREDS itself, when we looked at separately, we think well the whole thing was pretty good in AREDS 1, but AREDS 2 when we separated the beta carotene versus the
Lutein,
lutein,
yeah.
the
So
lutein was better. Lutein was better.
So you would think that AREDS2 with lutein would be better than the original formulation.
Right.
This,
But what's
so that's some
interesting-
evidence that it really is the lutein that
We think
is
it's really
important.
But what's interesting with AREDS1 is that we saw it through, we did looking at the fovea, but also looked at just the area growth in general, you know, how fast the area in, as a whole. The beta carotene was actually important in reducing that as a whole, in addition to that. We didn't
The
see
beta carotene or the whole AREDS formulation?
That's probably the whole AREDS formulation. So we
Because
can't,
it's hard to believe
can't
beta
separate out.
carotene.
We can't separate out. So the seed is probably really important there somehow as well too. You know. But we look at nutrition, lutein synthetase comes up. If you look at nutrient and GA growth, lutein synthetase also comes up as important as well too.
Right.
So I think lutein really is an important function. It makes biological sense because
Oh,
it's
yeah.
immaculate.
Well, that's why I was asking about AREDS. I would have thought maybe the AREDS wouldn't be as effective as
But obviously
AREDS2.
antioxidants. So,
Yeah.
so you know, when we looked at when I asked Non to look at,
But the antioxidants would seem like it would affect toward the fovea and away from the fovea, as opposed to lutein, where there's a difference in the foveal area compared to growing away from it.
so the lutein is in the macula, right? So it's naturally in there. So if you give it, should it be going towards the macula more or not? I don't know.
Well, I was saying that the more lutein you have, the lower the rate of growth toward.
Towards the macula.
Yeah.
Got it. That
And
makes sense.
so if
yeah.
you feed them, I thought there was evidence that lutein increased when you feed them. Of course, they were all those studies that we could never replicate with the.
Oh, you mean the macropigment stuff?
Yeah.
Yeah, I wonder why Paul Brinson never published this stuff, because he did the macropigment, he never published, I don't think he found any difference. I mean,
Yeah,
I never got an answer from him, and you know, it was so unpredictable, especially with the stuff that he was using. If I just shift my head, I got a different number, you know. So, but what's interesting also is I convinced Anand Swarup to look at some mice models, looking at daily supplement and Mediterranean diet. What was interesting was in the retina, it increased the HA was much higher. It's got to be sort of lipids. So it's the oxidative, it's antioxidant stuff, and it's helping mitochondria. It has to do with energy, energetics of the retina. That's really important. And somehow we're providing more energy to the retina, and then decreasing mitochondrial dysfunction is also really important.
I mean, that's the thing that the take-home message I get every year after going to the Ryan meeting is how multifaceted this whole
It's
thing
just not
is.
one
And
thing,
you've got all
exactly.
these different factors and
Yeah.
sorting out, what's important for Mrs. Jones is
Make a difference for
pretty
somebody
tough.
else.
And the mechanism, of course, you know my feeling about mechanisms. You've heard me say multiple times.
Well-
We make up mechanisms. Sometimes they're right.
But that's exactly what, you know, if everything's mechanistic in IH. I mean, you heard Paul. Like, you don't know how mechanism works. Now, why shouldn't we give this stuff? And I know Mary Ann said to him, if you're having a heart attack, ER, do you care how that works? Do you want to take the drug or not? Paul Seaving.
I like my treatment A versus treatment B, and what's the mechanism is. I said,
maybe not the way, right?
B is better than A, and the inversion time is the amount of time it comes up with an equally plausible explanation.
You may have some change in that with the new director in IH. Maybe she'll emphasize. I mean, epidemiology is dead now.
Oh, yeah.
It's totally dead.
So I went to the business meeting of the clinical research group.
I missed that one. I forgot. I wonder where I was.
So they want to change the name.
Population science.
Yeah,
right.
and get epidemiology out of it because they say epidemiology is dead. Well, I don't, traditional epidemiology may be dead, but epidemiologic principles aren't dead.
Well, clinical research. I mean,
population.
clinical trials is an epidemiology,
Yeah, sure.
right? So.
And so I view epidemiology as the application of statistics to medicine. And but I think they're right that traditional epidemiology
Well,
is being replaced by.
I'm on the Central Steering Committee for Epidemiology, NIH. Nobody comes in except for cancer. Nobody has money for it. Nobody's done anything. Right. Cancer, NHLBI occasionally, and NIEHS, but nothing from even NIA has nothing in there.
Well, cancer has
2.9
risk factor analyses that they can
2.6
do
Right.
related to the genetics of the tumor and stuff that are.
Well, they have also a lot
we
of
don't
big,
have.
they still have a big population. Like they have they have like a sister study for breast cancer. So they have a whole bunch of things like that. So, thank you. So it's interesting that they still have like some of these. They do bring patients in, and just some way we know what I really think needs to be done now, Rick. I think what needs to be done is that all the institutes get together, NHLBI, NIA, NEI, look at all systemic diseases. Can we look at the eye and see how it relates to all these other diseases? That's not been done very well. People talk about that now, but that's not been done. We need large populations.
So what's in this million man thing related to the eye?
All of us.
Yeah.
We don't even have eyes in it yet. All of us, we're just trying to get fundus photographs in there now. 1 million people are getting
all this information about them, but
yeah.
that's what I was
like
asking.
EHRs, getting
What are they
EHR
doing
and doing
about
some
eyes?
and genetics. There's nothing. So we've been trying to get some fundus photographs, and I guess they can connect it to EHRs. So Sally Baxter has written some papers on, the drug relationship to glaucoma, for example, and stuff like that. So it's not really, it's okay, but it's not really good. So you need some real eye exams. You need some HRs. Imaging is really cool. If without imaging, you don't know the diagnosis. It's just like you see diabetic, who cares? What does that mean? And you can wear Fitbit. So I'm actually registered. So I got a Fitbit. So I'm wearing the Fitbit. Plus my watch.
What does a Fitbit give you that your watch doesn't?
Well, I wear this all. This is good for 8 days. You don't have. So it actually gets my sleep and everything else. What happens?
It just gets my sleep. I just put it on when I'm showering, and by the time I finish showering, it's all charged up.
I don't do it for 24 hours. Maybe I could do it for 24 hours.
I do.
Yeah, maybe I could do it then.
So this tells me about my sleep.
Yeah.
Because I'm interested in my sleep.
Yeah. We should have lunch.
I increased my sleep time today.
How did you do that? Sleeping longer?
Came late.
That was good.
So Barb and I decided we'd come late. So we took a cab.
Oh, good.
And the cab drops us off and says, I can't go right next to this place. So he sets us off walking. We're walking around, we can't find it.
Really?
And, he said, well, it's right up that alley. Well, it wasn't up that alley. It was a totally another place. And eventually we...
Little vans that brought us right up.
Yeah.
Funziona? Ok. No. Sì, nel senso che siccome ho ereditato anche delle slide di un collega, ho messo tutti insieme e non ho convertito PowerPoint, ho attaccato il computer, però mi hanno detto prova a vedere se funziona il mouse. Non funziona? Quindi per avanzare ma non... no, il mouse accendo e spengo, ok, un attimo. Sto cliccando ma non va. È spenta. Veramente. Sì, si accende all'inizio ma poi resta spenta. E poi non basterebbe mettere questo e cliccare sull'altro? No, esatto. Non è uguale.
Yes, I know you.
I just wanted to say, it's so nice to see you.
Yeah, we need some beautiful women there.
You raise but there's something, I don't think it's working. We'll,
Whenever you want, I start. Don't worry.
usual trouble.
Yeah, we can start.
Okay.
Okay.
Waiting for, waiting for.
Okay, so
Okay, if you want to please take your seats so we can start relatively on time with 10 minutes of delay only. Okay, so we will start now this session, this afternoon session. And I'm very happy to chair this session together with my friend Francesco Viola. And I introduce the first one so you can introduce the second one.
That is most important one.
The second one. And the first speaker is Professor Rossi from Naples, and he will present biosimilars for the treatment of neovascular AMD and diabetic macular edema. Please.
Thank you, Professor Mandello. I talk about biosimilar for the treatment of neovascular age-related macular degeneration and diabetic macular edema. The anti-VEGF drugs have had a substantial impact on clinical outcomes of patients with neovascular AMD and DME. Despite the clinical benefits, the high cost can lead to challenges in patient access. To mitigate the financial burden, biosimilars were developed. About the definition, biosimilar is a biologic that has been demonstrated to be highly similar to original drug. Development is usually less difficult and costly. BioBetter, named also second generation biologic, is a biosimilar with more efficacy and safety than original drug. Generic is a copy of an already marketed chemical synthesized. About the approval, we have 4 steps: chemistry, manufacturing and control, quality data, preclinical data, and clinical data. About chemistry, molecular and protein characteristics of a biosimilar must be similar to original drug. Quality data, analytical studies are performed using state-of-the-art technologies to characterize the structural attributes of the biosimilar. About preclinical data, the EMA and FDA encourage sponsors to conduct comparative tests using human cells. Animal studies are not fundamental, and animal safety and toxicity are not essential. About clinical data, the phase 2 studies are not usually conducted, but are fundamental clinical trial, phase 3 clinical trial comparative study. An important concept is the extrapolation. When a biosimilar is approved for one indication, automatically is approved also for the other indications of the original drug. We have ranibizumab biosimilars. The first approved is SB11, but we have also others ranibizumab biosimilars approved as FYB201, XSB001, QL1205. The approval of the SB11 was based on a phase 3 clinical trial in 700 patients with neovascular AMD treated with intravitreal injection of a biosimilar or ranibizumab every 4 weeks for 48 weeks. The primary endpoints were change in best corrected visual acuity at week 8, and change central subfield thickness at week 4. The results is evident that there was a significant difference between both group for CST and for best corrected visual acuity. Adverse events are similar for both groups. SB11 was approved in Europe for all indications, but in the United States was not approved for diabetic macular edema because during the trial used another concentration of the drug. We have others ranibizumab biosimilar as FYB201 is approved in Europe and in United States. The approval was based on a phase 3 clinical trial in 77 patients treated with intravitreal injection or with ranibizumab or with biosimilar. The primary endpoint was change from baseline at week 8 in best corrected visual acuity. About the results at week 8 and at week 48, there aren't significant clinical difference between both groups. About adverse events, the adverse events are similar in both groups. Also for aflibercept, we have biosimilars. MYL1701P was approved in Europe and in the United States. The approval was based on phase 3 clinical trial in 355 patients with diabetic macular edema. Patients received aflibercept or biosimilar every 4 weeks for the first 5 doses at week 0, 4, 8, 12, and 16, and after every 8 weeks. The primary endpoint was the same, was the change from baseline in best corrected visual acuity at week 8. About the results, there aren't significant differences between both groups for best corrected visual acuity at week 8 and also at last time point at week 52. Adverse events are similar in both groups. We have also for bevacizumab, we have a biosimilar. In fact, Outlook Therapeutics is currently investigating an ophthalmic labeled bevacizumab biosimilar in 3 clinical trials for neovascular AMD and in 1 clinical trial for diabetic macular edema. We remember that and we know that bevacizumab is an off-the-label drug for neovascular AMD and also for the ME. For this is not valid the concept of the extrapolation. In conclusion, the biosimilar, thanks to the reduction in costs, could be very important in the future for the treatment of neovascular age-related macular degeneration and diabetic macular edema. The only limits could be represented by the skepticism of the part of the ophthalmologists and the patients, which, however, can be overcome with time and clinical practice. Thank you.
So I want to present my co-moderators. So Professor Bandello will talk about brolucizumab. Where does it fit in the new treatment landscape?
Thank you, Francesco. Thank you, Francesca. Thank you, Giovanni and Bas, for the invitation. I'd like to start saying thank you also to my Alessandro Rigo, who is one of the youngest in our department. He worked for this presentation. These are my financial disclosures, and I must evidence that I serve as a consultant for Novartis. So this is not a contradiction, but my mentor taught me that when you speak, the attendees will follow you for a few minutes. So please start saying the most important thing. And this is the most important thing. This is my last slide, where I tried to synthesize the main concepts. So the importance to have some reabsorption of fluid is one of the most important aspects of our therapies using anti-VEGF. And brolucizumab shows us that there is a very important effect to make the retina dry. For sure, we have side effects, and this made really the attitude of our colleagues in other continents with brolucizumab completely different from our attitude of European attitude. And although there is this very important efficacy, we must consider the safety problems related to the use of this drug, and this is the main reason for different attitude. So my last point is that it seems that for brolucizumab, the world does not seem a global one. I mean, we have differences that are not justified, and we must try to understand why these differences are so important in different continents. So the first point is that we know that the reabsorption of fluid is very important to determine the effect of our therapies. And it was shown already that if you have a very fast reabsorption of fluid, you have an improvement in visual acuity, and this will stay for a very long time. Patients with no reabsorption of fluid, with persistence of fluid, are usually the worst patient. Brolucizumab is very effective with this respect, in this respect, and the reason maybe is this equivalent molar dose, which is the highest compared with the other anti-VEGF drugs that we have today. So what we know is that we have a superior fluid resolution with longer treatment intervals. So we can have a very effective dry effect on the retina associated with a longer intervals among different procedures. And another important thing is that it was shown that with longer follow-up, this kind of therapeutic effect is maintained. And here you have the results after the longer follow-up. But as I was saying before, one of the most important aspects to be considered is the safety profile. And this was the main reason for different reactions in different countries. And as you can see here, in the two most important registration trial done for this drug, there were significant differences related to the intraocular inflammation, retinal artery occlusion, and endophthalmitis. And what we know from different clinical trials now is that the effect of brolucizumab is also very important in diseases where till now it was different to find any drug really effective. One of these is the polypoidal lesions, and the other one is the most important retinal pigment epithelium detachment. And in these kind of diseases, which usually are very difficult to be treated, the brolucizumab showed a very important therapeutic effect. What about diabetic macular edema? We know that in diabetic patients, you have a great percentage of eyes. They do not show a response to the anti-VEGF approach. And this percentage of patients is maybe to be considered, and we should try to understand why these patients are not sensitive to our therapeutic approaches. And as you can see here, the probability to have persistent diabetic macular edema is really very high in diabetic patients, and after 3 years, it is still 40% of our patients. So also in this case, the use of brolucizumab showed very important therapeutic effect, and also in this case, anyway, we had some problem related to the safety profile, although in diabetic patients, and we do not know why, the number of patients with some important side effect is lower compared with what happened in AMD patients. This is the long-term therapeutic effect with brolucizumab in diabetic macular edema. And what we can say is that the effect is very important in diabetic patients and is also maintained after longer follow-up, as you can see in this experience. And in this case, the safety profile showed that there was some difference in the two groups compared with the aflibercept, but anyway, the importance of the side effects in diabetic patients, for some reason that we do not understand why, the effect is less than what happens in AMD patients. So this is a slide where we are trying to show what is happening today. And as you can see, the number of patients treated in different parts of the world is completely different. Japan, Korea, and some other countries shows very important use of brolucizumab. On the left, you have the different countries, but if you consider the number of brolucizumab vials used compared with the inhabitants, you see that Japan is the first country, South Korea is the second, and Italy is the third. So it's really difficult to understand this huge difference among different countries. And I must say that we have some experience in our department. This is used, this is a drug which is not used as the first-line treatment for sure, but in our experience, in our hands, you can have very important therapeutic effect without significant side effects. So the problem is these are the side effects on the basis of the numbers, and this is in the two Oak and Darrier trials, and the overall incidence of side effects was 4.6. And an important side effect such as intraocular inflammation and retinal vasculitis, 3.3, and intraocular inflammation plus retinal vasculitis plus retinal vascular occlusion was 2.1. And what is important to remember, in the great majority of cases, you have the side effects usually after a short-term follow-up. In the first 6 months, you have the complication. And what is important to consider is also what are the risk factors for these kind of events, what you should consider in order to avoid that your patient shows some important side effects. We saw some important side effects in our department, thanks God, not in our patients, but in patients that were referred. And when you see some important side effects, you are really in trouble and you start to feel very bad. And so you are hoping to have your patients never affected by similar complications. What we learned is that if you start avoiding brolucizumab in any kind of inflammation, in any kind of patients having had some kind of inflammation, you can reduce significantly the number of complications. The other point is that the patients with complications usually are female, with some systemic inflammatory disease or some local inflammatory disease. These patients must be absolutely not treated. And finally, why is there a justification for this? Why these patients show complications? So what we must admit is that the presence of pretreatment anti-drug antibody is present in patients treated with brolucizumab in a higher percentage compared with the other drugs. On the basis of this, it was started some kind of guidelines or recommendation for using this drug in our patients. So this is my first slide, and I remember you at the end of this presentation. It's important to consider that brolucizumab is able to produce a very rapid therapeutic effect, and it is associated with very important dry effect on the retina associated with good visual function results. And the complications that for sure can be present can be avoided if you are able to avoid patients with some kind of inflammatory history in their life. So final question is this really a global world, or there is some difference in different continents? And this is a provocative question for our US colleagues that are here today. Thank you very much for your attention. Oh, sorry. And now it's the turn of my co-chair, Francesco Viola. He will speak about faricimab and benefits and dual pathway inhibition.
Thank you very much, Professor Bandello. Thank you very much, Professoressa Simonelli, Vas and Giovanni, obviously. Really nice place, incredible. So faricimab benefits of dual pathway inhibition. My disclosure, I'm a consultant with Roche. So angiopoietin signaling pathway regulates vascular homeostasis and controls vessel permeability, inflammation, and angiogenic response. You see here this cartoon with a healthy tissue with the angio-1 signaling maintains mature and stable vessels. And in the other side, the pathologic tissue where angio-2 signaling leads to vascular instability, to the pro-angiogenic action of VEGF. What about faricimab? Faricimab is a bispecific antibody that independently binds and neutralizes both anti-VEGF and angio-2. So, and what about faricimab in AMD trials? So we have the experience and the data of Tenaya and Lucerne. Tenaya and Lucerne, we have data of the first year, of the second year. So the primary endpoint was the first year, but it was very important to analyze also the second year. So if you see, the comparator was aflibercept with the loading, with free injection, and every 2 months for the rest of the study, so the first year and also the second year. And the faricimab 6 milligrams was studied with a loading of 4 injections monthly, and that's, in my opinion, very important to have very good result. And then there was a disease activity at 20 and 24 weeks. And as you see here, you can have a group treated with the Q8, a group treated with Q12, and a group treated in the first year with the Q16. And this is based on disease activity. And disease activity is based on OCT or visual function or new macular hemorrhage. And the second year was interesting because we did the treat and extend regimen. So we know the result. We know that at the end of the first year and also the second year, faricimab was very similar to aflibercept in terms of best corrected visual acuity. And also if we look at the data on OCT, so on the fluid. What is important, obviously, is the disease control with less injection, and we see here the difference. So we did this with, we obtained this with 10 injections of faricimab and 15 injections of aflibercept, but obviously it could depend also by the design of the trial. But the good thing is the second year, you see that faricimab was used for 3 injections, aflibercept for 6. So we know also this data that is the most important one from the trials. So we know that 80% of the patient almost achieved a cool 12, so a treatment or more. So every 3 months or more, the first year and also the second year. So it's very important, this data to control this disease and to treat less our patient. And you see the number of injections and the difference again. This is the slide I like more now, but please, this is a sub-analysis, but it's very important because it's the only period if we compare the two drugs, so aflibercept and faricimab, after the first 3 injection in the loading, because the loading was different after the first 3 injection. And if we compare the 2 drugs, we clearly see that at the in the week 12, so after the first 3 injection, we have less fluid with the faricimab compared to aflibercept. And the other good point is that we had less patient with fluid compared to patient treated with aflibercept at 12 weeks. So again, we have not the power of a trial, but we have a very good data to show that is maybe drier as molecule. The other data is that is faster than aflibercept to achieve no fluid or less fluid. If we look here, we see that faricimab patient achieve first absence of intraretinal or subretinal fluid faster and with fewer injection versus aflibercept. We see that the 75th percentile was reached with 2 injections and at week 8, compared to aflibercept with 3 injections at week 12. So this, in my opinion, it could be a good data to take in mind when you treat patients. The other post-hoc analysis, again, post-hoc analysis, but good data is that patients were with detachment of epithelial pigment detachment involving the fovea were similar in the both groups. So we had data of the treatment, and you see that they look to predominantly serous detachment to fibrovascular or to fibrovascular only, so based on the OCT, obviously. And if we look the result, we see again after the first 3 injections, so the only time of the study where we can compare it, the 2 drugs, that we have less fluid in patient with the presence of PED at the baseline in the faricimab group compared to the aflibercept group, if we put all the patients together. Not if we look only the fibrovascular PED at baseline, but if we look at the serous PED, again, the difference is more important at week 12. So it seems that it's drier more the retina than Eylea, that is the drug we were used to have it in our clinic. And again, these are the number of patients with fluid after the 3 injection. You see that patients with serous PED with faricimab are more or less 4% compared to 12% of aflibercept. This is the treatment regimen with faricimab. You can treat as suggested. So the loading, you can check the active disease at 20 or 25 or 24 week. The application in my clinic is a little bit different. We see that we do the loading, and then depending on the lesion, the activity of the lesion, we decided to extend with the Q8 or directly to Q12 in, for example, type 1 or type 3 lesion. And this is the classical example of a good result after the first 4 injections, how we treated. About the safety, we can say that from the trial we have very good data about the safety. And also on the real life, at the recent ARVO, the group of Murphy published this, the experience at Murphy's with more than 2,000 patients with very few inflammation. So we can say we have a very safe drug. What about the faricimab in DME? DME, the study, the Yosemite and Ryan was different. You see that we had the 2 groups of faricimab, one with the loading of 5 injections and then every 2 months treatment, and the other group with faricimab 6 milligram with the 4 injections monthly and then with the treat and extend, compared to the aflibercept, as you can see here. And again, the results are similar if we see the visual function, but we see that the number of injections are fewer with faricimab compared to aflibercept in the first and in the second year. And the OCT, so the fluid in the retina, is very, very similar. So again, we have these cartoons showing why faricimab could be important for our patient, and also for our life or for our clinics, in our busy clinic, to do less injection if we treat at the beginning. Again, this analysis show in the sub-analysis, in the post-hoc, also in DME, that faricimab drive more than aflibercept. We see that the 50 percentile is achieved with 7 injections compared to 9 injections. If we look at the fluid in the retina, and also if we look at the non-fluid, so less than 280. So in conclusion, also the safety profile in DME is, I would say, very good. So we have no concern about that. So in conclusion, I think that faricimab targets 2 distinctive disease pathways to promote vascular stability. It has a durable vision gains, can be used actually in DME and neovascular AMD, and the safety profile is very good. Thank you very much.
Thank you, Francesco. You had something around 15 seconds of delay, something around there. Yeah. So we have now a very small change in the program, please.
Yeah, unfortunately, Alessandro Ivernizzi is not here, so we will go through my friend, Professor Minnella.
Our friend.
Our friend, okay, so we know that. Our friend. Steroid therapy for neovascular AMD and DME. Please, Angelo.
Thank you very much. Thank you to the organization, the president, for the invitation. I'm going to, these are my financial disclosure. I will, first question, there is a role of inflammation, and so the steroids have a role in retinal disease. So we know that as well as vascular leakage and edema, and as well as the neovascular presence, we can have also inflammation. So just to the first question, we already an answer. And we know that the inflammation has a role in retinal disease in different way, depends from the disease. Mostly in diabetic retinopathy and retinal vein occlusion, less in wet age-related macular degeneration. This is the reason why usually we use steroids for retinal vein occlusion and for macular edema and uveitis, and much less for wet AMD. So we know that we have a lot of cytokines that we found in humor aqueous, and we know that pro-inflammatory cytokines in ocular media are reduced by corticosteroids. So this is the action of the dexamethasone and in general of the steroids. So we can say that in presence of DME, we have a double possibility, double chance to use steroids to as a counteract the inflammatory action, and also the VEGF and Then we also the anti-VEGF that are specifically indicated for the angiogenesis and for the VEGF. So, as Professor Bandello told us before, with this slide I can conclude my presentation, because these are the guidelines regarding the use of steroid in DME. And we see in the slide that we can have on the far right some case where there are patients that are really not compliant. I have some patient that usually comes not very often, just sometimes, and this just could be an approach to use as first-line steroids in this case. And then we have, the history, so some kind of motivation that are outside the disease. We can have, a situation where we have story of vascular attack, cardiovascular problem, and this is another reason to use in first line this molecule, this drug. And then we have a case where we have, persons that are they have a vitrectomy or a pseudophakic. And then there are some reason just due to the disease. Which are these cases? Are the case where we can find in the retina some retinal structural parameters like the drill, the disorganization of retinal inner layers, or the hyperreflective foci. So, which are the devices that usually use as steroids in our clinical practice? Mostly the slow release dexamethasone and the fluocinolone acetonide. We are using the port delivery system under a clinical study, but not for DME, but especially for neovascular AMD. What could I say a little more regarding the use of steroids that usually we have already used in our daily practice? I can say that there are some cases after the loading dose where we have a persistent macular edema despite the use of drugs. So in real world, this study that I'm going to present just to have an idea how is the situation regarding the possibility to continue with anti-VEGF therapy versus switch to dexamethasone implant. In fact, the question, shall we stay or shall we switch in this study, just point out the situation and analyze this fact. And in the study, as you see, we can, if we continue with anti-VEGF therapy, we can have, in comparison, weaker result instead of using the steroids. And these are the results at 12 months. But not only, we have also from the same study the result at 2 years that are mostly the same. So in this case, we can really think of use first or before waiting a long time for switching from anti-VEGF to steroids, so we can use this opportunity to have some result. This is another study from Professor Bonfiglio that just correlate what I showed you before, the fact to find inside the retina some kind of qualitative distinctive sign of inflammation that could allow us to use this drug. Safety, as well as the MID study, that is the most important study conducted for the steroids. We have the Saffodex study that just analyzed, and they saw that overall ocular hypertension incidence in the study eye was around 30%. The incidence did not change, increasing using multiple kind of injection, and was statistically higher in the patient with RVO, with the retinal vein occlusion, as we can have in our normal experience. People with RVO usually have at the baseline also a higher intraocular pressure. And so we can conclude that, in 90, almost the totality of the patients are treatable, the IOP with the drug. And so this is the conclusion of the study. As far as concern the other drug, the long-acting fluocinolone acetonide that is used as intravitreal injection, we know that usually the time of it lasts is around 3 years. These are the kind of technology where we have a continuous microdosing delivery, where after a rapid concentration peak, we have a stabilization during the 2-3 years. This is the slide that point out difference in comparison of fluocinolone versus anti-VEGF and versus dexamethasone, where sustained morphological and functional effects are obtained using this molecule, where we have a stabilization in the central macular thickness during a long time. This is our small experience. It's a retrospective study where we just found out the situation in some patient exactly. This is the scatter plot. Mostly the data are, as we know from the literature, so nothing special, nothing more. What is really we found a little different. We did a study regarding the electrophysiological aspect, and we found that the PNHR and the B wave amplitude significantly increased during the first 3 months, and the trend towards an improvement was observed also later. So in summary, we can say that fluocinolone is another steroid that could reduce the burden from intravitreal injection. It has a long-term control of DME with the stabilization of macular thickness. We can say that we can have a better result also in patients with not long term DME, but maybe in short term. In our experience, we confirm this data from the literature with the significant CMT decrease and the visual acuity stabilization. And we hope to better study in the future is that we find some positive effect of ILUVIEN or fluocinolone in the retinal function, different from other steroids. And last slide, just to make a point for later to have the discussion, the importance of using the dexamethasone in patients undergoing cataract surgery. I know that in the States, usually the surgeon, the posterior segment are completely different from people that usually do the anterior segment. In Italy, we do both, and we found some cases like this with patients with diabetes after the cataract could have this kind of situation. Thank you very much.
Grazie. Thank you very much, Angelo. Now we move to the last presentation, or we don't know, maybe that we will have the presentation by Professor Invernizzi. But at the moment, the next one will be Professor Testa. He will present maybe one of the most interesting topic of today, the gene therapy for neovascular AMD.
Thank you. Thank you, the inviters, for this kind invitation. I'll briefly review the main gene therapy approaches for patients with the neovascular age-related macular degeneration. As you know, the wet form accounts for about 10-20% of AMD cases, and it's particularly important because it is responsible for the vast majority of cases with severe visual loss. The existing therapies for neovascular AMD aim to inhibiting VEGF that plays a crucial role in neovascular AMD because it stimulates neovascularization. Randomized clinical trial has shown that intraocular injection of anti-VEGF block exudation, allow fluid reabsorption, and improve vision. Thus, the standard care for neovascular AMD consists of a repeated injection of anti-VEGF. However, Studies of long-term treatment in clinical practice show inferior visual outcome compared to those reported in clinical trial, probably due an undertreatment on non-compliance with the scheduled injection. Therefore, there is a strong need to develop treatments that provide sustained suppression of VEGF. To address the limitation of the existing anti-VEGF therapy, novel gene therapy-based strategy have been proposed. This approach is called biofactory and relies on a vector-mediated introduction of a genetic sequence to achieve the endogenous production of a therapeutic protein. For the specific case of a neovascular MD, this protein targets well-established pathways such as the VEGF. As you can see, there are different clinical trials that are currently testing the biofactory approach with different drugs for treatment of wet AMD. Today, I will focus on two most promising therapies based on the recently published results from phase 1/2 trial. This drug, ADVM-22 and RGX-314, are now under evaluation in further clinical trial to confirm their efficacy. ADVM-22 utilizes a novel vector capsid that carries a cassette with the coding sequence of aflibercept, a known inhibitor of VEGF. The adopted serotype facilitates the transit across the inner limiting membrane, and therefore it can be delivered via single intravitreal injection. A multicenter open-label phase 1 study has been designed to assess the safety and efficacy of a single intravitreal injection of ADVM-022 in patients with neovascular AMD, previously controlled with frequent anti-VEGF injection. 30 patients were dosed with a single intravitreal injection of the drug at 2 doses with 2 different prophylactic steroid regimens. In particular, 12 patients for cohort 1/2 followed the 30-day prophylactic oral prednisone regimen, whereas 18 participants for cohort 3 and 4 followed a 6-week prophylactic topical dipropionate regimen. Cohort 1 and 4 received the high dose of the drug, while the cohort 2 and 3 received the low dose. All participants were followed for 2 years. Any subject that showed a worsening in terms of visual acuity loss due to fluid accumulation or hemorrhage received a supplemental aflibercept injection. As you can see, the majority of the adverse events were dose-dependent and mild or moderate. And only 5 serious adverse events were reported. 2 cases of cataract, a case of dry AMD, a case of retinal detachment, and a case of current uveitis. The secondary point of the study were evaluation of aflibercept expression level in aqueous humor, evaluation of vision maintenance, evaluation of central subretinal thickness, and the reduction in supplemental aflibercept action. The trial showed that the concentration of aflibercept in the aqueous humor remained stable through the end of the study. Moreover, ADVM-22 treatment results in stability of BVCA in both dose groups up to 2 years. Moreover, CST decreased after aflibercept administration at screening with a mean change of about minus 90 micrometer in the low dose group and about minus 60 micrometer in the high dose group, and it remains stable over 2 years in both groups. After ADVM-22 administration, the frequency of anti-VEGF injection was decreased in both groups. Specifically, the mean annual VEGF injection were reduced in 98% in the high dose group and 81% in the low dose group. After 2 years,
the 80% of high dose group and 53% in the low dose group had not performed any supplementary injection. In summary, the optical trial demonstrated that a single injection of RDM22 induced a sustained level of intraocular aflibercept, improved retinal anatomy, preserved best corrected visual acuity, and limited the number of anti-VEGF injections. The superior safety profile at low dose compared to high dose, together with a comparable efficacy between the two doses, support the design of phase 2 study adopting the low dose of the OPTIC trial. The second gene therapy approach, RGX-314, uses an adenovirus vector to deliver in the retina a transgene encoding a ranibizumab-like anti-VEGF monoclonal antibody fragment to achieve continuous suppression of VEGF. The therapy is being tested in both subretinal and suprachoroidal delivery in wet AMD. The subretinal delivery of RGX-314 gene was evaluated in a local-label multicenter dose escalation study in 42 patients previously treated for neovascular AMD. The study tests 5 viral doses administered by subretinal injection. First, the patient received an intravitreal injection of ranibizumab at visit 1 to determine the anti-VEGF responsiveness. Only patients who showed a response in terms of a CRT reduction or central fluid reduction were eligible for subretinal injection of RGX-314, 14 years later. The design included 18 participants enrolled in the 3 ascending dose cohort and 24 participants in 2 additional dose cohorts at higher level. Overall, no clinical significant immune response, drug-related inflammation, or post-surgical inflammation were observed. Thus, the adverse events observed in participants were mainly of grade 1 or 2 in severity and rarely serious. Similarly to the OPTIC trial, the secondary points included the measurement of RGX protein levels in aqueous fluid, visual acuity, central retinal thickness, and the number of anti-VEGF injections. As you can see, the concentration of RGX-314 protein was detected in aqueous humor in all cohorts already 1 month after injection, while 2 years after injection, the main protein concentration were substantially higher in cohort 3 than to 5 than in cohort 1 and 2. In terms of visual acuity, the mean baseline BVCA was either maintained within 5 years later or improved in cohort 2 to 5 up to 2 years post-treatment, whereas cohort 1 showed a gradual reduction in BVCA over time despite the frequent supplementary injection. The CRT did not change significantly in cohort 1 and 2 and 3 over the follow-up, but in cohort 4 and 5, there was a substantial reduction in CRT between baseline and 1 month, which remained stable up to month 24. Finally, in cohort 1 and 2, required about 10 supplementary ranibizumab injection per year to control exudation. So there was no significant change compared to the year before treatment. Instead, there were a 60% reduction in cohort 3 and 4 and about 80% reduction of supplementary injection in cohort 5. In summary, the subretinal injection of RGX-314 in patients with neovascular AMD achieved a sustained expression of the protein at least 2 years after treatment and control exudation. These findings suggest that this therapy is a promising strategy to provide durable, sustained suppression of VEGF, and pivotal trials to test this hypothesis are underway. In parallel, One-time suprachoroidal delivery method is under evaluation in phase 2 trial. In conclusion, a new generation of gene therapy is expected to reshape the treatment of neovascular AMD. Instead of replacing a faulty protein as we generally apply for inherited retinal dystrophy, this therapy prompts the eye to make a novel therapeutic protein. Pivotal trials are ongoing, and it is estimated that if the trial gives the result comparable with the corresponding 1-2 trial, the one-and-done therapy for wet AMD may be available in 3-4 years. In any case, these new generation of gene therapies are a treatment for the consequence and not for the causes of wet AMD. And so there are not an ultimate therapy for wet AMD. Thank you for your attention.
Thank you, Francesco.
Thank you very much, Professor. And now we conclude with Professor Stawenghi with the 8 milligram aflibercept benefits of quadrupling the dose. Thank you, Giovanni.
Thank you. Yes, I actually substitute Alessandro Verlizzi that has no voice, so I have to substitute him. So again, apologize if I will not be as good as it would be if he will present that presentation, this presentation. So the 8 milligram aflibercept is it's already present in the United States, in Italy, and in Europe. It's coming now, and the new drug is actually an advantage, we think, and I will try to show with the data of the clinical trial. These are my disclosures. So we know very well the aflibercept, and we know that it's working quite well, but the problem is always the duration of the treatment. We have to inject at least every 2 months, or if we do a treat and extend, we can reach a 3-month interval. But one of the disadvantages is that at least in our social medicine in Europe, we have to have overloaded clinics, and so that it's important to have the possibility to prolong the treatment. So in fact, if you look at the effect of the current intravitreal agents, the possibility to move in this way, the treatment interval will be very, very useful. Now, again, we know that the VEGF aflibercept is inhibiting VEGF-A, B, but also the placental growth factor and the galectin-1. And it's important to have a high dosage. The high dosage is actually working in increasing the molar dose. And that, of course, will help to maintain the treatment for a longer time. You see, just with the cartoon showing, the time is and the quantity of dose that molar dose that can stay during the time. So if we look at the aflibercept at 8 milligram with a higher, molar dose, we can actually expect to have the effect of the inhibition of the VEGF, A and B, placental growth factor, and galectin for more time. The 2 study was conducted, one for AMD and the other one for DME. So the pulsar for wet AMD and the photon for DME. I will just show the data from the pulsar. There was 3 arms, a 2 milligrams with after the 3 injection at Q8, aflibercept 8 milligram after the 3 initial injection every 3 months, and or after or every 4 months. And the endpoint was at the 48 weeks. That was useful for the registration of the drug, but the study continued until 96 weeks. And there was also a possibility to extension. So just to show again how it's administered in the 8 milligrams, the 12 and 16. sorry, 2 milligrams, 8, and with the interval of 8, 12, and 16 weeks. And you actually see that, of course, there is a possibility to change the interval based on some rescue, if we can say rescue treatment, if the visual acuity was losing more than 5 letters, and I underline and, because probably in the real clinical setup, we probably don't, use the and, but or, 25 millimeters of increase in the central retina thickness, or a new onset of neovascularization. And that is, of course, can create the possibility to extend. The second year was actually possible to, based on the same, if you don't have such a wasting, you can actually increase the visual acuity. So these are the results. The results, sorry, the interval, not visual acuity. These are the visual acuity results at 14 weeks, and based on that, the drug was registered for the use in clinical practice. And here is the results at 96 weeks. And you see that it's quite interesting to notice that the effect in best-corrected visual acuity improvement is overlapping with the 3 doses. So it means that you can actually prolong the treatment up to 16 weeks. And here was the photon. Sorry, I skipped because okay. What about again, the pulsar? Here you actually see the fluid, and you see that no matter you use the different type of drugs and the interval, it's quite interesting that more than 50%, up to 65%, you actually have a patient with dry macula at 16 and 48 weeks. And here, just as we used to see in in a normal table. Another thing that I would like to show you is that is not so evident the up and down that you usually see with a aflibercept. That could be just because there are interval of 4 weeks, more than 4 weeks, so you can actually not see the small changes that you can actually see with if you follow and you report every 4 months. But it's quite stable, and it's interesting to see that even if you go to 16 weeks, you can maintain such things. And again, another important thing is the number of injection. Of course, if you inject every 16 weeks, you have less injection. That is actually one of the secondary endpoint that we really appreciate in going back to the clinical setup. And this is always a nice thing to see, because these are the patients that one by one, and every line is one patient, and you actually see the ones that move to a different treatment intervals. And from the one that started with the 8 milligram Q12 or Q16, and you see that there are quite a big number of patients that maintain such an interval. That it means that it is not by chance, but it's probably related to the drug. What about the safety profile? Looks quite good as the 2 milligrams. There is no big changes, and you see that again, all the drugs can give a IOI, but you see that in that case it doesn't look like that by increasing the dosage you get more side effects, because here you see that there are more IOI with 2 milligrams than not with 8 milligrams. So I think it's by chance. And of course, we have always to be careful in looking at the patients that are treated, but again, is not a big problem. So to conclude, the possibility to have the good results with just comparable to what you get with the 2 milligrams, with the 8 milligrams, with a longer interval, and so a longer duration of the drug. I think it's demonstrated by the PULSAR study. Of course, then we will do in our clinical practice, and that could be all as usual, a little bit different. Thank you very much.
Thank you so much, Giovanni. We appreciate the effort you did to present this file of slides, starting to know them maybe some minutes ago. Thank you very much for that. I think that now it's time for the discussion and for the panel discussion. I'd like to invite here Professor Barbara Blodi, Francesco Boscia, Angelo Maria Minella, Dr. Lim, and Michele Della Corte. So I must admit that I did not prepare anything for this discussion, so we'll start very freely. And I'd like to ask to our colleagues coming from other continents, I start from my presentation, and do you think there is still room for using brolucizumab in other countries, trying to take into consideration some of the guidelines coming from people who were using this drug much more in different realities? I mean, is it possible to reduce the number of complications considering some important criteria for inclusion or exclusion of our patients from treatment? Professor Blodi, please.
It's a great question. Thank you for asking it. I think in the US, we use anti-VEGF, and we're so comfortable with the agents that we have. We're so comfortable with Avastin for many of us, for Eylea, now the newer drugs. It's very difficult to accept a 4% rate of blinding, but could be potentially blinding. And so there's inflammation with the other existing drugs. It's usually at 1 to 1.5%. So in my practice, I wouldn't consider brolucizumab.
Okay, you are really stable on your position. And yes. Francesco, tell me something about the use that you are doing.
Yes, we did use quite extensively brolucizumab when it was first introduced into the clinical practice. I remember when I was introduced to this molecule, it was named RTH258, was in advisory board at the academy ages ago, and it was quite obvious how the molar dose of this drug could be so effective, either in terms of anatomy and function. And so when it was introduced in Europe, we did use quite extensively. To be honest, we did observe few inflammation, no vasculitis. So all the events eventually ended up into a better visual acuity. So I'm not that dramatic, but to be honest, nowadays, if I had to inject my mother-in-law, has been say, well, I wouldn't. No, seriously, I wouldn't, because I think that we have been all used to use these agents, as has been said, comfortably, and we really want to sleep well at night without any problem. So really, I would consider just as the end resource.
So second or third line?
Third.
Third line. Any other consideration, Angelo?
I agree with Francesco. I would consider in the drug the efficacy, especially the early efficacy. This is important because we have seen from the study that if you start and you have immediately a response, that could be really helpful also for the long-standing, situation. So it will help a lot. And I will consider and will take into consideration the 4%. Regarding your question, we always ask the patient and we look for a previous information, but even in the case that where we had some kind of information in the past history was almost nobody. Nothing. So I will ask the patient. The only attention is just to instruct the patient that I am going to use a very strong drug, and probably get in touch with me in case of floaters or some other things.
Do you give them a telephone number,
Yes.
something like that? Okay. Please.
Yeah, the drug is actually not approved in Singapore, so we use it as a last resort basis. Thank you.
Thank you. You want to say something?
Especially in type 3 of CNV, is very effective. So,
Francesco, I have a question here.
Usha, sorry.
So, in Italy, do you ever see occlusive vasculitis?
Absolutely, yes.
You do?
Yeah.
Right. The reason is, we know from looking at drugs, other pharmacology, that there sometimes are drugs that are used very extensively. For example, metamizole is used extensively in Spain, and they don't get agranulocytosis at the same rate as we do in the UK, where you have a 24 to 1-fold increased risk. So clearly there are differences between populations. So the question in a way is, perhaps there may be genetic variability which increases the risk in certain populations compared to others. And has that been looked into at all?
Maybe that the mother-in-laws are different in different countries. Yeah. Phil, do you have any consideration, Phil, Bas, about this topic, which is an important one? Nobody can say, I don't care about that.
So after 3 or 4 episodes of inflammation after the 3rd injection, I stopped using brolucizumab, and we've been fortunate in having faricimab and now the high-dose Eylea available. So if I was taking care of a lot of polypoidal choroidal vasculopathy, and
Or
I
huge DP.
and I was frustrated with not being able to extend the interval beyond 6 weeks or so, I think I'd be tempted to use brolucizumab. I know the Japanese are very happy with it.
I know.
Please, I was going to say, and Tokan, I'm sure will comment too, but I think it's first of all, I think it's a great drug. It's really quite powerful, and I think also the indication may make a difference here. Maybe for DME, you cited very beautifully, Francesca, how the rate is lower, but on the other hand, we have a lot of different options for DME, and also, we don't have the same urgency. But, sometimes we fear so much this rare complication, but we know a much more common complication is what happens with uncontrolled exudation. So do you want your patient to lose vision because you didn't control their exudation adequately, or do you take this? So I don't think it should be an absolute. I do think that we've probably prematurely abandoned its use in the US primarily. I think a big factor in that was it was surprise. By the time the drug became available outside the US, people were aware of this, right? So, and people don't like getting surprised. That was the big problem, I think, that we faced. And I think that we've probably made a mistake in our lack of utilization, because I think there are scenarios where it is probably appropriate. And maybe Tokan might comment, but I think if I had a higher prevalence of PCV, I probably would be using it more. But like Phil, I don't really use it now.
Giovanni, do you want something?
Yeah, well, I absolutely agree with Vas. I think it's probably the most potent drug. It's brolucizumab, and I think there are a series of things that I think it's important in the selection of the patients. First of all, you have to exclude any patient that has any type of general inflammation that they know. Second is that I would not choose a patient that is recovering in a
Far away.
patient
far away
hospital.
from everything.
Yes, exactly. But they have to be able to understand the small changes, floaters and so on. Up to now, we actually started, we injected more than 1,000 times, and we actually have only 5 subjects and no one a big problem, and also the ones that have some vitritis was solved in few times. So
It's very important to talk to patients, to inform them, and to have them coming immediately.
That is a key issue.
Absolutely. Glenn.
Yeah, you were asking about things to potentially prevent it as well. And one of the things, I was on the safety review committee for brolucizumab and saw those cases, and they were getting regularly evaluated by slit lamp exam, et cetera, as part of the study. But some of those patients had inflammation that there was sort of a stereotypical pattern of presentation where they would get some anterior chamber cells, and then they might get some vitritis, and then they started getting vasculitis. And in several of these instances, they had anterior chamber cells, they received an injection, they had persistent inflammation, received another injection, and in general, we're not as good at looking every time we see the patient to check for signs of inflammation. And I think that's something that certainly we can do. And
Sometimes I think it's our responsibilities. We saw a woman few weeks ago. She was treated with brolucizumab. She had an inflammation, and she came back in the emergency department saying that she had this problem. The doctor did not realize that she was doing brolucizumab, and the patient was injected again. And finally, the eye was lost, completely lost. And we saw this patient afterward. But this is our mistake. I mean, you must be very careful about these kind of reactions, and we should modify your therapeutic approach. Vas?
I think Barbara was
I
trying
was
to
going
say
to,
something.
no, that's all right. I was going to comment, something that was said earlier today was that the reporting system is not up to par, in my opinion, for inflammation, because the physician can report iritis, they can report anterior cell, they can report vitreous cell or vitritis, there's uveitis. There must be 5 ways to report inflammation.
Yeah.
And then it gets missed because percentage-wise, each of those 5 is quite low. I would also say that reading centers typically do report on excessive leakage, on optic disc staining, and vasculitis, but it is not part of the grading protocol. It's usually a comment for something that's happened, and that gets missed also. So I think there needs to be a lot of care during the phase 3 trial to look at truly what is the incidence of inflammation.
Thank you. I'd like to move now to, oh, sorry. You want to say something?
No, I was just going to ask you, Francesco, and maybe the experience in Italy, do you load the patient when you use brolucizumab? Because one of the things, as you know, is we were asking about, or talking about the risk factors. It seems like there's a higher rate, at least it was observed in the Merlin, when you treat more frequently.
Yeah.
And so I'm wondering, because I have to say, I've never saw a case of it, but I never treated a patient more frequently than every 8 weeks with
Absolutely.
brolucizumab.
Yeah, for sure. So, Giovanni,
just one thing.
I like to leave this session on time.
Yes, thank you.
For you.
No, but one of the things that we actually discover is that one of the very sensitive signs of inflammation is the enlargement of retinal vessels. It's something that we never pay attention, but it's absolutely unbelievable. You actually get a changing in size before any other terrible thing. So
I agree.
Please look at the retinal vessels.
Caliber of vessels. So I'd like to spend some word, really, we have 2 minutes maybe, about another important topic of this session, which is biosimilars. I think that we will face the biosimilars becoming more and more important in our practice. We are not familiar with that because we started to have some biosimilar in Italy since some months, but we did not have experience about that. So what I'd like to know is how do you foresee the future for us? I mean, how many of our injections will be made by biosimilar? I know that whenever you have a biosimilar entering the market in any field of medicine, it will become the blockbuster immediately. In few months, at least in our fields, biosimilars became the number 1. So what about the future for our field? Phil, Bas, whoever would like to speak about that. Phil Rosenfeld, there. Or Rick, sorry.
Yeah,
I think the transition from the inflammation issue to the biosimilars is very relevant because we have very reliable drugs right now. The Lucentis, the Eylea, the faricimab, the high-dose Eylea. I have no evidence that they're as good or better in terms of inflammation.
But financial reasons are very important for us. I mean,
But you need to understand, in the United States, it doesn't matter. That's the weird thing about the U.S. system, is that unless an HMO or a private insurer dictates that you use one drug versus the other, Medicare is agnostic to prices.
in Italy is the opposite because we are obliged to use the less expensive drug by our administrators. So we are pushed in the direction of biosimilars. And before we are pushed in the direction of Avastin, we have a lot of regions where Avastin is the first drug to be used. You are not allowed to use the others for financial reasons only.
That's the major issue with the U.S. system, that drug prices are non-negotiable when it comes to Medicare.
You want to say something?
Yes, because in Italy, we have the patient that know by the media the situation, and they ask us to do new drugs. Instead, we are obliged to use all
the less expensive.
Another problem.
I have a question about advertising. Is advertising permitted on commercial TV in Italy?
No.
No.
See, this is one of the great travesties in the United States, that years ago, advertising for branded drugs was allowed, and our television shows are filled with these ads.
You want to say something? You want to add something?
Just to add that the situation is very dynamic, because as biosimilar come onto the market, the incumbent oftentimes would also go into a phase of price adjustment. So oftentimes it depends on the payer, how do they actually work with the drug company.
I'd like to have some advice from Dr. Ferris about this. How do you see the future for biosimilars in our ophthalmology field?
So I agree with Phil that we have a very strange system in the United States, but as in Italy and in Great Britain and other places, inevitably, if a drug is shown to be as effective, there is a move to use that drug. And generics, I take all sorts of generics now because although it is Medicare is agnostic, the generics in all, I'm not talking about these biosimilars, but all biosimilars were being pushed in that direction, I think probably for good reason. And I wouldn't feel so badly about starting with Avastin. and now we're going to have other drugs that are similar. I mean, we've tested Avastin, and you always have the opportunity. We were talking about brolucizumab. We always have the opportunity of switching.
Please, there is there.
So just a comment from the UK perspective. So clearly we're very price sensitive, and the likelihood is we will be made to eventually default to biosimilars. But as Barbara said, the issue is how do we monitor inflammation? The trials are short, and the regulators say you have to have quite small trials to show efficacy, but quite small to show safety and inflammation, because they're biosimilars, they're not generics. And so we need to have rigorous monitoring systems within our countries, because we've got so many biosimilars coming through to detect inflammatory or unusual responses. So I think the direction of travel is we're going to default to biosimilars, at least in the UK market. And if there's non-response that you have to demonstrate, then you may be able to switch to alternative drugs. But I think we need to have more rigorous, standardized inflammation monitoring.
Glenn, do you want to say something?
You at least have a chance to do it. Great.
At the Reading Center, we've switched now for the reasons that Barb just mentioned, that when we do clinical trials now, even phase 1 and phase 2, we're changing the imaging that we do so that we get wide field even on the diabetes, on the diabetics and the AMD studies, so that it's easy for people to then, if they have any evidence of inflammation, or we see evidence, and we're actually now including them in our grading forms if we see
Yeah.
intraretinal or
Yeah, we
intravitreal
do as well.
hyperreflective foci, so we have changed the monitoring.
May I ask you, the Americans, how is the perception because we are talking about safety, but what about efficacy? I mean, this drug, these biosimilars, at best are similar to ranibizumab or aflibercept. So are you prepared to make a step backward in terms of efficacy? We are talking of extending the intervals and things.
So it's a great question. I don't want to speak for everyone because we're a pretty diverse group in terms of how we do things probably. But I will say that we use biosimilars in our practice at UCLA, Doheny. And so actually what the question that's recently come up, and maybe this is what you're getting at with this efficacy question, is the problem with having all these agents is by the time you step through them or you say, I'm going to use the less expensive drug and move on, is the game it may already been lost for the patient, right? So if you haven't achieved control, because usually if you're changing, there's probably a reason. You haven't controlled the disease adequately in some way or frequent or sufficient frequency, what have you. And the question is, by the time you go through these iterations, will the patient have suffered some irreversible compromise? So actually in our group recently, we've talked about only stocking maybe 2 medications that we might only use at present, HD, aflibercept, and faricimab. And then just have those 2, just because we want to shorten the escalation for fear that by the time we get around to it, it may already be game over.
Carl Chucky, you are not well listening to us. Do you have any opinion about this biosimilars use? And then Phil, and then it's closed.
Yeah, so I think the issue with biosimilar, I think you have to be really aware of is you have to, there's so many, but you have to be aware that, when the studies are done for approval, right, they're relatively very short. So it's really unclear.
6 months is enough usually, right?
Sometimes even less. And it really depends
3
on
months.
how much and the number of subjects that are being studied. So the safety profile of these biosimilars can be somewhat questionable, especially when you start to look at where they're being manufactured. So these biosimilars are very difficult, unlike drugs, the drugs that Dr. Ferris takes. The chemistry of these drugs is very straightforward. The production of these biosimilars is much more complicated. And if you start looking, I look very carefully at where these drugs are being made. And I'm not saying I'm going to not use them if they're made in a certain country or a certain country, but you have to be very aware of the potential safety profile of these
You
drugs.
don't think that the agencies are enough careful about this?
Again, if you look at the number of patients, they look at, remember, what is the FDA safety profile? Is roughly, where's Rick, 1% is their cutoff? So if it's above 1%, then they say there's a problem. But it can be below 1%, which could be significant, and it may not be detected in the BNA application.
Thank you. Phil, and then we close.
I was just going to say, I never introduce a new drug on a treatment-naive patient. I always go through my progression. I don't lose patients. It's just a question of how often we're going to inject. So if I'm going to introduce a biosimilar, it'll be on top of existing therapy. And if I can't get that prolongation, if I can't get greater durability, I'll just move on to the branded drug.
Thank you. Thank you so much. I think we are on time. Giovanni Abbas, Francesca. And I think we have till 2:50 for this coffee break. Thank you. Thank you. Thank you so much.
Sì, magari mettiamo.
Stanno già nei dove si rilascia, dopo vi lascio chiedi altra acqua nel caso
Ci serve
dovrebbero servire.
Perché non sono bevendo, devo dire la verità, cioè le ultime cose li porto quasi sempre puliti.
E lì, quindi siamo a posto. Il fatto che lì bisogna mettere un po' siamo a posto.
Partiamo?
Certo. Aspetta, dove sono? No, alle 15? No, questo. 50, 250, sì. Aspetta, 250, siamo già.
Dove sta Francesca? Aspettiamo che entra qualcuno.
Io lo vedo che se loro sono già qua vogliono visitare.
Francesca, iniziamo? Allora, quando dici tu iniziamo. Sì, sì. Andiamo? Stanno entrando. So, good afternoon to everybody. First of all, I would like to thank Francesca and Giovanni for this kind invitation, and I would like to bring the greetings for all Italian ophthalmologists because this meeting is under the patronage of Italian Society of Ophthalmological Science, which I have the honor to be the president, so welcome to everybody. So, the first lecture this afternoon is by Stanislao Rizzo, Artificial Intelligence in Retinal Detachment Surgery. So we don't need any more a surgeon.
Good afternoon. Thank you very much for this kind invitation. It's an honor, it's a pleasure to be here after 1,000 miles. Finally arrived. I'll talk about artificial intelligence, retinal attachment, and you'll say enough. But I like to talk about artificial intelligence because I think this will be something will change our professional life, and not just our professional life. In a few years, you will have a phone in which you upload a photo of your patient. The phone will answer about the prognosis, about the surgical plan, about the results. So I divide my talk into chapters, into episodes. One chapter I discuss about the possibility of AI, in particular chatbots AI, to suggest you a surgical plan in vitreoretinal surgery for retinal detachment. And the second part of my talk will be about, is it possible to have a prediction of functional results in vitreoretinal surgery for retinal detachment? So up to now, we have more than 3,000 papers about the use of artificial intelligence in ophthalmology, but just a few papers for retinal detachment. And the first question I like to have with you is, do you think that a chatbot, you know what is a chatbot? That many of my residents know what is a chatbot. Chatbot is an interface, a software between you and the artificial intelligence, between you and the computer. We have many chatbots available up to now. We have open access application, completely free, not just for you, but also for your patient. This is more dangerous. And we observe two chatbots, the most famous, the most diffuse, ChatGPT, in particular ChatGPT-4, released last year, able to describe image. In Saturday, ChatGPT was released, ChatGPT-5, and Google Gemini. Google Gemini is able to access to all current web data that we have now on the internet. So our aim in the first part of the talk is to assess the capacity of AI chatbots in suggesting surgical planning for retinal detection case. The source were 50 cases operating in my hospital, 50 cases of retinal detachment surgery. And this is the page of ChatGPT's chatbots. You write the question. For example, we wrote a case of 70-year-old woman with disturbance for 3 days. She was already pseudophakic, and the fundus examination showed a macular on retinal detachment with a single short retinal trace at 11. And the question to chatbot is specify what kind of surgical plan you would suggest at the eventual intraocular tamponade. The chatbots answer it, and we submit this answer to a group of which expert vitreoretinal surgeon in order to see the agreement between this, the computer, the AI, and our vitreoretinal surgeons. I'd like to show you some case in order to explain better these methods. So this was a first case operated in my hospital 6 months before, underwent the scalar bulking for retinal detachment. She was phakic, and she had a recurrent inferior retinal detachment with PVR. So these were the answer of the two chatbots, ChatGPT and Gemini. Both the chatbots answered vitrectomy, but one answered ChatGPT membrane peeling. Gemini went directly to inferior retectomy. This may be difference of literature. Maybe the second were more Americans. The first one was more Europeans. No? Both retinopathy and laser coagulation, and just ChatGPT used silicone oil. This is more European, no, you know this. So, second case, pseudophakic, macular retinal detachment, single short retinal break 11 o'clock hours in middle periphery. The answer were vitrectomy in both cases, fortunately. No, but they don't not maybe didn't know primary accuracy. Lot of autocoagulation, gas tamponade. So the results were similar. At the end of the 50 case, we observed 84% of agreement between the chatbots and the group of retinal surgeons. 84%. So maybe in a few years they can suggest also how can we our behavior in sense. And maybe, but chatbot GPT is the creator of Elon Musk, from OpenAI, Elon Musk. So you know the origin of this ChatGPT. What is the potential? If in ChatGPT-5, if you submit a photo, a retinal detachment, or the patient submits a photo of retinal detachment, he will have the answer. And maybe he can perceive you, because why you do not apply the vitrectomy instead of scalar blocking? Because ChatGPT suggests me they have to receive vitrectomy. This is very, very dangerous, because it's open access. So we published this paper a few weeks ago on a British journal, but we are other papers. For example, we have papers about prediction on anatomical assessment, and this was the group of Fang and Williamson from UK. We have also a prediction of late recurrence. So this was a group of George Caputo in Paris. But up to now, we don't have any paper about the prediction of functional results. We already observed some biomarkers. We published these biomarkers. We observe as a prediction of low visual acuity after retinal detachment surgery, subretinal hyperactive foci, outretinal corrugation, and intraretinal cystic space. And we saw that subretinal hyperactive foci is the worst purposely visual biomarkers. It may be therefore be considered a negative predictor. So the study is assess feasibility of segmentation of CT biomarkers in the image retinal attachment to predict with this automatic, with this deep learning structure, per proposal of the visual acuity using the biomarkers we already identified. Our study population were of course, macular retinal attachment with good quality of the image OCT, with follow-up of at least 6 months. We divided our population into groups. One group was low visual acuity group with possible best quality visual acuity more than 0.4 logMAR, and visual acuity restored with possible best quality visual acuity less than 0.4 logMAR. So this is the aspect OCT segment. So we saw we scan with preoperative 6x6 OCT scan crossing the fovea. And all this image, with this image, we trained the image processing of MATLAB. MATLAB is the open access software, is the deep learning software available for everybody. We trained with 50% of the image, first, of course, with manual labeling, and then tested with the remaining 50%. And we saw excellent result. As you can see that the conic kappa in all the preoperative factors were above 0.75. And the results were equal between the human graders and the automatic segmenters. But the true difference is that, of course, AI shows not just comparable segmentation accuracy, but with very fast, in real time, predicted time. So in 88 eyes, so low visual acuity group, present the high response in fovea flattening, in easy damage, ELM damage, and ONL cyst, and ONL hyperactive foci. So the only biomarkers that didn't reach statistical significance was outer retinal corrugation and outer nuclear layer thickness. So we can predict now what? Our patient can have more or less than 0.4 logMAR. This is the OCT of property OCT of my patient with macular retinal attachment. We submit to the MATLAB, and this MATLAB segment automatically. In about 90%, we have the probability the patient will have the answer. In this case, we'll have a low visual acuity. I'll show you in real time what does it mean. This is the page of MATLAB. So, of course, we have to upload the image. This is real-time video. This was the image of OCT in the preoperative. Then, of course, I write the command, segment the image. The image is segmented in a few seconds. And then I write the command, classify the patient. And after a few seconds, the computer gives me the answer. This patient will have less than 0.4 logMAR. This is real time, in few seconds. But not only the surgeon can do this, but also the patient. In conclusion, OCT imaging predictors post-operative visual acuity can be effectively segmented on post-operative OCT image using AI. The segmented biomarkers can predict post-operative visual acuity more than 0.5 logMAR with around 90% accuracy. This is the first scene of 2001 Space Odyssey. It was more than 50 years ago. It was a visionary, it was Stanley Kubrick. I think we're informed in something that will change our species. Thank you very much for your attention.
Stano, congratulations. I think there are several issues which might be raised in respect of your presentation. First of all, obviously, this is only a tomographic assessment. So were the results or were the prediction irrespective of the procedure used, whether scleral buckling, vitrectomy, or pneumatic?
No, absolutely not, because of course this was a preoperative observation. No, afterward. There was a complete, but all the patients received vitrectomy.
So it was vitrectomy?
Yes, just vitrectomy, not about this bias about different procedures. vitrectomy gas.
Was time course of the detachment?
No,
also
absolutely not.
calculated?
Absolutely
No.
not.
It was irrespective of the duration of the detachment?
Yes.
Also. The subject?
Was
Surgeon,
just
the surgeon.
one surgeon. Yes.
Yes.
The best. Of course.
What else? What else?
But why in one case there was only a break in superior quadrants? I remember probably it was phakic. Why vitrectomy?
No, it was completely two different papers. The first one, we interviewed the chatbots with submitting the case, but we, of course, the second part of the case is completely different. We observe the property of biomarkers in order to do it, but completely different cases.
So this is quite challenging, actually, because if this procedure were to be assessed also by the patient themselves, they could pretend this sort of outcome, and really it's worrisome to some extent. Any question or observation? If this is not the case, thank you. We'll go on with Grazia Pertile talking on use of amniotic membrane in pediatric and adult vitreoretinal surgery, when you need to patch it up.
Thank you very much for the kind invitation. In the next couple of minutes, I will share with you my experience with amniotic membrane and what you can do and where you can use it. There was a recent study that showed a number of properties of amniotic membrane. It is, of course, easy to get. You can preserve it, the lyophilized membrane. You can keep it in the OR, ready for use. There is no immunological reaction. It has antimicrobial activity and anti-fibroblastic activity. And it promotes cell migration and growth. And in a recent study, they showed all these properties. Today, in fact, I will focus on two possibilities of the amniotic membrane to be used as a patch and its anti-fibrotic usage. First of all, I'd like to acknowledge Stano Rizzo, the first proposed the membrane to be used for recurrent or persistent large macular holes. The initial use was the subretinal space. And to be honest, I was not very happy with this position because the membrane is pretty rigid. And to fit it underneath, you had to undermine the edges. And I thought it was possibly dangerous for the visual acuity. And so I started using on top of it. This is a case of only eye, high myopic patient. She underwent first a vitrectomy with ILM flap technique, and then with a free flap, and the eye was, the hole remained open. And so we decided to use a bubble of PFO and then to bring in the amniotic membrane, and you can use an additional perfluorocarbon and just glide it over the hole. And with the fluid air exchange, it is important to dry completely the interface and the liquid between the perfluorocarbon liquid and the air to avoid slippage of the membrane. And this is the postoperative result. After a couple of months, the visual acuity improved to 0.4, and you see there is a thin layer. This is the advantage of the lyophilized amniotic membrane, that it is thinner, and there is a nice closure underneath. In some cases, although you really have impression that you need to replace some retina because there is a shortage. This is a case that was operated on in 2015. The macular hole closed nicely, and recently she had a reopening, and you can notice how steep this layer is due to the high myopia, posterior laser treatment, and this layer is really, really short. And you can notice the condition when I tried to use perfluorocarbon liquid inside to move the fluid anteriorly. You see the very posterior laser, and immediately to the strong traction, the perfluorocarbon migrate under the retina. So we decided that it was not enough to put something on top, but we decided to put a layer underneath and to make a kind of sandwich, and then a layer on top to elongate somehow the stiff retina and to close this hole. Of course, there are atrophic changes, but this is the only eye of the patient, and even the recovery of some visual field was important. So you can use also with both layers. Sometimes you can replace the tissues in colobomas. You see how you can get a disc of amniotic membrane. If you stain it with blue, it is easier to see where you place it and how you move it. And if you use 27G forceps, it's easy to introduce inside. And so in this way, you can place this disc of amniotic membrane on top of the breaks on this very thin retina inside the coloboma. Another typical use is when you have a recurrent optic pit. In these cases, you can introduce gently this very soft membrane inside the pit, and I can guarantee you that you can really place a lot of membrane in case of large holes, and so you can close. Of course, you use this technique only in recurrent macular holes, in a recurrent retinal detachment to optic pit. This is another case, also a boy with, this is the only eye bilateral malformation of the globe, and you see the large break here at the edge of this kind of morning glory syndrome. And apart from an accurate delamination of the epiretinal traction of epiretinal tissue, you can fit some amniotic membrane inside this hole at the edge of the pit, and it works nicely to close it. And because in that position, you cannot do any laser or other kind of retinopexy. But the most interesting use of amniotic membrane, in my opinion, is for its anti-fibrotic properties. We have here a case of a trauma with a full thickness perforation. So the foreign body was full thickness inside the posterior sclera. And in these cases, you have a problem that if you remove the foreign body, you have a posterior break, and BSS is going to migrate in the retrobulbar space, pushes the eye, and so you have a loss of pressure from one side and a pressure behind the eye. So also in this situation, I thought that it was a good idea to have ready some amniotic membrane. You see it can be introduced. This is fresh amniotic membrane, by the way. And it was prepared ready inside the eye, so ready to be introduced into the break as soon as the foreign body was removed. And if you pay attention, you can see how it is sucked into the, you see, all of a sudden it is sucked inside the hole. So you can fit it, and then the foreign body is quietly removed after sealing this break. And then you can trim the exceeding part of the membrane with a cutter. And as I said, the most interesting thing is that when you have usually such a posterior break, you have fibrotic ingrowth, and the retina tends to be pulled inside the break. While if you close it with amniotic membrane, you see the postoperative condition. It is very quiet. It seems really to create a kind of Brooks membrane and a separation, and it is not pulled into the break. And in this, in the next example, it is even more evident how it is. This is a case, 21 years old male, blunt trauma, retina attached, but you can see there is a large break in exposure of the sclera in this position. And this was the autofluorescence picture and the traction. So you see the macula is really pulled toward this break with subretinal and pre-retinal fibrotic tissue causing an ectopia of the macula and drop of visual acuity to 0.2 despite being attached retina. So I was really in trouble. I didn't know if I had to suggest anything or do nothing on an attached retina. And after discussing with the young man and his parents the possibility, decided to give it a try with the amniotic membrane. And so you see the condition, how much fibrosis there is. Let's see if video, is it possible to start the video? No, if not, we go, okay. Just to have an idea, so you start removing the vitreous, and then you see how thick this fibrotic tissue is on top of the retina, and the retina is incarcerated into these breaks. And it was really see from the blood vessels that it is completely incarcerated. And after a cleaning, an amniotic membrane was placed underneath and peeling of the eye lamp, and the amniotic membrane was put inside and fitted on top of the retina. And we will go to the postoperative condition. You see, after 3 months, the visual acuity was 0.6. And after 1 year, completely stable, and the macula in place, and no dragging or fibrotic recurrences. Thank you very much for your attention.
Thank you, grazie. Amazing surgeries. I've got a couple of questions. First of all, the sizing of your membrane they look quite oversized as respected to the area to be patched.
Yeah, it is, especially when you put it on top, because in my opinion, it is important to have it quite large because it is more stable. It acts like a contact lens, the opposite of a contact lens, and also because you can have some postoperative slippage. So if you have a hole, you position a precise size on top, you can have some surprises in the postoperative period.
My second question is the one which really puzzles me most. Why does, and I also want to ask Stano his opinion, why, if it's, an epiretinal placement makes sense because it's patching a hole, why should an amniotic membrane positioned underneath the retina act as well in closing the holes? And what is your, why do you choose one way or the other to deal with these breaks?
You mean for breaks?
First of all, I would like to ask you, at least speculatively, otherwise, why should a membrane positioned underneath the retina, underneath the hole, act as a patch? Should be an inverse patching. And second, what is your way of deciding why, if putting it over the retina or underneath the retina?
Then concerning the first question, I think that if you put it underneath, you close the hole from the inside. You have an elongation. Yeah, and so it remains attached to the outer retina, and it acts like a wall retina. I think this is the mechanism, and this is why in the case with a very large hole and a lot of traction, a shortenage of the retina, I was not sure that it would act like this, but it could remain due to the atrophy as well, attached to the RPE, and in that case I hadn't solved the problem, and this is why I put underneath and on top to create really an elongation of the retina.
Do you use only for larger hole?
For larger hole, yeah, it is for sure. Usually my first choice is for large hole, inverted ILM flap, and then if possible a free flap of ILM, and only if I...
You have experience with the platelet?
I tried. I think it is a very good system, probably in combination with somebody else. It works properly.
It is easy.
If it is easy, but it works. It is short term. So if you just need to patch it for a temporary closure and then the retina will seal, then it is okay. If you need a real patch and elongation of the retina, then you need something else, maybe in addition to platelets concentrate.
We are observing the evolution of the technique. We start with the subretinal space, and they're all closed, but the functional results were very poor. Now we change apretinal space. We change also the membrane, because now using the membrane because it's more transparent. It transmits 95% or less more than fresh membrane. But the last technique is to combine the subretinal amniotic membrane as a basal membrane, because it's a perfect basal membrane, because of course all the photoreceptors, real cells are coming up to the membrane. With autologous retinal transplant, because we need some more noble tissue. On the subretinal space, the amniotic, and on the top, autologous retinal transplant.
Although it is very nice, if you look at an OCT after a retinal patch, it is perfect. Sometimes you really cannot distinguish what is the transplant and what is the original retina. Although I'm convinced that there is no function. On OCT, as you put the retina on top of a functioning choroidal, and so the outer circulation, let me say, for the outer retina is immediately restored, it is function, but if you interrupt the inner circulation, you have no function. So I think it looks on OCT much better than what the functional results potentially can be.
Thank you, Grazia. Next and last talk of this session is by Matteo Ferreira on OCT and robotics in VR surgery.
So thank you so much, Professor Stavringhi, to invite me. This is my financial disclosure. We were co-investigator in this trial using a robotic surgery for VR, involving the Rotterdam Eye Hospital, Eindhoven University, and the PRECISE system. Our purpose for the study was to obtain OCT recordings of human retina during robotic VR surgery, so to evaluate the signal quality of the OCT itself, the characteristic of the scan, and to compare the image quality to preoperative standard OCT. The operation were performed at the Rotterdam Eye Hospital in 5 patients at the end of the standard surgical procedure. There were 2 surgeons. Me was one of the 2 surgeons. We use this suite, this robotic suite, that is a PRECISE suite. This is a telemanipulator robot. That it means that you control the single robotic arm using a remote control, and it follows your movement that is created to follow more or less the same movement you do during the normal surgery with your normal vitrectomy. And it's amazing how good it is. It's definitely better than me. I say the first time I use it, it's amazing how stable it is. The precise system was then acquired by Zeiss. We inserted in the eye this kind of instrument that was an OCT optic fiber that was put inside a needle that was mounted on the robot. The sensor consisted of a single fiber that was a focusing optics that produced an intensity profile of this shape. At the end of the standard surgery, we had to move the arm inside the eye, close to the retina, following these steps. So from above to upside, right to left, and then stop in a position and then nudge the probe to the retina and get away from the retina itself and try to find if we put a boundary limit to the robot, if it was able to stop the movement towards the retina itself. So this is a movie of the surgery. The robot is power on. I take control of the remote controller. And then you can start to cradle the system and start to use the robot. I don't know if there is the sound. You will hear that I'm getting closer and closer to the retina, nudging with the needle, and it's incredibly stable. It can stay at the distance we fit to the retina without any kind of movement.
1.2, 1.1, 1 millimeter, 0.9, 0.8.
This is continuous recording, and then I move back. So OCT signal was obtained from all the patients. The quality was assessed using the signal-to-noise ratio at the variation of the angle of the probe. There were no specific correlation between the signal-to-noise ratio and the inclination, because we used a very few inclination from 50 to 80 degrees. At the different distance of the probe from the retina, the signal-to-noise ratio remained very stable, using a helpful hint that is increased the averaging of the A scans and enabling the retinal detection up to the measurement windows of 3.2 millimeters. And then at different velocity of the probe, the fringe washout of the probe itself was 8.4 millimeters per second. That is quite fast. The instrument velocities are much lower in our ends, and more, the fringe washout is easily prevented by restricting robotically the velocities of the probe itself. So these are the quality of the images. As you see, closer to the retina, the images are better compared to where you move away from the retina itself, but the ILM and the RP remain constantly easy to detect. These purple line are the movement of the robot following the retina itself. And watching at the retina, we see a lot of blip, this kind of movement, and we analyze it. Using a mathematical extrapolation, we see that this retinal movement creating two harmonics. One have a frequencies of 56 BPM, that was heart rate of the patients. So the robot was able to follow the heart rate of the patients itself. And the other harmonics was the breathing of the patients. And those were the amplitude, 8 microns for the heartbeat and 5 to 12 microns for the breathing of the patients itself. Then we compare the intraoperative OCT to standard OCT. Standard OCT were acquired before surgery. Movies of the surgery were synchronized with OCT signal, and in post-production images from microscope were overlaid to the preoperative OCT scans, and then the scan lines were used to find the correspondence with the OCT obtained during the surgery. This is an example. And we were able to detect the inner retinal layers, the outer retinal layers, and the photoreceptor RP complex. And these are a couple of examples of the same measurement in the same location pre-op and during the surgery. And the mean difference was 16 microns compared the preoperative OCT to the intraoperative OCT. So in conclusion, OCT recordings of human retina definitely is feasible with a robot integrated distance sensor, can record movement as heart rate, breathing, and can easily follow the movement of the retina itself. And the OCT acquisition have a good quality with the Chernobyl retinal layers, and even we can approximate more or less the retinal thickness we have before the surgery using the standard OCT. So in future clinical applications are one we expect to have, so is plan and perform a subretinal delivery of gene therapy. Definitely, you can use a robot to stay there and inject for the time you want, precisely in the position you want, even controlled by the OCT, to plan and perform a very precise, very small tumor biopsy, I think, for some oncologists. And, for example, keep the vitreum at a safe distance from the retina, even if the retina is moving in a detached retina or something like that. Then the future is open to find a new solution for the robot. Thank you so much.
Thank you. Very impressive. My question is, I might have lost during the presentation. How quick is the feedback?
It's so fast. It's faster than our response.
So even if the patient moves suddenly, it's
there's an emergency exit. So you have a red button. You can push whatever it happens that really eject the robot from the eye of the patients. But the reaction of the robot itself, of the system itself, is faster than what we have normally during our surgical procedure. And that was an experimental setting using, I think, 3 or 4 different computers that makes even longer the reaction. In a commercial available setting, it will be definitely faster because there will be just one CPU, more than one CPU, but one that commands everything.
So you envision its use as a sort of a standard, also for standard procedure, not only for those you listed in the last slide.
In respect of the standard procedure, the problem with the single arm surgery, but probably is a problem with all the robotics arms, is that you have not a full degree angles of movement, but you have quite a cone of movement you can follow that is quite close to the temporal side, to the nasal side, and to the posterior pole. It's difficult to go in the far periphery, inferior, superior, and temporally nowadays.
So Grazia, feel safe. Your peripheral peeling still is needed. Okay, if there are no more questions, I think we are done.
Thank you.
Thank you.
So the last session, it's a Masquerade mini symposium, is actually a case presentation. So I ask to the panel, David, Bailey, Giuseppe, Glenn, to come with us. Please have a seat. And Marco and the...
Okay, so good afternoon, everyone.
I think there is another seat.
So, sorry for the title, Full the Expert. I really didn't like it.
I'm sorry.
Okay, so it's not working the clicker. Okay, so you probably noticed some weird titles, and these were given by a famous poet from Milan, which is Professor Invernizzi, which is not here today. Anyway, we'll do our best to discuss our cases, and let's start with the first one. It's a 70-year-old female with history of sudden decrease in vision right after cataract surgery was performed OS. So vision was hand motion, 2 millimeters of hypopyon, and here you have a fundus picture where you can appreciate definitely reduced opacities, some foci of possible retinitis and hemorrhages. And here's the corresponding OCT. So we have mainly intraretinal fluid, chororetinal folds, and diffuse retinal thickening corresponding to the area of wetting that you see in the fundus picture. So for this reason, we decided to proceed with via parsplana vitrectomy plus intravitreal injection of vancomycin and amikacin, and dismiss the patient the same day with the topical dexamethasone for 4 days. So we monitor the patient, and at 14 days, here's the result. And unfortunately, the cultures came back negative for both bacteria and fungi. So what to do? I mean, we decided just to follow up the patient, and the patient came back just 10 days later with floaters OD. So what do you suggest in this case? So what would you check?
What was the exam prior to cataract surgery? Was there a review?
I mean, cataract surgery was done with no complication.
No, but did you show us what the eye looked like before the surgery?
No. We just received the patient in the emergency department, and that was the pictures. No images before. Apparently, according to the surgeon, it was a normal fundus, similar to the right eye. So that's the reason why, when he saw in the private practice, he said, "Okay, that should be nephtomyelitis, so go to the emergency department of that hospital."
This was all subretinal choroidal or sub-RP stuff. Is that line in the there? Is that a photographic artifact, or is that like a giant Schlegel line?
We show you other slides. So this is the OCT.
I mean, the OCT that you showed us now almost looks like something that was like a resolved multifocal choroiditis or lymphoma, where there was maybe massive sub-RPE infiltration. Now you're telling us there's cells in the other eye. Could this have been lymphoma? You just partially treated it with whatever you injected, or multifocal choroiditis, you put steroids in.
So at first examination in our clinic, the vitreous opacities actually did not allow a reliable imaging of the entire fundus. From what we saw from the right eye, there were no sub-RP infiltrates or involvement like in multifocal choroiditis, at least in our first
The retinal
examination.
circulation was fine, right? So there was no vancomycin
Correct.
induced. It wasn't some toxic thing.
I have the
right?
angio to show you if you want.
So this was something going on probably before.
Yeah, I agree. That's a good point.
Yeah,
so
that's exactly what I was going to say.
It's quite slow in working.
Now that
I wanted to show you.
for FA and
Sorry,
ICG.
the fluorescein and ICG. So I go back. So definitely you have some vitreous involvement that may be secondary to inflammation or any other process. Fluorescein just shows some minor perivascular leakage in the late phases, especially in the temporal side, but ICG, I agree with you, does not show any significant changes that may indicate either occupying space lesion or infectious disorders or poor perfusion. And so we decided to investigate the patient since, perfectly agree with you, we consider this eye as affected by an ongoing situation right before the cataract surgery that probably was not diagnosed. So we prescribed to the patient a full set of blood tests, including also infectious and inflammatory disorders. But just 6 days later, the patient came back with another area of whitening that may be retinitis. But the problem is now that you have a decreasing vision in the good eye of the patient, and we advised to the patient to do a diagnostic vitrectomy, but the patient refused the surgery because that was the only eye. She was actually aware of the risk of the procedure, so how to behave in this situation. So
I was gonna ask Glenn what he's thinking right now.
Well, my first thought was to do a diagnostic vitrectomy, but she's not going to let you.
yeah,
do it.
I agree. The problem is that when we did not perform, I mean, we just did the cultural examination on the very first vitrectomy. Now definitely would be the right moment to do a diagnostic vitrectomy, also, looking for any malignancies. So the problem is that the patient refused any surgical approach. Here's the OCT. And so going back in 10 days and putting more attention to the autofluorescence, you can realize that at the level of the equatorial region, that definitely a significant change in the autofluorescence features that correspond to a continuous remodeling in the outer retina.
This is vitreoretinal lymphoma. I think Bailey was right. So I was a little bit confused because when I saw the initial picture on the left eye, I saw retinitis. And so I was thinking this could be a vancomycin-related retinitis, but then you injected vancomycin and it suddenly resolved. And the fact that it resolved, I think, that leads you more to a vitreoretinal lymphoma because these can just spontaneously improve.
Why
But
not
now,
syphilis?
What's that?
Why not syphilis or other infectious uveitis? I mean, these
No,
RP changes.
that appearance you had on the OCT was a retinitis, that diffuse hyperreflective change. So that was always my suspicion. And then I saw it again on the right eye with cells, and now you have the classic RPE changes. That we had 120 cases of OCT, cases of biopsy-proven vitreoretinal lymphoma, and these RPE detachments are the most common. That retinitis that you showed is very, very unusual. That's very atypical. So it's a masquerade syndrome. But now I think it's quite clear that this is a primary vitreoretinal lymphoma.
The attempt was to fool the experts. So
yeah, it is unusual. But what I would say is that the other conditions that we're talking about are not going to improve spontaneously or improve after vitreoretinal surgery. If it was syphilis, for instance, that's not going to get better with your standard treatment. And I've seen lymphomas get completely back to normal and then do exactly what you see here.
Okay, so good point. You have a very characteristic features of lymphoma now, but I'm just curious with all of these experts here, was there any clue aside from the fact that there was spontaneous improvement to suggest lymphoma early on, except for the fact that it's in the differential?
For me, it was the immediate OCT you showed. There was sub-RPE material. That's a very limited, number of entities that can do that.
But in the beginning, before you had the OCT, the features were atypical. You don't usually get a hypopion with a lymphoma.
And I agree that full thickness retinal involvement is rare, but it does definitely. We have 2 cases at Sloan Kettering.
For us, it was really interesting to observe how the situation was triggered by the cataract surgery, whereas in the literature, there are several reports where indeed the intraocular surgery may lead to improvement of the lymphoma. So probably everything runs so fast.
It's also a very fulminant case because in the 120 cases that we published, I never saw one case like this. They were usually multifocal deposits. The majority were drusenoid RPE detachments or subretinal bands, like the cloudy vitelliform lesions or the subretinal hyperreflectivity. We had maybe 10% where you could see intraretinal extension to the inner retina, but never a diffuse fulminant process like that. So the first thing you're thinking is a retinitis, but then you have to keep into consideration that retinitis can masquerade as lymphoma can masquerade as retinitis, but it's a very unusual case of vitreoretinal lymphoma.
I have a comment on the OCT picture. Yeah, just to say that it happened to be a couple of months ago, since we have in our clinic a subtracheal laser trial for drusen. I had a patient that was referred to me, was a young patient, like 52, 55, from after having seen 2 other retina specialists with this picture, and was referred to me for subtracheal laser. So think about this, that is mimicking a drusen or drusenoid PED. So let's think, especially that patient was quite easy to make a diagnosis because it was unilateral at just beginning in the other eye. And she was seen for the first time just for eyeglasses. And the physician saw this drusenoid appearance, and then it was vitreoretinal lymphoma.
Definitely, these are
so
rapid
I think for the audience is useful. Think about something different than just drusen, even if the age is almost the right age for drusen.
Yeah, pay attention also because it's an uncommon condition, but definitely the diagnosis, the prevalence is underestimated. And whenever you see this rapid turnover in terms of pseudo-drusen, you should think about this. So we concluded as a as presumed PVRL, since the patient indeed refused to perform the diagnostic vitrectomy. Always keep in mind primary vitreoretinal lymphoma as a possible differential diagnosis of any intraocular pathology and inflammation. And it's not so uncommon to see cases like this one, which is a cytologics-proved PVRL that may mimic indeed retinitis. So not always your retinitis is an infectious retinitis. Always consider lymphoma as one of your differential. And the role of multimodal imaging definitely has been increased over the last years. These are the recent consensus recommendations for diagnosis of PVRL. And you see that one of the main points is the role of ocular imaging, in particular of OCT features. And as Professor Saraf just mentioned, always consider which are the main possibilities. So to both study sub-RPE, subretinal, and both intraretinal deposit. So we decided to treat the patient with intravitreal methotrexate with the typical protocol, so twice a week for 4 weeks, and then slowly tapering. So this is the baseline at 2 weeks, at 8 weeks with a global, and I would say good restoration of the retinal anatomy. We lost the patient at this situation due to a great, to systemic sickness, and the patient was found to have later CNS lymphoma despite continuous MRI that were performed by the systemic oncologist. And it's good to observe how the intraretinal lesion actually resolved, improved after methotrexate, as well as the autofluorescence and the OCT pattern. Okay, so
I just want to add that I find also particularly useful the autofluorescence appearance.
Yeah, I think this is quite, it's definitely listed in your imaging tools, but I think this is even more misleading because that's not so common unless you don't see a really rapid, quick turnover from one visit to the other. So probably you should consider also this one in your differential. And if you're okay, we can move to just an imaging challenge. This is really quick, so any guess that's an early phase fluorescein angiography. And if you want, I can also give you a Sarengi lens wide field approach. Looks like you have really severe non-perfusion. There's a huge blockage on the temporal side that may indicate maybe intraretinal or subretinal bleeding.
Any suggestions?
Is it definitely an eye?
Oh yeah, that's an eye, I agree. And it's a human eye.
Human eye?
That's the second.
Does the patient have like an Argus 2 implant or something?
I give you other hints. So early and late frames.
Is there a hemorrhage there, do you say?
I mean, it's really, hypofluorescent. So there's definitely blockage over there from the surrounding background fluorescence. So that may be, I don't know, bleeding or maybe.
Is it an ILM hemorrhage that you did YAG laser and that the blood released afterwards? No.
That's a good point. I mean, I would be quite worried if it was the results of my laser, but no, it's not an ILM laser. I didn't consider this option. That's a good idea. For next time, I will show the case.
I think you may have fooled the experts on this one.
At least with one case, please.
I don't even know what this modality
Okay, I can
is.
give you, if you want, the history of the patient. So that's a 17-month male with history of drifted OD. So visual acuity was fix and follow OU. Unremarkable systemic or ocular history. Can give you. I mean, next slide is the solution of the case. I cannot move so fast. No, okay.
I think they're cool.
Okay, so this is the anterior segment photography where you see the Mittendorf dot. You have the tunica vasculosa lentis, and you have the persistent papillary membrane. So let's move to the fundus. And here you have your normal retina with the hyalid artery. And again, so this was a PFE.
Yeah.
So we made a mistake. We asked, is it an eye? We should ask, is it the retina? That's the mistake we made.
Okay, so let's move to more regular cases. So that's 48 years old Italian male with a history of decreased BCVA OD and headache. He went to the emergency department just saying that he came back from
Sorry,
safari.
Marco, but if it was a Belgian, it should be different.
I don't know. We should ask Alessandro. He was so, strictly saying, please say he was Italian. So I'm just replying, it's Italian. I don't know if it makes any difference, your systemic background. So vision was the 2025 ODIV and 2020 OS. So a remarkable anterior segment and a normal IOP. So this is the montage, fundus photography. Any comments? So definitely we have chororetinal folds. We have optic nerve swelling OD, which is definitely more severe of OS. There's something on the superior pole of the disc. And you may appreciate a subretinal bleeding just nasally to the disc. Yeah, absolutely. So let's take a look at fluorescein and ICG. So I would say pretty much unremarkable, the fluorescein angiography. You definitely appreciate better the folds in ICG. Late frames where you have these errors of hyperfluorescence that may just indicate, I don't know, chronic CSCR. OS. Any comment? We can move. I think this is a good picture to get something. In particular, ICGA has quite an interesting pattern with this filiform area of hyperfluorescence in correspondence of the choroidal vasculature.
I mean, if you're showing something weird, those could be tracks of some type.
What did you say, Billy?
I said those could be tracks of some parasite if it's something weird.
That's a good point. So OS has a similar ICG pattern in the extreme temporal side. And here's the OCT. So do you think it's helpful for your diagnosis? So we're talking about masquerades. So definitely one of our main differentials should be into inflammatory disorders. There's not CME indeed in OD.
No, the choroid's thick for sure.
Yeah, the choroid is definitely, I mean, it looks quite thicker.
But you can see the vascular pattern of the choroid. I mean, I don't know that the choroid is infiltrated, but it is thick, as Glenn says.
Yeah, it's not infiltrated. I agree with you because the anatomy of the choroid is preserved, but it's definitely thicker.
Is he on any medications like topramate or something like that?
No systemic medication.
And it's hard to tell here, but there were no inflammatory cells in the vitreous.
I agree with you. Also, the vitreous on B scan was okay. So there are several differential, but probably if I just saw this picture, posterior scleritis could be helpful, but no pain complained by the patient.
The patient had no pain in his eye?
No pain. Just painless, decreasing vision, OD, and headache.
And it was in a good health? There was no hypertension, no...
That's a good point. It was not yet under therapy for hypertension.
I've seen cases of VKH look just like this.
Yeah, so we actually focus mainly on the ICGA pattern with these errors of hyperfluorescence and the choroidal vasculature, and there are indeed several papers.
But you didn't really show any chronic, hypertensive features. Certainly no Malrick wedge, no Elschnig spots. Really.
You're right, but it's also true this is another case where we know that you may have also sudden onset of hypertension that may be, despite being uncommonly, also pretty much asymmetric with just some features. Anyway, the patient was just put under systemic medication for hypertension, and this is the follow-up at 1 month. So no other procedures or medication were done.
So this is acute hypertensive choroidopathy.
Yeah.
I can't tell you we see many of these patients yet. we are still seeing, I mean, in my practice in our clinic, we still see many patients like this.
We don't have
So
so many. I mean, it's. I think.
I mean,
it's that's one
relatively
of the
many.
few-
I think it's should be quite unusual to see patients like this in our
Definitely.
current era. But still, I see many. And we are those who are making the diagnosis of hypertension. So,
Any other thing to consider on this patient? Would you have also screened something different? Something more?
The other thing that it's quite strange is that if you look at the retinal vessel, you don't have hemorrhage, you don't have any
Absolutely, which is different from this
change,
case.
exactly what usually you see for hypertension.
How did you figure that out, Giuseppe? I have to say that was not the first thing that came to mind, just because you've seen other similar cases. Huh?
Right. And also the ICG is quite typical, that appearance that you showed in the ICG.
Yeah, I mean, when you see,
That is,
you know, hyperfluorescence on ICGA in correspondence of vascular structures, mainly you focus usually on like Kyrielase plaques, but it's on the retina, so you're not so used to observe a similar sign on a choroidal vasculature. So, actually, I didn't follow the patient from beginning to the end of the case, so I don't know if he had any other, you know, systemic cause like no hematologic reasons. I cannot tell you if he did any, you know, oncologic workup for other reasons for hypertension, but I have no other news. So the patient was admitted to the emergency department. We just saw him during one of the regular consultation for the OCT, but actually I didn't follow the patient on the last follow-up. And other case, so 44 years old man with a history of a decreased vision OD. So nothing in the previous medical or ocular history. The vision is definitely low, OD, so 2200 and 20/20 OS, normal anterior segment, normal IOP, and this is the fundus photograph of the right eye.
Again, Alessandro will present the international cases.
Yeah.
Okay.
You got it. He's always really, you know, strict in specifying the nationality of the patient. So, any comment on this picture? Definitely, we have massive vitreal opacities, OD, and something which is definitely less bad, but definitely going on also in OS on the supratemporal vascular arcade. And here's the corresponding OCT. So, no real evidence of CME or other signs of inflammation.
Is that not some focal vascular sheathing in the supratemporal arcade?
That's a good point. So let's move to the fluorescein and ECG. So it's unremarkable OS. OD is quite difficult to be, you know, recognized. But if you look at the periphery, nothing major. Also no FA, no vascular leakage, no hypoperfusion areas.
There were, something was on the inner retinal surface. Was it, what was the details of it? Was it?
I will give you details. I think that's
amyloid.
That's one of the key points. The surface. So anyway, the uveitis department decided to perform a full test, and probably they were, you know, quite struggled with the quantiferon. It came back positive, and they decided to put the patient under systemic steroids plus ATTT. So this is the B-scan OCT. Maybe there was some improvement OD, but the left eye definitely got worse. And if you check the OCT, do you think this is what you were looking for?
Yeah,
that's amyloid.
Amyloid.
Amyloid. So we should ask our colleague. We decided to do a diagnostic vitrectomy that came back negative for malignancies, but
Did you do a Congo red?
The problem is that the request for examination did not include at that time by the colleague study for amyloid. So the problem is that indeed the patient had later some complications related also to the surgery, so increased the uncontrolled IOP. were underwent surgery with the implant of hammer glaucoma valve. And these are the anterior swept source OCT from the back of the lens. And again, if you look better, just right posteriorly to the posterior capsule, you can see this typical deposit that, as you just mentioned, were so indicative for amyloidosis, and that were actually missed at the first examination. And also the OCT after the vitrectomy showed this spike, the level of the inner limiting membrane, which is one of the main signs, one of the main indicators for these pathologies that have been widely described as, like needle shape pattern. And so the patient accepted to do, again, a vitreous biopsy searching for amyloid, and actually was confirmed. So the patient was not aware about this situation, but was interesting to notice that also the brother was photocoagulated in another clinic as a possible, diabetic retinopathy, because he was considered to have vitreous bleeding. And when he came back during the OCT, we found the same deposit on the inner retinal surface. So
there weren't any choroidal changes, though. The ICGA was pretty normal. These patients can get a spectrum of choroidal changes that you see along the choroidal vessels, that I didn't see any of that.
Yeah, no. So but David, isn't there a difference between, this is between primary ocular versus systemic in
that's
terms
the
of
genetic, which is, I know, mainly, cardiologic involvement. I think you're talking, I remember we did some studies with Professor Pulido that was looking about the choroidal changes. That was, I think
it was in a Japanese paper, but I thought you could get it in both the genetic and the acquired type. So
we can go back, but in this patient, if you look at the ICG, does not show.
These definitely don't show.
Yeah,
they're quite characteristic. They're
I would consider normal.
Little comma-shaped changes along the choroidal vessels, and they can be a bit mild to very severe, and that's what I typically look for when I have a patient potentially with amyloidosis. The other thing you can look for with choroidal amyloid is a thickened choriocapillaris band, and you actually can see that in the left eye. It does look like the choriocapillaris band may be a little thicker.
Yeah,
so I wonder if there is, in addition to these vitreoretinal lesions, if there is evidence of choroidal amyloid in this case.
Yeah, that's a good pickup. If you go back for a second, Marco, yeah, that's a good pickup, David, especially on the nasal portion. That's where the
it does look very thick on the nasal. You don't see usually a choriocapillaris band.
Is it something that you consider definitely pathologic if you don't know the history of the patient, or on a regular basis you would consider like a normal?
Yeah, I don't know that I would look at the choriocapillaris band in all patients for this kind of finding. If there was indicators that you were looking for amyloid, then I would definitely want to look at that choriocapillaris band, and I think then you might be able to lean one way or the other.
Great. Any other comment, or we can move to other patient? So that's 67 years old male, history of the type 2 diabetes, and the patient was sent because not responding to anti-VEGF treatment. So visual acuity was 0.3 and 0.5, and some nuclear sclerosis, a normal IOP, and definitely you have some flame-shaped hemorrhages, plus some microaneurysms that indeed are more visible on fluorescein. I'm going to show you the fluorescein in a few seconds. And here's some CME and retinal fluid.
Yeah, I was wondering about that. Does this
I was afraid about this comment.
Waldenstrom's, or I was wondering about it with the left eye.
I put your CT so small just not to show you the features.
No, if you go back to if you go back to the color, I was wondering about Waldenstrom's or multiple myeloma, just because there's a serous detachment in the center.
Yeah,
that's a little bit atypical for diabetic macular edema. So when you see serous detachments, especially large squared out ones like this on the OCT, you have to think about a monoclonal gammopathy, and you would expect there's no leakage on the FA.
Absolutely. So the OCT pattern, I think, here was really, really helpful. Rather than being considered as cystoid spaces, it looks more like a schisis. Plus, I will show you the magnified image later.
Yeah, there you
definitely,
go.
yeah.
No
so the fluorescein, I understand the doubt of the referring doctor that actually considered the patient as a possible, diabetic retinopathy because he had a lot of microaneurysm. So probably this could be quite misleading on a regular basis on a regular clinic. We did also ICGA just to better document the case. You definitely appreciate the microaneurysm, plus you better see the vitreitis. So I think the good point of the muscular session is that definitely you usually consider hyperviscosity syndromes and this kind of schisis is one of the differentials for vascular disorders. But in this case, you have also vitreitis. So would you be concerned about vitreitis in a patient with Waldenstrom, or how would you move in this case?
The patient had Waldenstrom's, but you're also seeing some cell in the vitreous. Are you sure it's inflammatory? It's not maybe vitreous hemorrhage?
It could be. No hemorrhages at all, but definitely
you can also
could be
get
maybe some
protein
interest
aggregates.
involvement. Okay. So we did. These are some of the examination we prescribed, but definitely we also focus on hyperviscosity syndromes. I just wanted to show you first this slide just to show you that the patient had a really high erythrosedimentation ratio speed, but this is misleading. And again, was positive for quantiferon. But as you just mentioned, the OCT, when it came back just to give us the result of the systemic examination, was definitely so clear, so obvious for hyperviscosity syndromes, in particular when you move also nasally to the disc, you are able to appreciate a situation which is definitely different from what we observe on a regular basis on diabetic retinopathy, with the schisis going also beyond the nerve. And here's the typical fluorescein angiography where in the late phases, despite the presence of severe amount of fluid, you have a silent edema. So here's the main reason for semi-Sanniton FA, so vitreous macro attraction syndrome, retinitis pigmentosa, X-linked retinoschisis, and definitely in this case, hyperviscosity syndromes and leukemia. And here's the serum protein electrophoresis with a spike on the gamma globulin region. The patient was found to have a really high IgM level. And again, normochromic, normocytic anemia plus high ESRR. And the patient then underwent a bone marrow biopsy and was confirmed to have Waldenstrom disease.
The other thing is that the patient had diabetes, but if the M protein is very high, there could be a component of hyperviscosity, and that's going to exacerbate the retinopathy.
Perfectly agree.
I think his A1C was around 8, so presumably not so poorly. Was it 9? Oh, was it 10? All right, so maybe not. But
yeah, it was 10.8. So here we had a
10.8.
high HbA1c. We had the other misleading factor that could be somehow.
But maybe if you bring down that M protein with treatment,
lower.
you'll see an improvement not only in the macular
You will
edema, but
observe
also
it.
the
Yeah, once the
hemorrhages.
chemo is introduced, you will see a lowering of that parameter. So what about Waldenstrom? You're perfectly aware about this condition. I just want you to remember that actually this patient may show elevated ECRR, may show microangiopathy, may show pseudovitreitis, and also optic nerve swelling. So the patient underwent systemic chemo with an improvement of the gamma globulins and IgM, and at first, indeed, the OCT showed the first worsening, so that the patient later went under plasmapheresis, and the last follow-up, you see that there was a reduction on
Yeah, it can
this
take a
in the-
long time. That's
Yeah.
been my experience. I haven't seen a lot of these, maybe just a few over many years,
Or
but
it's
the
really
macular
slow.
edema, you have to get the monoclonal antibody down, and it can take many months or longer to get that edema controlled once you've reduced the elevated blood
What about
parameters.
adjuvant anti-VEGF or
No,
steroid
I don't think the
injection? You think it's
anti-VEGF is
helpful?
going to work if you don't get the M protein down.
We stopped the injection once we performed the systemic screening and just waited. Yeah, that's the last picture. It's interesting because it's a 12 years follow-up, so the patient now is
And
about...
This is also a very nice example or representation or indicator of fluid and how it is toxic to the outer
Yeah,
retina.
to retina.
And if you have chronic fluid, you're going to lose your outer retina and ellipsoid, and this really shows that very well.
I think it's striking because we had a similar case where the response to plasmapheresis indeed was faster, but he really had just in a few months an RP aperture and the retina just collapsed, giving again a sort of pseudo hole, and again a really bad systemic, visual acuity prognosis. Should we move on?
We'll have to be very, should we? Okay, we'll be very fast on this one. So far you have a 25% fool rate. So we can see if we can.
This is really easy, but I think it's really instructive. I wanted your opinion, indeed, on the management of this case.
The TFV is the only one.
PHPV?
PHPV. So, I mean, you're the expert. It was really, difficult to fool you. So, arterial hypertension, history of retinal detachment, OD, and floaters. And we know that the patient had in the same eye also unilateral glaucoma diagnosis plus CRVO. So the patient underwent intravitreal injection with the implant of desamethasone and anti-VEGF and was referred to our ocular oncology clinic for possible pigmented lesion, epipapillary. So this is the clinical examination and fundus picture.
Maybe we can hear from Adnan too, because we haven't.
Yes, so you've got multiple pigmented lesions at the back there, and systemically well, and pigmented lesions in the other eye?
No, in the other eye.
And no anterior chamber changes at all?
Not yet anterior changes, and that's a good point. And the other thing is that when we did the examination on the superior periphery, we saw this ciliary body mass. So as I mentioned, this is not such a challenging case, but I think it's interesting to see what happened because these are the OCTs. So you have some CME due to the CRVO. Definitely you have these retinal hyperreflective lesions which are not consistent with the diagnosis of uveal melanoma, but rather of possible retinal metastasis. That may be the same you see from cutaneous melanoma or from ciliary body or other tumors. So periphery. Fluorescein and ICG, I would say not helpful for your final diagnosis.
And do you have an OCT, sorry, the OCT through?
OCT, I mean, you see just, diffuse thickening of the inner retina, but you have so much masking on the outer layers that you're not able to appreciate if it's choroidal or hydroretinal. So I just move here. So on ultrasound, you see that the lesion indeed is mainly retinal rather than choroidal. And I want you to focus on this differential. So you just mentioned properly that when you have multifocal fundus pigmentation, there are several conditions you should rule out, and in particular metastases from cutaneous melanoma or other intraocular dissemination. And here is some, pigment at the level of trabecular meshwork that was unilateral. We did UBM, and you definitely can observe some ciliary body thickening. And when you do the scleral transillumination, you have what it looks like a possible ring shape and ciliary body melanoma. So our conclusion was that the patient indeed had already diffuse ciliary body melanoma, pigmentary glaucoma, CRVO, and then iatrogenic spreading of the lesion with intravitreal injection. So, yeah, these are some, typical images, histopathologic, but my question was, okay, any chance to avoid this when you have this case? I don't know. I mean, when you have a CRVO, I usually don't put any 3-mirror lens or go and check for the ciliary body. I just...
Well, when they have a CRVO, you're supposed to check for iris rubeosis, and check the angle rubeosis. So if you're being very disciplined, like I teach my residents, we should put the gonio lens on, but in a busy clinic, many of us don't.
You're right, but sometimes it's a posterior shape, ring shape. We saw the patient with already dissemination. Sometimes it's not so obvious in the anterior portion of the angle because actually the alterations mainly occur posteriorly. So anyway, pay attention when you inject a CRVO.
Great.
Okay.
Wonderful. Thank you.
Thank you for your attention.
Awesome. Thank you for the panel's expertise.
So, Russ,
Okay,
what do you think?
I guess I'll start. First of all, I think we had a phenomenal meeting. Thanks to all of the great participation from the audience, but also the great talks from our speakers. Thanks to our wonderful moderators for keeping things on time or helping us catch up as needed, because we're finishing on time after a remarkable day. Many thanks to Angela and the entire team at AVEC. I think they need a round of applause because they did a superb job. I'm just, as I said, we wish you all the best, and thank you very much to Francesca and to Giovanni for this great meeting.
And just a few words from my side. Again, I actually wanted to underline for the young people that, meetings are important for discussion. And so I think you have an experience here that the discussion it was the key issue of all the meetings. So I hope you will go back in your clinic, and, thinking that you have never give anything for granted, but think and try to verify if everything was told to you is correct. So thank you again, and
thank you very
Francesca?
much for the high level of your communication and contribution, and I was happy to have you here. Thank you again.
Thank you.
Meeting is now closed.